in India and globally, those who are joining us in this online. And the theme of this conference is immunology. So I am as much, as far from immunology as probably Donald Trump, and so I should be careful. Only difference between him and me is I should be more careful about making the statements about it based on the little bit of knowledge that I have got in this area. But at the beginning, I would like to tell something about my university and this particular project, which is very, very encouraging. First thing is, what is very important and encouraging is the scheme which Government of India introduced, which is called SPARC, the Scheme for Promotion of Academic and Research Collaboration. You might remember that we had a scheme, and we still have it, which is called GYAN. For last 10 years or so, India has started understanding strongly that one of the reasons why either the research work in India is not getting decimated, or we are not able to connect and do higher quality of work, is due to lack of proper collaboration. Because collaboration is the key to success in research world today. Once I remember, I met, when I was a Dean of Research at IIT Madras, I met Bruce Albert, the chief editor of Science magazine. And I asked him that we Indians are doing pretty well to go to 1B journals. But what is the problem that we are not figuring out in 1A journals like Nature or Science? So Bruce said that, well, the answer is not very simple, but one of the major reasons why you are not able to do that is that you do not know how to collaborate. You look at the papers in Nature or Science, most of them are collaborated by large number of people across institute, across continents. And this is exactly what we need to do. Fortunately, Government of India at one point of time felt it's important to invite the scholars from abroad and acquaint them to the Indian academia. That was done through the GYAN project. And the feedback of GYAN project induced them to introduce this project called SPARC, which is mostly aimed at developing collaborations, writing projects together, doing research together, student exchange, faculty exchange, which can really build up, very interesting collaborative work in every field of science and technology. Now, these projects, the SPARC projects, one of which is the project under which this current today's symposium is being held, these projects are extremely competitive. Something like 3 to 4% of the submitted projects only get funded. And if you look at even this Chandigarh region, there were only 6, from about 15, 20 institutions, only 6 projects have been accepted and funded. And out of these 6 projects, 4 are from IIT Ropar. Now, this tells something about IIT Ropar because as the director, that is the president of this institute, I take pride in the performance of my faculty members. By the by, the faculty members of this institution is pretty young. So, because the institute itself is very young, we are just 12 years old, and within 12 years, this is from 2 things. During last 1 week, it has been found out that we are one of the top 50 institutions in Asia. Now, this is great because if you look at Asia, all the institutes which are 100-year-old in places like Japan, Korea, China, Singapore, and so on. So being among them in the top 50 is something really great. Even yesterday, among the institutes of this country, which is about 9,000 in number, IIT Ropar has come out again within top 25, which is again another achievement, because if you really look at the achievement of the institutes in India, which are anything between 50 to 100 years old, a 12-year-old university getting into that is really very, very commendable and challenging. As I was saying, that one of the reasons that we could get into that position is the very young and enthusiastic faculty member. You may be surprised to know that the average age of faculty at IIT Ropar is only 38. And these youngsters are really fearless. In fact, during the recent COVID crisis itself, they have worked. I mean, we have got a faculty strength of nearly 180, and they have worked in 25 different areas related to the COVID crisis and brought out products and methods. At least 6 or 7 of them are under the process of commercialization. So it tells something about the energy and the enthusiasm in this university, particularly with respect to research. Even last year, in the Times Higher Education ranking, IIT Ropar came out between 300 and 350. But what is more important there, that in research citations, IIT Ropar got 100 out of 100. Which means it's not that only new things are being done, but the quality of research is one of the major concerns, because in our mission statement, we made it very clear that we want to contribute to knowledge and society, but we will only encourage high quality research. We are not looking at counting beans. We are not looking at how many publications a faculty member has made, but what kind of impact his research or her research is making. That's what the institute looks at. So as a result of this institute is being seen as one of the premier institutes today in this region of the country. And the SPARC projects being funded from the government of India, a large number, is one of the indicators of how this institution is doing. Believe me, I am not although I told about this international ranking, I am not a great fan of this ranking system, whichever it is, because I feel that university has got two aspects which it must address. One is the diversity, and other is this whole definition of what a university is. The term universe comes because it should be all pervading. A university should be something where the people should come from all over the world, where the ideas should be connected to all over the world. Probably the term university or the first university was something which developed in this country itself. Hundreds or thousands of years ago, we had Takshashila and Nalanda universities in India, which showed path to the world, what a university is, where people used to come all the way from Middle East to the Far East to get knowledge in various fields. So I think it's this all-pervading knowledge on one side and diversity on the other side, which is more important for a university, not so much the rank or the marks that it gets. And in that direction, IIT Ropar wants to make a very good contribution. Now, with this preamble, I would now, move a little bit to today's topic, which is immunology. Now, talking about immunology is, on one hand, difficult because that's not my subject. On the other hand, it's very easy because in this age of COVID, Everybody, a man in the street, you ask him something about immunology, he will talk about antibodies. He will talk about, there is no drug, so only our immunity is going to protect us and things like that. Because every day, now here comes the problem, because every day we hear a lot about immunology in the media, in the electronic media, in the print media, in the internet. But what is more dangerous is that half or maybe more than half of that is garbage. We hear all kinds of myths, we hear all kinds of unscientific things, and we start believing it. So it's very, very important today that we, the immunologists, really set the right perspective for the future work that we would like to do, not only in COVID, but in other areas also. Now, why this is important? Because if you look at the kind of expectation, on one hand, I would say that it has got a positive side, that the focus is turned for the whole community, the entire world community, on science, on scientific methods. Because everybody is understanding that if anyone, any community, or any method which can save, the humanity from this crisis, it is the scientist. So in that sense, the focus is on science, which is a very positive thing. But on the other hand, the problem is that the expectations are sometimes really, so high, and we need to convince the people. And these people means the common man, the media, and most importantly, the politicians who are policy makers, that a scientific outcome requires a scientific method. And scientific method takes its own time. I'll give you an example. Everybody in the media is talking about the vaccine for COVID is coming in 3 months or 6 months. Now, everybody, every one of us, if we look back, we know the history of vaccine. How many years did it take to come with the vaccine for measles? I think decades, maybe it's, nearly a century. How many years we have spent for finding out a vaccine for, SARS, other kinds of, viruses of this family itself. Centuries have gone to find out vaccine for flu. Have we got one today? No. The answer is absolutely no. About 30 to 40 years we have spent on the vaccine of AIDS, and it is still not there. We don't have a vaccine for malaria. So if you look at, in a large number of cases, we are not even able to get the vaccine after decades of research. In a number of cases where we could really get vaccine, is again very small, and in many cases we took decades to develop it. On the contrary, today's media and politicians are saying that we should get a vaccine within 6 months. Now, this is, we the scientists have to clearly, tell them that this is something which you are asking us to tell, going against, the rules of not only the, scientific method, but even ethical research. Because you see, these are not the days in the old, probably Nazi concentration camps, people could have put a vaccine and then infect the person deliberately with the disease. There was no ethical standards and nothing like that. But today we can't do that. We can't put the vaccine to a COVID vaccine to a person and deliberately infect him with COVID virus. This is ethically not acceptable. And hence, if we have to go to level 1 clinical trial, level 2 clinical trial, level 3 clinical trial, it takes time. Each trial takes 3, 4, even with international collaboration, we require 3 months, 6 months kind of thing. So if at all we can get a vaccine for COVID, if at all, because that if is a big if, still, I think it's at least 1.5 or 2 years away, even if we get it. So we have to tell this in unequivocal terms to the policymakers, politicians, media, that look, we are scientists, we follow a scientific method, and by that, this is what is possible and this is what is not possible. You know, I think all of you came across a very funny statement by a biochemical or microbiology researcher in Spain, when there was media interacting with her, asking about the vaccine development. She got so irritated. She said that you pay, millions of dollars per month to Messi and Cristiano Ronaldo. Why don't you go to them and ask them to save you? Why have you come to me, who gets hardly 2,000 euros a month for survival, and you ask me to rescue you? Now, that may be a little bitter on her part to tell, but the story is quite close to our heart, in the sense that I think, instead of finding out the heroes in a romantic world, the world has to look and find out the heroes in the laboratories. And the laboratories, I mean, not only the doctors or clinicians, also the laboratory researchers who bring out the real breakthrough, which is finally implemented by the medical practitioners. Immunology, in that sense, has got a much, much wider perspective, and immunology has got a very important future. Because if you look at the diseases that humankind has been able to really combat, are diseases like polio and smallpox. These are the two diseases everybody will say that we have been absolutely successful in completely exterminating from the world, and thanks to immunology that we have been able to do that. So the discipline of immunology, I think, more sophisticatedly will have to be developed, although the concept of immunology was there hundreds of years back. In fact, in 431 BC, Greek historian Thucydides, he said that the people in that war, they had a severe war, in that only those people should give services to the people who are infected with the epidemic of plague, those who are themselves infected by plague and recovered from it. So that an idea that only those who have recovered have developed an immunity, that kind of idea was there in prehistoric ages. But then the progress of this particular discipline was very closely linked with the progress of medicine itself. And slowly we developed, probably Jenner and Louis Pasteur can be told in the modern age the fathers of immunology in that sense. But it developed with that. But what gave a completely different turn is the discovery of the DNAs and RNA structures. And from there, today, as all of we know, the first thing that everybody demanded after this COVID broke up is the sequence of this virus, the genomic sequence. So our approach today is much more fundamental. But then, what is more important, that the people who teaches the immunology teaches medicine how to really combat such wide-scale infection. But the people who teach immunologists are the viruses and bacteria themselves, because these viruses and bacteria themselves change so quickly from one form to another. Today, even in India, they are talking of 3-4 different strains of the virus. And so it's very, very important that while we are looking for vaccine, we are looking for medicines to treat them, trying to use some existing medicine. There is big debate on remdesivir or the hydroxychloroquine and so on. But the fundamental research on immunology is extremely important, because we do not know these viruses, these bacteria, in what form they are going to come. And that's why it is important that institutes whether it is a small one or a big one, comes up and starts thinking that disciplines such as the biological disciplines, which are in usual terms quite far away from the focus of institutes like IIT Ropar, because IIT Ropar is a technological institute, and so what is important that even a technological institute focuses on the areas such as immunology, such as other biological areas in which different diseases from cancer to diabetes, and from epidemiology to immunology, we cover all of them with an idea to develop such disciplines. IIT Ropar set up about 7-8 years back a small center, and this is called the Center for Biomedical Engineering. But recently we have elevated this center as a department of medical technology, and there we would like to take up such challenges. Dr. Javed himself is a very well-known immunologist, and I sometimes tell that we have poached him from a nearby reputed microbiology institute, because I felt that he can be a person who can build up our biological research setup, and he is already showing that through this particular project on SPARC. Dr. Javed has already shown that he is at the forefront, and he is able to attract people from all over the world. And I really, I'm very, very hopeful that these collaborations will end up in very, very concrete research projects which will take IIT Ropar, this country, and the world towards better understanding of immunological perspective, right from its theoretical basics to its on-field applications. And IIT Ropar, on its side, I promise you, will be helping you with all its resources, by all means, to be successful in developing a very successful stream of immunological research. Thank you very much. I'm sure that I have provided very little which you didn't know, but I have been interested always in developing immunology as a very important stream of research at this institute, which will help not only this institute, but this entire region and world at large. Thank you very much. I hand it over to Javed for further proceedings.
So thank you very much, sir, for a nice introduction. And now we welcome Professor Peter Doherty. He has joined us. So Professor Peter Doherty does not need any introduction. He's a Nobel laureate. He got Nobel Prize in 1976, and we learn immunology through his discoveries. In fact, even today, I remember I used to read his papers, very classical papers, which showed MSC restrictions. So how he used to treat guinea pigs with anti-serum, removing antigen-presenting cells, and then adaptive transfer. He developed chimeras and showed that, yes, MSC molecules are responsible in imparting immune response. And the key role of cytotoxic T cells in killing targets. So he, with Rolf Zinkernagel, they developed very fundamental, very basics concepts of immunology. So we welcome you, sir.
Hi, are you hearing me okay?
Yes, we are hearing you.
Oh, good.
We welcome you. And let me tell you that about 2,700 people have registered for this conference.
Okay.
And right now, about 500 people, they are watching us. And we open this now question answer session with Professor Peter Doherty. So we welcome you.
Okay, good to be there. Where are you?
So we are in Punjab, India. This is
In the
at
Punjab.
the
Okay.
foothills of Himalayas, about 290 kilometers north of Delhi.
That must be a very beautiful place.
Very beautiful place, and we really like to welcome you over here. This is a new institute. Only 12 years back, this institute was established. So this is one of the series of IITs. IITs are quite renowned, very premier institute of the country. They produce engineers, but IITs, they have also entered now into biology.
Right.
So we welcome you, sir.
Thank you. So you're going to ask questions and I'm going to answer us.
Yeah, sure. So definitely, people will first like to know about your discovery of a Nobel Prize, your MSc restriction experiments, how cytotoxic T cells it kills, targets, and then, of course, people will like to hear about your connections because you have a lot of experience working in virus, and in current situation, we are facing SARS-CoV-2. So how it's ultimately going, how you want to address this. And
Yes,
I would also like to welcome Professor N.K. Ganguly. He's also with us. And probably Professor Peter Doherty may be knowing Professor N.K. Ganguly. And we are expecting Professor G.P. Talwar as well.
right.
So he may also be joining us. So you're
Professor
welcome,
Talwar,
sir.
I do know. Yes, yes. So,
Yes, right.
okay. Well, and are you having cases of COVID-19 in your region?
Very less.
Not many. Okay.
Yes.
But Delhi is quite badly hit.
Yes.
Yes, yes, yes. Okay, so if you want to answer questions, I'm happy to. Or do you want me to say something? You want me to talk about the discovery of MHC restriction?
Yes. If you could.
That was a very long time back, of course. 1973, we did the first experiments. I was at the Australian National University, the John Curtin School of Medical Research, after returning from doing a PhD in Edinburgh. I trained originally as a vet, so I'd been working in veterinary research. And this was my first experience of biomedical research. And we were looking at killer T cells. I was joined in the lab by Rolf Zinkernagel, who'd come out as a young medical graduate to also learn some immunology. And working together, we discovered that the killer T cell is in a virus infection model, lymphocytic choromeningitis virus, is focused onto the transplantation molecules of the cell of the syngeneic cells. That is, that we postulated that the transplant molecule was acting as altered self, that it had been in some way modified by the virus, and that the killer T cell was seeing the modified transplant molecule. Of course, a decade later, that was shown to be because there was a peptide from the virus inserted in the tip of the transplant molecule, and that was the nature of our altered self. That allowed us, the altered self argument allowed us to develop a kind of theory of how the cell-mediated immune system works, and to explain also what the transplant system is for and how it must have evolved. And as a consequence of that, we wrote some very short letters to nature. A letter to nature at that stage could only have about, I think it was about 1,500 words and 4 pieces of data. And of course, as it's way before the online era, we couldn't have any attachments or anything of that sort. And we also wrote a hypothesis article in The Lancet, which turned out to be substantially correct. And as a consequence of that, some 23 years later, I got a call, and when I was working in the United States, telling me that Zinkernagel and I had won the Nobel Prize for Medicine for 1996. And all that time, of course, we'd both been working on aspects of the cellular immune response, cell-mediated immunity, and virus infections. And that's been my career, really, an interest in virus infections, virus pathogenesis, and viral immunity. And of course, that translates into the type of situation that we're facing at the moment with SARS-CoV-2 and COVID-19. So my main activities at the moment are really acting on the public communications front for our institute with respect to COVID-19. As I'm almost 80 years old, I closed my lab a few years ago, and I've been mainly writing about science, and particularly trying to communicate with the general public about the nature of science and what it is and why it's important. And so this has been a logical extension of what I've been doing.
So now the forum is open for question answers.
Javed, could I start the ball rolling?
Yes, sir. So this is Professor N.K. Ganguly. He was earlier he was Director General of Indian Council of Medical Research. He was a health secretary.
And currently I am the chairman of Immunology Foundation, and I was the past president of Indian Society of Immunology. So I have a few basic questions, and I will ask them all at once so that it is easy to handle them. At this given moment, Professor Doherty, what is the basis of the protection from the COVID-19? Like neutralization antibody is taken as a correlate of protection, and many times the serologic tests which are conducted are taken as a passport or a surrogate of neutralization antibody, but we know that they are assayed in a different fashion. So do they truly, these are surrogate, or this doesn't really say that how we are protected from virus? And the if are the cytotoxic or cell-mediated immune systems are equally important from protection of COVID-19? And just to related question is this, that we also know that body's immune system starts attacking its own body during the infection at one stage of the time. Can we know that who will do that? Is it the virus genome or the host parasite relationship? So this is an interrelated first question. And my second question is this, that what is your take on the viral vaccines? There are nobody really did so much concerted effort in creating a new vaccine so early. We know that in AIDS virus, we couldn't get a vaccine till date. And latency and few other things were the problem. Here, is there a latency in COVID-19? And will we face the same issues as we faced in the AIDS virus vaccine development? So these are the two questions which I have from you, for you.
Well, thank you for those questions. They are, of course, very, very, very big questions. And to some extent, they're the questions that we're all asking because, as we all understand, we're at very early days still with the COVID-19 pandemic. We first received notification of the gene sequence from China on 9th of January, and it's now about 5 months later. So it's not really all that long. So with respect to the neutralizing antibody assay, firstly, my understanding is that we can do a neutralizing antibody assay. At the moment, we're doing it mainly in vero cells. The virus grows well in viros. It's being done largely by Kantar Subbarao, who runs the World Health Organization Collaborating Centre for Influenza at Melbourne, one of the 6 worldwide centres. And Kantar is, you may well know her, she's a very experienced and very smart virologist who was at the National Institutes health for many years. So the neutralizing antibody assay itself is robust. It works well. But of course, it does require a reasonable amount of serum. So that means we need to bleed people to get enough serum to do a good newt test. And that means, of course, that it's rather limiting in the sense of serological surveys. The data on whether newts are protective, I think not a lot so far. We'd like to see a lot more done with convalescent serum and so forth. But I understand that there is information that cocktails of monoclonal antibodies can be protective. And there's also experiments from Dan Baruch's lab in Harvard that shows that monkeys that were vaccinated or infected with SARS-CoV-2 give a pretty good level of protection. I think most of us are thinking that the neutralizing antibody is protective, but absolutely robust evidence. It will take time because we have the confounding factor when, of course, you've got an immune response, what other effects are going on. Surrogate assays for the neutralizing antibody assay, many of the blood pinprick, what we call point of care or lateral flow kits that came out very, very quickly, turned out to be not particularly useful, probably because of IgM, perhaps because of cross-reactivities. My sense is one of our groups has been doing rigorous evaluation of the lateral flow assays that have come from many different companies and has been finding that the IgG-based ones are best. Some working on a kind of competitive ELISA type principle seem to be the best correlates of neutralizing antibody. And I think we are now, as I understand it, getting to the point where we think some of those lateral flow kits might be useful for broader serological surveys, because there are still unanswered questions about background infection that really limit our understanding of this disease. I think some of those assays are coming through rather strongly, but I also do believe that the most robust assay, of course, is still the neutralizing assay. Then with respect to the issue of vaccines, the vaccines are moving very fast, as we know, and there are many different candidates in test at the moment, and a number already into human arms, which is pretty impressive considering we've only known the sequence of this virus for 5 months. And so we have, for instance, the Adeno-5 vectored vaccine from the Jenner Institute is fairly advanced. It may be the most advanced as far as I know. I think it's already in phase 2 trial in Britain. And as I understand it, a lot of that vaccine is being manufactured in India. There are DNA vaccines, mRNA vaccines, RNA vaccines, protein-based vaccines, a nanotech type protein approach, then also an adeno approach, killed virus vaccines for some of the Chinese researchers. And also we have a protein clamp vaccine in development here. The clamp idea being that it will maintain the morphology of the spike protein. I think CEPI, the Coalition for Epidemic Preparedness Initiative, I hope I've got that right, which has been running for about 2 years, primarily started by Gates, but with other supporters, has about 12 vaccines that it's been sponsoring over the last 2 years, including the one from the University of Queensland, the protein clamp vaccine, which gives very good antibody responses. They're seeing them through their testing. And of course, the mandate of CEPI is that whatever vaccines are developed under their framework, there is no patent involved, and those vaccines have to be globally available at the best possible price. That is basically more or less at cost. And of course, many vaccine companies have now committed to providing vaccine at cost. So production of vaccine, there's going to be a big issue. And of course, as we all understand from some earlier work from SARS-1 and from the MERS virus, we did get some immunopathology signals, or the investigators who were doing that got some immunopathology signals from some experiments that were done in rhesus macaques. And whether that's immune enhancement, as with dengue, or it's strict immunopathology is maybe not that clear. So far, I've not heard of any indication of that from any of the vaccine trials that are in progress right now, that have been through ferrets, mostly being the main test animal. Now, sometimes ACE2 transgenic mice. I haven't heard anything about immunopathology type effects. But of course, if we're going to give a vaccine to large numbers of humans, especially younger human beings who are not going to be severely affected in the main, we have to make sure that vaccine is absolutely safe. So that's why there's a certain slowness about the emergence of some of these vaccines. You know, if we were making this vaccine back in the time of polio virus, we probably already have it out there, but we've learned to be a lot more careful on that. My personal conviction is we can make a vaccine. There is no evidence to my mind that this virus goes latent or that it persists as an infectious virus. We are finding, there was a lot of confusion early on about the possibility that people who had tested positive with the PCR test, which, as we all know, is incredibly sensitive, that people who are testing positive with the PCR test and then went negative and then were coming positive again, there was concern they were being reinfected. I think now most of us are convinced that's actually there is some virus persistence at a very low level, probably. And most thinking is that unless someone is so sick that they have to go into hospital and they do develop severe disease, that their excretion of infectious virus will be over in about 14 days at the most. But we can still pick up PCR product for much longer at times, both in respiratory material, sputum, nasal swabs, and in gastrointestinal tract content. So there may be some hanging around a bit, but it's not like HIV. This is a fairly straightforward virus, I think, in that sense. It doesn't have a reverse transcriptase. It doesn't copy back into the genome, and it's not mutating at a very high rate. I mean, we do pick up mutants, and we can use those mutants to track particular lineages. For instance, we had an outbreak here that came from a cruise ship, but and we were able to track all the cases that came from that cruise ship due to some mutations in those viruses. But I don't think we need worry about it being like influenza or like HIV. The question of how robust immunity will be is another issue because we don't really know. We haven't had any long-term capacity to look at this thing in the long term for obvious reasons. But my personal conviction is we can make a vaccine against this. The other strategy, of course, is to develop antiviral drugs. And we're just starting to hear a bit more about that, and monoclonal antibodies too.
Thank you, Professor Doherty. This was a most comprehensive answer to my questions. I couldn't expect more, and it is amazing how you remembered all these intricate, interlinked questions and answered every piece of that. Thank you very much for that. Thank you.
Thank you very much.
So we welcome more questions.
Dr. Javed, can I ask a question?
Yeah, definitely. You can introduce yourself. Anybody who's asking a question can introduce.
I am Aman Sharma. I'm a professor of medicine looking after rheumatology services at PJ Chandigarh. The interlinked question to what Professor Ganguly asked is that although we are at the middle stage of the epidemic or pandemic in India, and we're starting to get more patients in Chandigarh, there has been a stark contrast in the severity or...
Can you be a little louder?
Different parts of the world. Can you hear me now?
Yeah.
There has been a significant difference in the severity and mortality of COVID-19 from different parts of the world. And there have been various theories floating around that the part of the world which is BCG vaccinated has a mild
We are not able to hear.
Can the other people hear me?
Yeah, yeah. No, I think there is a problem.
So the question is about severity in different parts of the world.
Yes, I think so.
Yeah. I'd say a little bit about that. I think, one of the problems we have with knowing, for instance, what the mortality is really knowing what people are reporting as a case. And if case reporting is only people who are hospitalized, for instance, or people who are diagnosed with symptoms, because in many cases, including in the United States, I think you can't really get tested in most places unless you have symptoms. Even then it can be difficult. On the other hand, we're testing large numbers of people who are completely normal in some what we call blitz surveys. And we've also been testing now anybody who has any respiratory symptoms. So I think the varying estimates of mortality rates are very much conflicted because of the differences in defining case. If we define a case as someone who tests positive for SARS-CoV-2, then of course the denominator goes way up and we get much lower incidence of mortality. I think from the Australian series, we got the disease late. We're only 25 million people in an enormous landmass, and we're an island. So we were able to shut down the country quite effectively. Everybody coming in from another country where they're potentially exposed is quarantined compulsorily for 2 weeks in a hotel under government supervision. They're tested 2 days after they come in and 2 days before they're let out of quarantine, and they're not released from quarantine if they're still positive. So we've kept our case numbers with a lot of community testing included in that. We've kept our case numbers down to about just over 7,000, and our deaths down up to just over 100. Now, the highest estimate across the country for fatality rate is 1.4%. In Victoria, the state where I am, it's 1%. So it's very low. And of the cases where the people we're testing in the community who are coming in with colds and flu-like symptoms, anyone who comes in with cold and flu-like symptoms can now be tested. Our positivity rate has been less than 0.2%. So it's a very, very low incidence. And I think around 1 to 1.4% might be around the actual mortality rate. But we all have questions about whether there may be some seasonal effect with this. We have questions about where there could be local effects, initially at least, and I'm not sure of the situation now. We were hearing very low incidence rates from countries like Cambodia and Vietnam, and Thailand, all of which I think have good public health systems. So I'm not sure what the status of that is. The countries that have had low incidence on the whole or have handled it very well are the East Asian countries that had the experience of SARS, Singapore, Taiwan, and South Korea, and so forth. But they have had spikes in incidence as well. So it's quite a confusing situation, I think. And also we're worried about a lot of failure of reporting, even in the United States, which is a bit chaotic at the moment for that country. We're seeing different things from different jurisdictions. And for instance, there have been instances where a lot of deaths in nursing homes were not reported. We had the same thing in the United Kingdom. So even in countries you would think had very advanced medical systems, we're getting all sorts of inconsistencies. So in the end analysis, I guess, it'll all wash out as excess death rates. Now, so that would be some time off in the future before we get those numbers. In talking for years about the 1918-19 flu pandemic, I was always a bit embarrassed that I would say when we think between 50 and 100 million people died, which of course is an enormous number in a world population that is probably a third the size of it is what it is today. But now I see why it was so difficult to come at a more precise measure, even in our times of modern communication.
Okay. May I ask a question?
Sure.
Yeah. See, as a complete layman in this area, I keep on hearing these theories that in countries like India, because of the BCG vaccine, we have got some kind of extra protection, the population in general. So what's your opinion?
Yes, of course. Yes, there is this phenomenon which is really quite strange, which is called trained immunity, I think, which has been around for a while now. I think it was first discovered in Britain. That BCG, which of course is the tuberculosis vaccine, which is a very big vaccine with an enormous amount of protein, but is a live vaccine, attenuated vaccine, that gives protection against a number of other infections. There's also evidence that live vaccines given to children, not kill vaccines, but live vaccines, can protect against other things. So we think that this is somehow, maybe through epigenetics, training elements of the adaptive or the innate response, rather not the adaptive response, training elements of the innate response to respond in ways that could help control an incoming pathogen like COVID, like the SARS-CoV-2. So I hope that's indeed true for India, because that would be wonderful. We're also wondering whether infection with other organisms that might be more common, say in Africa than in a country like Australia, whether those infections might be giving a measure of protection through the same sort of phenomenon. So at the moment, this is being taken very seriously, this BCG suggestion. We actually have a trial all ready to go in our healthcare workers, where one half of them will be vaccinated in one of the big hospitals, and a lot will be vaccinated, and others unvaccinated, of course, but we'll be vaccinating people in both the patient contact group and the non-contact group. Now, the problem, of course, is for doing a trial is that we don't have any cases, so that may not go ahead here. But we do have a big trial system called ASCOT, and we are interacting with the global network REMAP-CAP. And so it may be that some strategies that are developed here, including possibly antiviral drugs, we will be testing in other countries where there is a lot of disease incidence. But we indeed hope that this story about BCG and that other infections will be a factor. The other thing that, of course, is a factor that we're all wondering about is possible microbiome effects as well. And again, we don't understand any of that.
Professor Javed, may I ask one question? Hello. Yeah.
Yeah,
am I audible?
Yes, I can hear you. I can hear you.
Professor Doherty, in your opinion, in view of the...
You can introduce first. Dr. Sen, you introduce yourself.
I'm Pradeep from CSR Intech. And my question is that in view of the genetic polymorphism, how effective it would be if any vaccine would be coming up, would be useful universally to treat the COVID patient infection?
I think if we were looking at T-cell responses, I'd worry about that. And we do worry about that with our Australian Indigenous population with influenza, but with response to the infection, not the vaccine. But I think there can always be polymorphism effects in anything. But I think if we're going basically for neutralizing antibody, the general experience has been, I think, that the universal vaccines that work okay in the West work elsewhere. But the main problem being is if you've got a lot of background infection or malnutrition and so forth. So I'm not terribly concerned about it this time, though we don't know. One of the things that we have as an advantage in Australia is that we have quite a substantial population from India, Bangladesh, Pakistan, of immigrants. We have a lot of people from Asia, as well as the usual Caucasian types. And we do have quite active phase 1 and phase 2 clinical trial networks. And one of the attractions of our phase 1 and 2 clinical trial units, we may not be able to do phase 2, of course, we have no infection around, is that we can draw on a diverse population of ethnicity to make sure that we're not missing things for particular groups. So I think it's always something we have to keep in mind, and I think we should try and test in a spectrum of genetic backgrounds if we possibly can.
Dr. Javed, I was wondering if I can ask a question.
Yeah, definitely.
Good morning, Dr. Doherty. Myself, Dr. Pawan Gupta. I'm working at Imtech Chandigarh. My question is this, that there has been some wondering regarding the vulnerability of different age groups towards COVID-19. So one sees that it is largely the elderly who are vulnerable to COVID-19, but that is not the case in case of influenza. In case of influenza, you will see that both the elderly and the children, they happen to be vulnerable. So what could be what could be the reason which actually exempts the children from vulnerability to COVID-19? Could it be the polio vaccination program? Could it be the polio vaccination
We're all very curious about that question. Why is it that children aren't getting COVID-19 in any large numbers? Some do, and occasionally a small child dies, but it's very much an age profile that your risk starts to go up dramatically after age 60, and by age 80, I think the death rate is somewhere between 10 and 20% of people who are infected. So there are various speculations about why children should be less affected. One of them is that the receptor for this virus is the angiotensin converting enzyme 2 receptor, the ACE2 receptor. And one possibility is that the children are actually expressing less ACE2. That's one thing that's being discussed. Otherwise, I'm not really sure that we're clear at all about why children are much, much less susceptible. They can be severely involved. We've had some children developing a Kawasaki-like syndrome. And of course, that goes with the observations that now this virus is very different from influenza. We thought, when we first saw this disease, we thought it would be like influenza. We thought that, we would get massive pneumonia, people would be drowning in their own lung fluids and all that sort of thing. That doesn't really seem to be the case. I mean, what does seem to be causing a lot of deaths in this disease is basically anosmia, oxygen lack, and that's basically due to microclots in the alveolar circulation, and then other people are dying from strokes due to larger clots. So basically it's a clotting disease. And so what we're getting the sense now is that the SARS-CoV-2 virus is actually going systemic. Though we don't find it as a viremia, as you would with an arbovirus, we can find genetic signatures in the endothelium, in the heart, in the kidneys, and there's evidence of damage in all those sites. So we think the virus is actually, if it gets into blood, it's taken out very quickly by ACE2 expressing cells. And that's the reason for this pathogenesis. A lot of the discussion now on treatment is very focused on anticoagulation, even thrombolysis, as I understand from my clinical colleagues. I'm not a clinician, so I'm not very, I'm not seeing patients or any of that sort of thing. So it's a complex disease, more complex than influenza. But the children thing, it's being looked at, but I can't give you a better answer than I've given.
So my question regarding, in connection with the BCG question. So do you think it is BCG is imparting because of the immunomodulatory effect of BCG? And again, in context with India, we Indians, we are infected with many non-tuberculous mycobacteria.
Yes.
And if you are aware of one trial which was done for 10 years in Chingalpat, South India, where it was shown that BCG does not protect from tuberculosis. Rather, this is the environmental mycobacteria which protects. So is it BCG or our environmental mycobacteria which are doing this job against COVID?
Yeah, I would imagine it could be both. But what is your perception of the percent mortality or morbidity with this virus that you're seeing in India so far? Do you think it's lower than in other places or similar?
It's lower as far as the mortality is concerned.
Yes, so that could be a factor, I think. You may be relatively protected. They're having a terrible time in South America, of course, where you might expect some of the same conditions, but I don't know whether the South Americans use BCG. We've heard of very bad, obviously we know the situation in Brazil, which is very bad, also in Peru, I think, and some of those countries, but really, I don't know. And I think the BCG thing is probably this training type effect, I think, but it's been around for a while now and it's never really been worked out properly. It's difficult to work these things out, as we all know, because they're so complex. One thing that is obvious is that SARS-CoV-2, right at the beginning of the infection, is really turning off the interferon pathways, turning off type 1 and type 2 interferons. Now, there's just a study that's come out. It's using transgenic mice, which are actually using, you can't infect normal mice with this virus unless you modify the virus. But if you make a transgenic mouse with the ACE2, you can infect them. And some people are doing transient transduction experiments with adeno5 virus that has ACE2 in it. And if you blow that up their nose, then you will you can develop the infection. And one of the things that's been shown is that evidently type 1 interferons are actually protective in this type 1 and type 2 can be protective. So we think it's going around the interferon mechanism, and maybe treatment with interferons might be helpful. But of course, that's an expensive treatment, so it's difficult to roll out en masse, but you'd have to roll it out very early too, probably.
So there's a question.
Hi. Javed.
Okay, right.
Dr. Doherty, I was wondering that this lack of vulnerability of children to COVID-19, could it be by any wild hypothesis, by any permutation combination, be attributed to the prior polio vaccination being given to them? After all, both of them, they happen to be the positive strand RNA virus.
So what polio vaccine are you using in India?
We use both the attenuated one and the heat-killed one, depending on whether it is an intravenous or whether it is a...
So on that kind of BCG issue, you might expect maybe something with the live and not with the dead. I mean, it's with respect to children's vaccines, there is this story, which has come out of Africa mainly, I think, that the kill vaccines give you no protection or may even make things a bit worse, but the live vaccines do. So maybe there's something there. But also I think that maybe having a lot of background infection, particularly in the young, may be part of that. But it doesn't explain the situation in the Western countries where, again, children seem to be relatively protected. We're also, there are fair numbers of asymptomatics too in the adult population. We're not really too clear on how important they are as spreaders, whether they're putting out a lot of virus. And of course, if they're asymptomatic, they're not coughing and sneezing, which is where, obviously how we think most of this is transmitted.
Dr. Javed, can I ask a question?
Yes.
Yes, please.
Good morning. I'm Dr. Uma Khanga from AIMS, New Delhi. Thank you, Dr. Doherty, for the very nice discussion. And I just read some recent literature on the Chinese population indicating certain HLA alleles predisposing for SARS-CoV-2 infection. How much do you think that the MHC diversity in a population would influence the susceptibility to SARS-CoV-2 infection?
That's an interesting point, and I haven't seen that work. In general, MHC diversity doesn't have a big effect in infections. There's some signal with malaria, I think, again from the Jenner people, and there is.
We have with HIV.
And with HIV, yeah, a bit. There's also in our indigenous populations that lack A2, we think they may have a T-cell mediated immunity defect. And that may be common for a lot of indigenous populations who are normally very severely affected with the flu. But of course, India, as I understand it, HLA genetics in India is essentially Caucasian, isn't it?
Yes, and we are very diverse. We have a mix.
It's diverse, but not that different from Europeans. So, yeah, I don't know the answer to the question. It would not be surprising if it comes through. I mean, if you really want to get a good signal for MHC effects on infection, you have to go to chickens because they only have one. They only have one MHC locus.
Yeah.
So you can see very, very clear signals about MHC-related susceptibility in chickens. But of course, we have 2 or 3 functioning class 1 loci, so that's much more difficult to pick up. But would not be surprising, but it will take time for that to emerge, I suspect. And I'm not sure how much typing is being done.
I understand.
There's a question from a student. It's been checked, and Umakanga, can we give? There are a lot of questions, so
okay.
Umakanga take only one question at a time from a person. So there's a question that has been checked by probably a student. He's asking about the role of hydroxychloroquine in treating COVID patients. So what is your opinion about it, Professor Doherty?
Well, of course, in asking about hydroxychloroquine, we're sort of asking about drugs being repurposed. And the one drug that has come through so far that is an approved drug, a pharmaceutical, that actually looks useful is remdesivir, which is the antiviral nucleoside, I think, that was developed by the Gilead company for Ebola. And it looks as though that nucleoside may have, there's trial evidence that it is useful in SARS-CoV-2. The problem with remdesivir, it's very hard to get hold of. And at the moment, you don't expect to get much out of the United States with the present administration until their needs have been satisfied. We do have some, but not very much. And so far, there may be some signals that it is beneficial. Hydroxychloroquine, the anti-malarial, I think there's some intellectual reason for thinking it might work. I think there's been a lot of publicity around this and quite a bit of trial work, but not very good trial work, not properly controlled clinical trials. I think the general consensus might be that you don't want to use it late, that it's not much benefit then, and that when people are really ill, where there hasn't been any really good data is on using it early as a preventive or as a prophylactic or early in infection, though what has been done there hasn't shown much. I'm not personally a fan of hydroxychloroquine, and there is this latest data which shows that hydroxychloroquine may be influencing that BCG type effect in a negative way. So I'm kind of very dubious about it, and I'm looking forward to seeing a lot more antiviral drugs emerging. They're just starting to come through, I think, as the rational design guys and so forth get onto it. The nucleosides, I think, are fairly prominent in some of this. They can be developed fairly early, and of course, it's drugs that we use to handle HIV, both as a preventive in the HIV PrEP strategy and as a treatment. That's always the backup for a vaccine if vaccines fail drugs, but of course they're very expensive.
Well, there is another question. Yeah, please.
Professor Doherty, this is Dr. Naidu from IIT Roper. Could you please comment on possibilities of developing herd immunity against this virus?
Well, I think we've been thinking, or the epidemiologists have been arguing all along, that it has an R value of about 2.5. That is if you don't do anything to limit it by social distancing or anything. And that means one person will infect 2.5 other people. That means the disease ramps up very quickly and multiplies very quickly. With an R value of 2.5, the general sort of consensus is you would start to see real signs of herd immunity and a major drop-off in new cases and so forth when you get about 60% of your population infected. if it was higher, for instance, if it's like measles, you would see 90% before you're actually protecting people that are still vulnerable. So we don't know. Some countries may have reached that, but mostly or some regions may have reached it, but mostly the numbers we're getting out of places like Britain and so forth for different areas of the country suggest 10%, maybe 20% at the most of people have actually experienced the infection. So they're way off herd immunity. So with respect to herd immunity, I think the idea that herd immunity created by a vaccine rather than by natural infection is the way we want to go. So everyone is keen to get vaccines out there as fast as possible. But of course, there is this safety concern. So from what I can understand, the earliest we might see vaccines in widespread use would be late this year, early next year. The earliest, the Australian vaccine that we're discussing, the earliest we'd see that out there, I think, is mid next year. It's a protein-based vaccine, so it'll take more to produce. Vaccine production is going to be a problem too, getting enough vaccine made. That will depend on the nature of the vaccine. If it's a protein vaccine, obviously that's much more difficult than making a nucleic acid vaccine or something of that sort.
Dr. Javed, can I ask a question?
Yeah, sure.
This is Dr. Sharman Shahrawat from ISER, Mohali. So my question actually is about the kids not that's susceptible to the infection, or they don't develop severe disease. So could it be that they have actually a much more prominent type 1 interferon response in addition to the ACE2 receptor?
Possible. I don't know. I don't know if anyone's looked at it, quite frankly. I haven't seen anything on that. And of course, I guess there's not that many kids that are being looked at closely because they're not getting infected. Whether you can look at that in culture or organ culture, I don't know. It's an interesting possibility. One of the things that we're doing is, or that Cantor Subbarao's lab is doing in association with the Cancer Institute across the street, Peter MacCallum Cancer Institute, is looking at stem cell cultures, differentiated stem cell cultures of heart. And I think she's got a gastrointestinal epithelial culture. I guess you could try and generate those from individuals of different ages, but whether that would sustain under culture conditions, I don't know. I think, as always with kids, it's firstly that you don't want to be intrusive resampling, and then really the focus is not on them in this disease. So unlike flu, but we really don't know much about specifically about flu in kids, as far as I'm concerned, except of course they are major introducers of the disease into the household, which doesn't seem to be the case with SARS-CoV-2.
So the other question is actually on the vaccines. So because most of these vaccines actually are focusing on the spike protein.
Yes.
So could there be a problem with the escape variants emerging for the virus? Because there is only just one protein that a lot of people are focusing on, right?
Yes, I mean, of course, we're all concerned about the possibility, but this virus doesn't have the high mutation rates associated with influenza. It's a much bigger, it's a very big RNA virus. It's 30,000 base pairs. Flu is about 13,000, HIV is less than 10. So it doesn't have it does have a proofreading mechanism. They don't ask me to describe it. I'm not enough of a virologist. But it doesn't throw off mutants at a high rate. No suggestion yet of immune escape mutants that I know of, or of really virulence mutants. The only thing I've heard about so far, there was some suggestion that there might be a mutated form that was more infectious, but I've heard no more about it, and people were very dubious about the paper. So I really don't know the status of that. I'm not particularly concerned about antigenic escape mutants. And of course, if we did see them, say next year or later, if it persists, once we've got a vaccine strategy developed, it would be probably fairly easy to tweak the vaccine as we do with influenza and come out with a vaccine against the variant. But I'm personally not particularly concerned about them. And I don't think the community is at this stage until we actually see some sign. If you think of influenza, we've looked at influenza in kids that are persistently infected. If some children who don't have a functioning immune system can actually get a fairly persistent infection with influenza without developing severe symptoms. And in those kids, they are throwing off mutants at an incredible rate. So if you think about influenza, there are probably millions, if not billions, of mutants being thrown off each year, but only ever one gets selected as a mutation that actually comes through as a seasonal influenza. So there's a lot we don't understand about flu, but it does sort of tell me that you need and probably a high mutation rate to actually select these things out. Also, I wonder about selecting the mutations with such a specific functional protein. The receptor binding domain is very, 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 very targeted. The flu hemagglutinin binds to sialic acid. So that's a pretty ubiquitous sort of thing, but this thing is very targeted. So whether you would get, if it did mutate in the key antigenic site, whether you would get a good fit virus out, I wonder. So nobody's doing the experiments to try and force mutation. We don't want any lab escapes.
Yeah, sure.
So right now we end the question and answer session, and since because of the time constraint, and we would
I'll have one more question at most. Yeah.
Professor Doherty to release our monogram. It's a part of this conference and this project. So I'll put it on screen mode, and could you please release it? We'll be happy.
What am I supposed to do?
Yeah.
You only announce that I release it.
I believe it. Okay. I haven't seen it yet. And then I'll say goodbye because I have to go. That looks wonderful, and I wish you luck with it. I'm sure it must be full of extremely valuable information. So, is that what I'm supposed to say?
Yes. Yes.
It looks great.
Thank you.
Thank you for the conversation, and I hope all goes well with the disease in your part of the world.
So could you see this monogram?
Yes, I can. A monograph on immunotherapeutic strategies for autoimmune disease, edited by Professor Javed Agrawal, and I'm just trying to read Imitiaz Khan. So,
thank you very much. So we release it.
It is very green.
Right.
So I will say goodbye. Thank you for the
very much,
conversation.
Professor Doherty, for your time, and we appreciate your time and your valuable information about COVID-19 and other information. And we hope there were a lot of questions, at least 100 questions people have texted, and we hope that they can email you. And
please don't. I'm really too busy to answer too many questions. And I would be impolite, and I would hate to do that. So thank you very much, and I will say goodbye. I have to go to something else. So thank you.
Right. Thank you very much. Take care. Bye. So, actually, there are many questions, and we cannot take them since the shortage of time, because Professor Doherty, he has to leave. So now our second presentation, we're going to start, and this is by me. I'm Javed Agrawala, and right now I'm working at IIT Roper. And today I'm going to talk about something which is about my studies on immunosuppressive molecule. So, could you see my screen? Another one? देखना स्क्रीन अशु स्क्रीन देख रही है
Stop present now entire present now entire screen थोड़ा पीछे
नहीं आ रहा
नहीं उधर नहीं आ रहा ऑनर
Okay, so can you see my screen?
Yes, sir. Please make it full screen.
Okay, is it okay? Right. So today I'm going to speak about one molecule, which is cerulomycin, and it is immunosuppressive molecule. And why I have given this topic, Mere Desh Ki Dharti, Sona Ogle Ogle Hire Moti. This is in 1967, there was a movie, Upkar, by Manoj Kumar. And this is a song which I try to connect it, that there are a lot of valuable wealth in our dharti, which is our country, and in the mud of country. So I was encouraged to identify some molecule which may have immunosuppressive molecule. The reason was that if you see autoimmune diseases, in case of autoimmune diseases, it's largely growing, it's considerably increasing globally. And this autoimmune disease can affect, it can influence any part of the body. Okay, for example, it can affect brain, nervous system, which is multiple sclerosis. It can it can influence heart, myocarditis, pancreas, diabetes 1, type 1 diabetes, joints, rheumatoid arthritis, and multiple organs, which is systemic lupus erythematosus. So any part of body you think, and it can be affected by autoimmune diseases. But unfortunately, there is no treatment. Rather, we use as a preventive or secondary measures to suppress the autoimmune diseases. And the treatment for autoimmune diseases, these are anti-inflammatory drugs like corticosteroids or anti-rheumatoid drugs or immunosuppressive agents. And the biggest limitation of these side effects is this treatment is associated with side effects. And these drugs, they have a lot of severe side effects. And there are a lot of hunting going on to search different strategies to treat autoimmune diseases. So many radical changes have been made in treatment. For example, recently people have been trying to use regulatory T cells. Regulatory T cells, these are the most recent discovery in immunology about any cell. So the scientists have discovered regulatory T cells, and their role has been identified to suppress the immune system. So once the job has been done by the cells of the immune system and have cleared the pathogens, so now these cells which are activated, they needs to be brought into the normal stage, and this is the work done by regulatory T cells. Plus, regulatory T cells, they are also important in suppressing the autoreactive T cells. So although 99% of autoreactive T cells, they get deleted in our body, in our thymus, but very tiny percent of population can escape, and these are autoreactive T cells, and they can be very dangerous. They can play havoc in our body. And these cells now, they roam around in our body. So how these are suppressed? So there were many theories, and recently it's been observed that regulatory T cells can suppress these autoreactive T cells. So there are many side effects associated with currently existing therapies. And for example, you may know that what are the side effects, since it has been mentioned about 100,000 Americans are hospitalized every year because of side effects of ulcers and gastrointestinal bleeding by treatment with drugs to suppress autoimmunity. And as a result of which, about 10,000 people, they die because of drug-induced injuries. So the current therapies, they have very limited options. So therefore, this is a challenge for the scientific community and urgent need to develop alternative therapies. So what we observe that we thought, why not we explore and exploit different niches of a country. So what we did, we collected about 10,000 soil samples from different parts of the country, and we tried to develop, extracts of these. And if you see here, we went to as far as north of the country. This is Rohtang Pass near Himalayas, and we explored cold deserts, hot deserts, Western Ghats. So from east, north, south, west, everywhere the samples they were collected, and the soil samples were then bacteria were separated and they were grown, and we developed extracts. So about 1,000 extracts they were developed from the microorganisms which were isolated from the soil. So interestingly, what we observed that one of the extract out of 10,000 samples collected, one of the extract which we prepared, it showed immunosuppressive activity. And when we looked into this organism, you can see this organism was a filamentous actinobacteria. So actinobacteria, these are gram-positive bacteria, and whatever it secreted, it showed immunosuppressive activity. So after this active ingredient was isolated, purified, and when structure elucidation was done, it was observed that it was cerulomycin A. So cerulomycin A, the structure is this is a bipyridyl compound, and when we identified the structure, we came to know that this is already known molecule. And this molecule was reported by Hext in 1956. It showed antifungal and antibiotic properties. Okay, so this is something like, again, repurposing of a drug. So the first thing was whether this cerulomycin, it retains, or rather cerulomycin also shows immunosuppressive activity. So whatever we observed in the extract, is it the right molecule which we have isolated? So what we did very simple assay, and in this assay, we did T cell proliferation and added different concentration of cerulomycin. So here, if you see that there was a dose-dependent suppression of T cell response. So indicating that this cerulomycin, it has got immunosuppressive activity and it works on T cells. And similarly, what we did allo response, that is MLR, mixed lymphocyte reaction. That is, we took lymphocytes from two individuals, mixed them, and then we looked into the proliferation and added different concentration of cerulomycin, and we could see that the proliferation was decreased in a dose-dependent manner. So this indicates that cerulomycin, it has immunosuppressive activity, and this was reported for the first time by our group that it can inhibit the proliferation of T cells. So just to confirm it, we also purchased cerulomycin from LKT. It's a company which was manufacturing cerulomycin. We also purchased cerulomycin from Sigma, and the result matched very well with the cerulomycin which we purified. Now, the important thing is whether the cerulomycin, it can suppress the activity of cells which are responsible for inflicting two autoimmune diseases, that is Th1 cells and Th17 cells. So Th1 and Th17 cells, they are considered to be the key cells in provoking autoimmune diseases, whereas it has been proven, as I mentioned, the regulatory T cells, whether they can suppress the activity of these cells as well. So what we did, that we generated regulatory T cells in vitro, and then we did some of the experiments. So we observed, besides that, even rapamycin, which is also immunosuppressive drug, and it is used to treat autoimmune diseases, rapamycin can also successfully expand regulatory T cells. So it was already reported that rapamycin, it does. And rapamycin is also a molecule secreted by the microorganism. Besides, there's another molecule which is immunosuppressive, LY294002. It is also known to induce Tregs. So we thought that it may be probably first thing we should look into whether cerulomycin generates regulatory T cells. And importantly, rapamycin, even though it is used to treat autoimmune diseases, but it has a potential to increase the incidence of diabetes. So this is the big drawback with rapamycin. So first question we asked whether cerulomycin can induce regulatory T cells, and at the same time whether it can suppress, because both of these cells, they reciprocally regulate each other. So if you see this slide, probably this slide is loaded with many of these graphs. So if you see here in the upper panel, what we did, we generated T cells, and these T cells, they were CD4 T cells, they were stimulated with anti-CD3 and CD28. And so this was a clean system so that there is no interference of other cells or other molecules. So there were only CD4 cells stimulated with anti-CD3 and CD28. That is, they were given both the signals to activate T cells. And this upper panel over here is shown that different concentration of cerulomycin was added. So you see here, when no cerulomycin was added, these cells, they showed about 1% of regulatory T cells. So this y-axis is FOXP3 staining. So this is the flow cytometry data, and the x-axis is CD4. So both these molecules were selected because regulatory T cells, they express CD4 and FOXP3. So FOXP3 is a transcription factor for regulatory T cells. So it was very interesting to observe that as we increase the dose of cerulomycin, there was an increase in the percentage. So these figures indicate percentage of regulatory T cells. So these regulatory T cells, you can see here in the box, they increase gradually. And this is the positive control rapamycin. So if you see the same molar concentration of rapamycin and cerulomycin almost showed equal increase in the percentage of regulatory T cells. Interestingly, when we added TGF-beta, so TGF-beta is a differentiation factor for regulatory T cells. So we thought, why not we add TGF-beta with cerulomycin, see what is the impact. So if you see here, when TGF-beta is added without cerulomycin, there was a substantial increase in regulatory T cell. It went to 27%. And as we added cerulomycin, there was a gradual increase in the percentage of regulatory T cells, and it went from 27% to 47%. Similarly, when TGF-beta was added with rapamycin, the increase was 57%. So this is data plotted from this flow cytometer graph. So you can see there's a gradual increase in the percentage of FOXP3 positive T cells. So this was quite exciting result for us, that cerulomycin can induce the activation of regulatory T cells. So besides this flow cytometry data, it is also important that you have to prove being from India, you have to prove by many methods that whatever you are saying, it is really true. Okay, it's not. Otherwise, the reviewer will tell, how do you know that is artifact? So besides flow cytometer, we also did RT-PCR, and through RT-PCR, we could show that when cerulomycin was added, there was an increase in the percentage of regulatory T cell with cerulomycin, and this is rapamycin. And when cerulomycin was added with TGF beta, there was substantial increase in the percentage of the fold of FOXP3 positive T cells or regulatory T cells, and this is rapamycin. Right. So the important thing is that now whether this cerulomycin, although it is increasing the percentage of regulatory T cell, but regulatory T cells, they are suppressed by Th17 and Th1 cells. So it was important to see whether interferon gamma producing Th1 cells and IL-17 producing Th17 cells, they're also suppressed by cerulomycin. So what we did, again, we stimulated CD4 T cells with anti-CD3, CD28, and then we added different concentrations of cerulomycin. So upper panel indicates concentration of cerulomycin. So you see here flow cytometry data, it indicates that Th1 cells, which were stained with interferon gamma. So interferon gamma, as I said, is a marker for Th1 cells. So as the concentration was increased, there was substantial decrease in the percentage of interferon gamma producing Th1 cells. And similarly, we observed that Th17 cells percentage was also decreased. So this is a graph which we have plotted from this data. So there's a dose-dependent decrease in the cells which are deleterious towards autoimmune diseases, right? And we also did ELISA, right? So flow cytometry, and also we did ELISA, and in supernatant, we did measure interferon gamma and IL-17, and we could see that IL-17 secretion was decreased, interferon gamma secretion decreased, and IL-17 secretion was also decreased. So we also observe whether these cells, which are now showing the decrease in the secretion of interferon gamma and IL-17, whether their activation state is also decreasing. So we use CD44 as activation marker and observe that there was a decrease in the expression of activation marker. So also we did the staining with the JC1, and we observed, and JC1, or the mitochondrial potential, membrane potential, was measured through JC1. This is inversely proportional to the activation. So as the activation goes down and this increases, so we could observe that mitochondrial potential, it increased from 17 to 29. So this shows that there was decrease in the percentage of Th1, Th2 cell, and their activation or their activation stage was suppressed. So now the important thing was whether the Tregs which we generated by cerulomycin, are they capable of inhibiting function of effector T cells? So in this case, what we did, again, we stimulated cells with anti-CD and CD28. These were CD4 cells, purified cells, and we could see that CD69, which is early activation marker, it was also decreased. Right, and when we added TGF beta, we could further see decrease. And this is the functionality we tested. That is, we generated regulatory T cells, and then we did MLC mixed lymphocyte reaction over here. So the responder cells and the stimulator cells were added, and we added regulatory T cells. So when regulatory T cells were added over here in this MLC, we could see that there was a significant decrease in the activation of effector cells, so indicating that regulatory T cells, they can suppress the function of regular effector T cells. Now, the next question was whether the presence of Th1 and Th2 polarizing cytokines can influence the differentiation of cerulomycin-induced Tregs. That is, if you give condition where you are stimulating the activation of Th1 and Th17 cells, whether Tregs, they are suppressed, because as I told you, Th1 and Th17 cells, they suppress the generation of Tregs, and Tregs also suppress the generation of Th1 and Th17, depending upon the condition. So in this case, what we did, we measured both interferon gamma secretion or expression through flow cytometry. So this is intracellular cytokine staining, which we did. And side by side, we also did the expression of FOXP3 positive cells. So this x-axis indicates FOXP3, y-axis indicates interferon gamma, that is, this is Th1 cell, and this is regulatory T cells. So if you see over here, we could observe that in polarizing condition, when Th1 cells were produced, so 67% of interferon gamma secreting cells were present, that is Th1 cells, when no ceromycin was added, and there were very less percentage of FOXP3 positive cells, that is 2%. As we added ceromycin, this percentage from 67 went to 64, 45, and 26. But at the same time, you see here, in even Th1 polarizing condition, the interferon gamma secreting or Th1 cells, they increased from 2% to 7%. So interestingly, same thing was observed over here with Th17 cell. So the Th17 cell, when no ceromycin was added, were 41% and 3% of FOXP3 positive cell. And as we added ceromycin, so these cells, they decreased from 41 to 25, 17, and then there were no Th17 cell. And interestingly, regulatory T cells, they kept on increasing. So from 3%, they went to 49%. So this was very interesting observation that in Th1 polarizing condition, we could see that the percentage of FOXP3 positive cells, they increased. And similarly, in Th17 polarizing condition, these cells, they increase. Okay, so this FOXP3 positive population, it increased. Similarly, in case of interferon gamma secreting cells, they decrease, and they increase. Similarly, FOXP3% positive cell, they increased, and Th17 cells, they decrease. So this showed a significant decline in Th1, Th2 cells. Now, the proof of the pudding is in eating. So whether this ceromycin can ameliorate clinical symptoms in an experimental model of arthritis. So we induced arthritis in animals. So if you see here that when arthritis was induced, so this is a normal animal, and this is placebo. So we induced arthritis using myelin basic protein, and this was given in complete fluids adjuvant. And you see there's a significant swelling in the paw of this mouse. And as we injected ceromycin over here, there was decrease in the swelling. And when 10 milligram per kg body weight was injected in this mouse, you can see it is as good as normal paw or foot pad of a mouse. So here we have plotted the clinical symptoms, which were taken as mild erythema 1, and with inflammation in the limb digits. Then number 2, there was severe inflammation up to paw, and in 3 was given to severe inflammation up to the ankle. So when placebo was given, there was increase, substantial increase in the thickness of the paw swelling. And when we gave ceromycin, you can see there was decrease. We also proved this through histopathology. And if you see when ceromycin was given as compared to highly inflammatory position over here, so there was a decrease in infiltration of cells here, and there was less synovitis in the joint capsule. So this is histopathology of joint capsule. So it became as good as the normal, and it's a high inflammatory response. Then we did whole body imaging of this mouse. So this is a foot pad, and you can see the control. There is no swelling, and this is placebo. So the blue, more the blue, indicating that there is swelling. So in this case, what we did, we measured this swelling through injecting osteosense and ProSense into these animals. Okay, but when we gave 10 milligram kg body weight of ceromycin, we could see there was substantial decrease in the foot pad swelling. And this swelling we have plotted as a clinical score. So when 10 milligrams of ceromycin is given, you can see there was substantial decrease in the foot pad. And similarly, you can see the synovitis, it was substantial decrease. And ProSense, which measured inflammatory response, there was decrease, and osteosense as well. So what we did, we also measured in this capsule through RT-PCR the expression of interferon gamma, TNF alpha, and IL-6, and both of these three cytokines, they are responsible for inducing pro-inflammatory response. So you see that there is decrease in the inflammatory response when ceromycin was given to these animals. Similarly, we also looked into the presence of FOXP3 positive cells into these animals. And if you see here that this is control and this is placebo, and the animals which were injected 10 milligrams ceromycin, there was substantial increase in regulatory T cells. So similarly, we also did ELISA and measured the secretion of interferon gamma. So animals which were injected with ceromycin, they showed substantial decrease in the secretion of interferon gamma. TNF alpha was also decreased, and we also measured MMP3. This is matrix metalloproteinase 3. So all these pro-inflammatory responses, they were substantially decreased in these animals. So this shows that this molecule have sufficient potential that it can suppress the inflammatory response. Now, the important thing is whether the ceromycin is influencing cytokine-mediated signaling of STAT molecules. So important part is that you have to see whether this is an artifact or it is really some mechanism which is operating into the system. So for that, we looked into the molecules which are responsible for induction of interferon gamma. These are signal molecules like STAT1. STAT1 is responsible for induction of interferon gamma, and we did Western blotting. So if you see here in this case, we could see that there was substantial decrease in the expression of STAT molecule with the increase in the concentration of ceromycin. So here, if you see, this is a control, and as we added ceromycin, there was decrease. And all of these, we added interferon gamma to induce the expression of STAT1. So when you added interferon gamma, there's high expression of STAT1. And when we added ceromycin, there was a gradual decrease in this intensity of band. And not only by interferon, by Western blot, we also showed this through EMSA. So through EMSA, we also showed, and we also showed the decrease in the expression of IL-6. And we also looked into other molecules which are responsible for induction of Th1 cells, like JAK1, JAK2. So since JAK2 does not induce any response, and therefore we could not see any difference, so indicating the response is quite specific. Similarly, we also did RT-PCR and to see whether there is any decrease in pro-inflammatory molecules, and we looked into IRF1, IRF7, and ISGS15. These are all pro-inflammatory molecules, so there was decrease in them. And SOX1 is a regulatory molecule for it is responsible for stabilization of FOXP3 by controlling the Th1 and Th2 differentiation, and therefore it suppresses STAT1. So in case if STAT1 is decreasing, there should be increase in SOX1. So we could observe that there was an increase in SOX1 molecule. So just to see whether it is the response is specific, we look into other molecules, STAT3 and STAT4, which play no role in controlling IL-6 or interferon gamma mediated response. So we could not see any difference in the expression of STAT3 and STAT4, indicating the specificity of this reaction. Similarly, we could also looked into other pro-inflammatory molecule like fast ligand, and with ceromycin there was decrease in the expression of fast ligand. You can see here, and also SMAD7. It is SMAD7 also controls the expression of TBAT and interferon gamma molecule, and TBAT is also differentiating factor for Th1. So we also could see decrease in TBAT molecule, and we also did through Western blotting, and if you see here, that is a clear-cut decrease in the intensity of SMAD7. And we also did Western blot beside RT-PCR, and through Western blot, the results were reproducible of RT-PCR. So now the next question was whether ceromycin enhances TGF-beta mediated SMAD3 signal. As I told you, that SMAD3 regulates or stabilizes the expression of FOXP3. So the next experiment was to see the expression of SMAD3 with ceromycin. So if you see here that we could observe when we increase the concentration of ceromycin, there was increase in the expression of SMAD3. And when we added interferon gamma, we could see that there was not much influence on the expression of SMAD3. And in such case, we observed the expression of FOXP3, and when interferon gamma was added, we could see that yes, there was constant increase in FOXP3 expressing cells. And finally, what we did, we looked into we use inhibitor. SIS3 is an inhibitor for FOXP3, and when this inhibitor was added, we could see that ceromycin induced enhancement in the expression of FOXP3 was substantially reduced when SIS3 inhibitor was added, and we could not see much enhancement even though when we added TGF-beta. So this proves that the induction of regulatory T cells of FOXP3 is specific and could be observed through ceromycin. So the major finding of this work is that the result indicates that the novel role of ceromycin in inducing the generation of FOXP3 positive regulatory T cells, and it significantly suppresses the percentage of Th1 and Th17 cells, and this has been mediated very well by suppression of STAT1 signaling and augmentation of SOX1. So this is through increase of SMAD3 activity, and ceromycin can rescue Tregs from interferon gamma induced inhibition. That is, even if there are presence of Th1, Th2 cells, this interferon ceromycin T cell can still induce the expansion in the pool of regulatory T cells. So this finding suggests that ceromycin have potential to be considered as a potent future drug for treatment of autoimmune diseases. So we have got a patent on this, US patent on this molecule, and we also license this technology to a pharma company, US pharma company, for $3 million US. And finally, I would like to thank my team. So this work was carried out at Institute of Microbial Technology, and whatever work I have shown to date was mainly done by Rama, and then it was followed by Veshle and Sudhi. And I would also like to thank Dr. R.S. Jolly, who did structure elucidation of this molecule, and Dr. R.M. Vora for microbiology, and also would like to thank CSIR for financial support. So thank you very much. And this is India. You can see how beautiful India is. This is not far, this is far off from here. This is Timble Trail. I welcome now for question answer session. Okay, I'm ready to take question answers.
Dr. Javed, can I ask a question?
Yeah, definitely. So,
Yeah. So have you also looked at the induction of FOXP3 in antigens in an antigen dependent manner as a result of cerulomycin treatment?
No.
Probably these cells that were used in all these experiments were a polyclonal population, right?
See, in case of in vitro, this was a polyclonal population, but you can very well say in case of in vivo, in arthritis model, where we injected MBV proteins, so most likely the cells which were generated, these may be MBV reactive. So in vivo, whatever we measured, of course, they may be antigen reactive, but then again, whatever in vitro experiments we did, we stimulated them again with anti-CD and anti-CD28. So this is, of course, a polyclonal population.
Thank you.
Okay, so let me check the text box as well, if there are any questions. Because we are alternatively using another system as well, and this system so there's a question where one of the students want to ask this is Ipseta Jena, and the question is this drug has any role in immunosuppression in cancer? So actually we have not checked yet about the immunosuppressive role of this molecules, but this is a very interesting question. And there's a question. The question is from Prashant Kumar, and the question is, could this drug be used as a general immunosuppressant? So yes, as far as it is concerned, it appears to be, it is inducing general immunosuppression. But at the same time, the interesting part is that even though it suppresses pan T cells, but it increases the pool of regulatory T cells. So this is quite interesting part, and in this molecule, which is also seen in case of rapamycin. Okay, so another question from Manjeet Chopra from Naipra Mohali. What is the role of this molecule along with probiotic in post-menopausal osteoporosis? We haven't checked about it, so we really cannot comment on it. So another question is from Abhishek Chattar, and the question is, cerulomycin suppresses the differentiation of naive T cells and alleviates symptoms of experimental encephalomyelitis. Yes, it also suppresses encephalomyelitis that we have seen. And there's another question that from Shilpa Keshta. How would antigen-specific T cell against B cell of pancreas antigen act in the presence of cerulomycin? These experiments will address the issue of diabetes induction as seen in case of rapamycin. Yes, this will be really very interesting, and but right now we don't know. It appears from the data, whatever we have generated, yes, it can suppress Th1 and Th17 cell. And if it, in a case of diabetes, if it expands Tregs, it will be really wonderful. So there is another question from Imtiaz Rashid. Did you check the role of this molecule on CTLA4? Yes, we have seen the role of this molecule on CTLA4, and it increases the expression of CTLA4. Then Anjali Patil, is their NK cell also responsible for effector Tregs and suppression of cerulomycin? So we don't know about, we haven't looked into NK cells. There is a question from Kumar Subham, how to find that which microorganisms have potential role for immunosuppressive molecules. So same way, this is what I presented, that you can collect the different samples and then you can look into their, you should have people who are expert in making extracts of it. Then you should have people who can isolate these microorganisms, they can grow, and finally, if you find any activity, then what you do, you have to see whether you have a chemist who can separate it. And the screening which we developed was very simple. We took we used to take total sphenocytes, and then we stimulated them with CON-A. Okay, this is a mitogen, and then we added different extracts, different concentration of extracts, and this is how we did in mitoteter plate screening. Okay, so there's a question from Manjula Jain. It's used in COVID cases to care of lateral damage that is reported. Yes, this is again very important question, that in a COVID patient there's a cytokine storm, so cells get activated. So whether it is possible that it can work, but yes, it can work. But the important part is again that is going to ultimately suppress the immunity in general, and the person may likely be exposed to other opportunistic infection. So this is the important part of this therapy. Okay, so there are many other questions, and there is a person, Hilal Ahmed from Central University of Punjab. The question is, what is the effect of this molecule on different vaccination? Yeah, that is again a very important question, and it may have impact, yes. It may have a big impact on vaccines as well. So another question from Sayyid Aziz Tehseen, is antigen presentation affected by CA treatment? Yes, this is again a very interesting question. Even though T cells and even B cells, we could see more T cells are being influenced by this molecule, as you have seen, that there are about 99% of T cells, they get suppressed. But B cell, they will not suppress more than 50%. And important part that macrophages or dendritic cell, they were not at all affected by this molecule. So when they are not affected, probably antigen presentation is also not affected. Okay, is there any there's a question now, this molecule have equal beneficial effect on SCID like DiGeorge syndrome and ZAP17. See, this is an immunosuppressive molecule, and in case of SCID already these animals, they are immunocompromised, and in DiGeorge syndrome, these people are born without thymus, or they have got a very rudimentary thymus. These are like nude mice. So what will be the effect? Already people are immunocompromised, immunosuppressed. So I don't think one should try any immunosuppressive molecule on these people. So is COVID-19 an autoimmune disease, and can this help in treating COVID? COVID-19, no, it's not autoimmune disease. This is a viral disease. Can serolomycin be used to treat hyperallergic reactions? Yes, it can be used. It can suppress T cells. So there is a question whether COVID-19 gets suppressed by traditional foods. Okay, we don't know about it. Another question by Yatin Bhatia. How far this molecule has been probed towards anti-diabetic treatment and by which mechanism? No, we haven't tried yet. Sailesh Vishwakarma, when will the vaccine of COVID-19 come? As you have heard from Professor Peter Doherty, yes, many people are working, and we hope that it should come soon. So there is another question by Anjali Patil. Is the mechanism to suppress immune system is also same for other microorganisms? No, it depends on. Many of them, they can suppress through calcineurin pathway, PD-1, and there was different pathways through which it gets suppressed. There is a question by Sita Jena. Were dendritic cells affected by these molecules? No, as I said. Okay, then Dayanidhi Sahu, can serolomycin affect endocrine stimulation? We really don't know about it. But we have done toxicology studies. This was done by Naipur Mohali, and they have seen that this molecule, the concentration which we have used, it does not affect it does not inflict animals with cytotoxicity. Anuj Bandaran, do immunosuppressive drug like serolomycin paves way for other opportunistic infection in case of compromised immune system? Yes, that is the big problem. Right, so there is another question over here, some questions over here from Debrun Patra. Is this drug have a role in targeting phosphorylated NF-kappa during immune cells? Yes. Yes, we have seen in case of NF-kappa as well. And I think I missed that data. We have seen, yes. Then there's another question from Rashmi Kumar. What is the effect of this molecule on B cell? And have you checked whether antibody against the molecule is generated in vivo? Right. So as I told that it has a major effect on T cells. 99% T cells get suppressed, and B cells we could not see suppression and proliferation of B cells or activation of B cells or secretion of antibodies. We looked into antibodies like IgM, IgG1, IgG2A, 2B3, and we could not see more than 50% of suppression. And whether you generate antibody against this molecule, no, we haven't seen, because since it's a small molecule, it's just 300 kD molecular weight, 300. So I don't think, since it's a hapten, it won't induce any antibody response. Okay, so with the constraint of time, I would now like to stop my presentation, and it's time for invite another speaker, Professor Dipankar Nandi. Professor Nandi, are you available?
Yes, I'm coming on.
Okay, welcome, Professor Nandi. So we welcome you over here, Professor Nandi.
Thank you.
All right, so let me introduce Professor Nandi. Professor Dipankar Nandi is a very renowned immunologist of this country. And he did his, currently he's working as a professor at Indian Institute of Sciences, Bangalore. And before he joined Indian Institute of Science, Bangalore, he worked with a Nobel laureate, Jim Allison. Okay, Jim Allison, who discovered CTLA4 and worked on checkpoint therapy. And today, Dr. Nandi is with us, and he's going to talk on a very interesting and emerging topic on COVID-19 and roles of other infections and epidemiological studies. So we welcome you, Professor Nandi. Over to you.
Can you see my slides?
Yes, we can see your slides.
Fine. So should I start now?
Definitely, you start, please.
Okay, fine. So very good morning to everyone, and I'd like to thank Javed and the organizers for giving me this opportunity. What I will do is, this is a small story that we worked on during the pandemic, and I'll tell you a little bit about it. And I thought it would be of interest. The main agenda that I have over here is to get some inputs on this. Okay. So as Javed mentioned, that I'm at the Indian Institute of Science. I've been here for quite some time, and this is the Tata statue, and I think we have a flower show on what's known as Founders Day, which is 3rd of March. So I've been associated with the biochemistry department. It is one of the oldest departments in this part of the world, and we were in this iconic building for quite some time till 2011, and we have recently moved some years back into this biological sciences building where a lot of other departments are also there. Okay, so my lab works on different aspects, and as I've said that, and I'll give you a little bit of a background as to how we got into this, but we work on many things, and I thought we would sort of share this news story with you all. Okay, so I will tell you a little bit about coronavirus. Professor Doherty has already given and answered a lot of questions, so I think a lot of people are quite familiar with this. And the subject of today's talk and subject of for quite some time for all of us are coronaviruses, and these are RNA viruses which are mainly associated with respiratory tract infections, and they can cause anywhere from mild to major ones, and the major ones we know of the SARS, MERS, and the current one. What is interesting is coronaviruses were discovered quite early in 1967, and this is a very famous paper which has now become very famous by Almeida and Tyrrell, where they describe, these are TEM images that are shown over here, where the first images of coronaviruses were found. And the name corona comes from crown, and so these are not the ones on top are not that particularly clear. The one in the bottom is much more prominent, and this is a TEM image that have been adapted from the CDC, and you can see the nice crown that these viruses get their name from. Okay, so two major diseases have been associated with coronaviruses until the current one, and which is the SARS, and which SARS started off again from China, and it had a fatality or mortality rate about 10%, which is very high, but thankfully did not spread too much. The second one, MERS, was in the Gulf. It had a much higher fatality rate, about 35%, but again, it did not spread so much. So we were warned actually twice, but perhaps we did not take the warning very seriously, and so we have to deal with the current one. Most people think that there are only 3 types of coronaviruses, the 3 prominent ones, but actually there are a whole bunch of coronaviruses you have, and they can be classified into different forms. You have the alpha coronaviruses, the beta, the gamma, and the delta ones. The ones that we are concerned with mainly arise from the beta ones. And this is where the SARS, MERS, and the SARS-CoV-2 are part of it. And you can see that most of them they infect mammals, and the beta ones are thought to infect animals, but it's the rare ones or the mutant ones that are able to jump into humans, which cause problems. But suffice to say, there are a lot of coronaviruses out there, and who knows what will happen in the future. And this is some sort of a warning for all of us. Okay, so the disease that we know and that we have been grappling for the past few months is COVID-19, and this started off from what we know in Wuhan, China. And the virus has been called, the disease is COVID-19, the virus is SARS-CoV-2, and you have a variety of symptoms. But again, it's mainly a respiratory infections it causes, so we need to keep that in mind. And it occurs in close contacts. And so therefore, you often find cases within families because they're living together. And so I think one easy way is, of course, to wear mask, and also social distancing comes into place over here. So where did it arise? As I mentioned, that these were in mammals, and bats have been thought to play an important role, and it's also possible that other animals were also involved before some of them started jumping into humans, and you have pigs and pangolins also thought to play intermediate roles. Okay, so I had shown you the structure of SARS-CoV-2, and the prominent one is, of course, the spike protein, which is shown over here, and these bind to the ACE2 receptor, and that's how it enters the cells. And you have another protein which plays a very important role, which is the TMPRSS2, which enhances this binding between the spike protein and ACE2. And so you have different drugs that are obviously being tried that may play important roles over here. Okay. So as mentioned before, that this occurs mainly through respiratory diseases, and initially it's the upper respiratory tract that is affected, and with time, of course, it moves down to the lower respiratory tract, and it can culminate into other systems, including sepsis, cytokine storm, which might lead to multi-organ failure, increase in blood clotting, and so on. Okay. So this disease has created, a lot of problems for us. It's a pandemic, and currently it has there are about more than 7.4 million cases, with about more than 4 lakh deaths. In India, we have almost 3 lakh cases, and with about 8,400 deaths and so on. Of course, mortality is one big issue. The other big issue is economy, because a lot of people depend on jobs, and if they don't have jobs, that affects them. So both lives and the economy is hurt in all over the world. Okay. So let me now start coming to what are the questions that we addressed and how we came up with this. So we've been involved in teaching immunology for several years to both undergraduates and postgraduates. And one of the key questions that comes up is, why is it that some people are more sensitive to allergies, and we see that around, whereas others are not as sensitive. There are some people who are more sensitive to infections, so tuberculosis is a good example, whereas others are not. So we've also, as part of the course, we've also been keeping track of disease spread. So, you had AIDS, which I think some of us are familiar with. And then, of course, some of the ones that I mentioned, we had SARS, MERS, and because with increased globalization, increased contact with animals and so on, some of these viruses are jumping, Ebola, and so on. So this is in the background. So these are questions that most of us ask. Why is it that some people are sensitive? Why is it that some people are resistant, and so on? And this becomes important as we'll go around. Okay. So how did it all start? In 16th March, students in IISc were asked to go home based on a government of Karnataka advisory. The lockdown in India, based on Government of India, started in 25th March. And so this is clearly, the government of Karnataka was much more proactive and encouraged students to go home. And there was a skeleton group that was there in IISc to take care of labs and all. So there was one student in my lab, Sanmoy, and me. So we could not do any experimental work because there was a lockdown. So what we decided to do was we initiated, a study on epidemiological data that was available on the public domain. And thankfully the internets were working, and of course we now realize the importance of it because a lot of work gets done through that. We also collaborated with Dr. Mohit Jolly in the bioengineering group over here because we needed some help with statistical analysis and so on. So he brought in his strength into our sort of analysis over here. Okay, so what is the observation? And I've sort of hinted before. The observation is as follows. Some countries show a very high number of COVID-19 deaths, whereas others don't show that such high numbers. So, for example, the US, Italy, Spain, France, these countries the mortality rates are very high. And as Professor Doherty mentioned, you have senior centers being affected. And there are some groups that are much more affected than others. So, I mean, this is an important question. There have been multiple factors that have been thought to be responsible for this. And so, obviously, there's a heterogeneity in it. And so, what are some of these factors? Some of these factors, most of us, are quite familiar with. One is age. So one of the thoughts, and most people are also aware that seniors above 65 are likely to be hit by this, and so therefore they need to be careful. This is you have other comorbidities. So, for example, people having hypertension, diabetes are also in the likely sensitive group. Then you have cultural aspects. So, for example, hugging people is, a Western cultural thing. People from Asia don't usually hug as much. Of course, the younger ones, there's a change in the cultural aspects, and the younger ones may tend to be more prone for hugging than the slightly older ones. But in general, hugging is more of a cultural thing that is often seen in the West. Then there are other factors. You have temperature. So some people have said that usually in very warm temperatures, it's unlikely to play that much of a role. You have government healthcare policy. So in some countries, you have very good healthcare, whereas other countries, healthcare is not all that great. So there are multiple factors that are involved. You also have, where people have sort of taken care that the spread is less. Professor Doherty mentioned the case of Australia, and they are an island. They were very careful about this. Anybody who comes in is quarantined. Whereas others may not have been all that careful. They base their decisions on aspects such as herd immunity. And unfortunately, in this case, it has not worked out all that well. And we can discuss that a little bit later. But suffice to say that there are multiple factors involved in this. All right. So the questions that we asked was, there are multiple factors, but we wanted to look whether there is any association with any diseases. We also wanted to correlate these diseases with vaccination, and so that becomes important. So both look at diseases as well as vaccination to try and see if any of these would affect both COVID-19 incidences or deaths. Okay. So this is pretty much how we did it. So we initially started off with 25 countries. These 25 countries were based all around the globe. So we wanted to ensure that at least there are representative countries from all around the globe. And we also, selected for the more prominent ones, as you will see. But they are pretty much represented around the globe. So it takes a lot of time to look at this. Get all the data for this, and there was Sanmoy who did all this work. And so then we analyze for different diseases. I mentioned tuberculosis, flu, measles, hepatitis B, polio, and this is all using public databases. As I mentioned, we also look at vaccination. So this is, another aspect of our lab works on different responses, and we use genetics. So we usually look at wild type, what happens with wild type, what happens with a mutant, and that is very informative. So in terms of epidemiology, we've looked at disease and we have looked at vaccination to try and see what happens. So just to give you give students a sort of flavor of what happens, and then of course we did correlation and significance studies to try and see if they hold. All right. Okay, so this is to explain the role of vaccination. So vaccination is very important, and vaccination has clearly improved the quality of our lives. Okay, a brief example is students these days have not been vaccinated for smallpox, but people in Javed and my generation have been vaccinated for smallpox, and smallpox has been eradicated. So for a long time, smallpox was a big problem. The second example is polio. Now, polio has been eradicated in most countries. Okay, and that has clearly it's been very useful because you don't want to have kids with polio. However, there are issues, and in 3 countries, polio vaccination coverage is less. It's less because some people don't want their kids to be vaccinated for whatever reasons. And these countries also have other issues, and the countries are Pakistan, Afghanistan, and Nigeria. And unfortunately, you have kids in these countries with polio, and this is something that can be easily prevented. Overall vaccination has clearly been very useful, and we must keep that in mind. Okay. So what we have done is initially looked at disease, COVID incidences, COVID deaths, checked with respect to measles, vaccination, diseases as well as vaccination. We did this with measles, hepatitis, and polio, but there was no correlation. Okay, so that's an important part over here. Okay, so the initial ones, there was no correlation, and which acts as a negative control for us. So just because you have some diseases or you vaccinate, it's not that or we were unable to find any correlation over here. So in this first part, I just want to mention. So the next one that we chose was flu or influenza. That's known. It's interesting because flu is also a respiratory virus, and it has mild symptoms. And you have different types, and the 2 big ones that people worry about are influenza A and influenza B. And you have often different waves of this. You have influenza A, and then I have influenza B, and this is seasonal because these viruses mutate a lot. And there are quite a lot of cases. The mortality rate is about 0.1%. What is interesting is the vast majority of deaths with influenza is usually seen in people who are 65 and above. And so that is why a lot of vaccination is also done of people. So in fact, and there are ways by which people try and boost up the vaccines so that they are more effective in this group of people, so that you are able to generate a better response. Because remember, these people are also aged, and they might be entering immunosenescence, and so people are thinking of ways of increasing dose, increasing or diversifying the adjuvants in order to get better responses. So this is something one needs to keep in mind. So often when we see the reports, it's usually vaccination for the group that is above 65. Okay, so this was our first interesting finding. What we found is that countries that there was a negative correlation over here. So countries which have more flu incidences, so you have Iran, Bangladesh, Pakistan over here, have less COVID deaths, whereas countries that where you see less flu, they have more COVID deaths. Now, why is it that you have less flu incidences? There are less flu incidences over here is because people are vaccinated, right? And so that is again we see that. So this was a nice correlation in that sense. So again, where there is no flu vaccination, for example, there are less COVID deaths. Where you have more flu vaccination, actually you see more COVID deaths. Okay, so there is some sort of a correlation between both flu incidences and flu vaccination with respect to COVID deaths. Okay. So what are the different reasons that could explain this? So again, it's multifactorial. And it's something that we have not looked into. One of the possibilities is to see if flu infections downregulate the ACE2 receptor. So maybe so as a result of which SARS-CoV-2, it's difficult for them to enter the cells. So people with high flu show less. So is it that they have much better immunity? There are other examples where viruses can also impart non-specific immunity to other pathogens, and that's what. But one of the more interesting ones that have come up is apparently in the respiratory tract, it seems that one virus affects other viruses. And this is known as what the virologists call as viral interference. There have been cases where flu infections affect other infections by other viruses, including coronavirus. So that plays an important role, and it is possible that in countries where there are higher flu infections, like India, that we are somewhat better protected, and the reasons are. On the other hand, if you have more flu vaccinations, then the host does not get to see the flu virus, as a result of which, it's possible that in this case, SARS-CoV-2 has a much better chance of causing an infection and causing this. Okay, so this was our first finding that we found with flu incidences and flu vaccination. And so we wanted to look at other viruses, and the other respiratory disease is tuberculosis and, of course, BCG, and that's something that we again tried to correlate. Okay, a little bit about it. I think quite a lot of people over here are quite familiar with this. Tuberculosis caused by Mycobacterium tuberculosis, and there are a lot of cases over the world. So in 2018, there are almost 10 million active cases. Countries such as India and China have very high TB cases. In India, for example, the deaths due to tuberculosis is about 4.4 lakhs per year. So naturally, most countries use BCG as protective, but BCG, as was mentioned, is a live attenuated vaccine, and it's one of the often it's given to newborns, and it's often one of the first vaccines that they get. And so BCG is a very good pediatric vaccine. The question whether the BCG is useful in adults is controversial, especially in places like India, where you have quite a lot of TB bacteria around, and there might be other environmental bacteria also too. I must mention over here, because it's a live attenuated vaccine, in kids who are immunocompromised, they come down with a disease known as BCGiosis. And that is because these kids, it's a rare event, but these kids either lack the cytokine or the receptors for interferon gamma or IL-12. So it's a live attenuated vaccine, and that's the important part. Okay. So if we take a look at the correlation with countries with high TB incidences, there was none. The correlation came up with countries with low TB. Okay, if in and deaths were much higher. So these are countries mainly in the West and in more clean environments over here. Okay, so with BCG, it turned out again to be a negative correlation. The R is negative over here, and as can be seen, that countries such as which have higher BCG coverage have lesser amount of COVID deaths, whereas countries with no BCG coverage have countries with no BCG have higher deaths, when countries with greater BCG coverage have lesser COVID deaths. So this is a negative correlation. Okay, so there were two aspects over here. As mentioned to you previously, BCG is given to newborns. So how is it that that people are protected several years later? And so this was a question that was intriguing, and people have shown apparently that, and these are published reports that BCG is protective, and it has quite a long-lasting protection. So how is it that BCG is protective? Apparently, and this is what, again, Professor Doherty talked about, and this is something known as trained immunity. What happens is over here, BCG apparently causes epigenetic changes in stem cells, which increases protection. And often trained immunity, one of the markers is IL-1 beta over here, and so which is able to generate better protection. And it's possible this non-specific protection may be helping us. Okay. At this point, we had two factors at play. One is flu and flu vaccination and BCG coverage. So what we have done is then what we did was segregate the countries based on this. So you have three different types over here. You have countries with flu vaccination, but no BCG. You have countries with flu vaccination as well as BCG, and you have some countries like us that don't have flu vaccination but have BCG coverage. And so this is what is shown. So we can segregate this into three categories: one, the high, the moderate, and the low over here. And so the high countries with high COVID deaths are the US, Italy, Spain, France, as you can see over here. And these are the ones that don't have BCG but have flu vaccinations. The ones in the moderate are ones that have flu vaccination, but they also have BCG, and you can see that they have turned moderate. So it shows you the importance of BCG vaccination. And of course, the lowest ones are seen where you have BCG, good BCG vaccination coverage, but no flu vaccination. What we have done is the same data has been normalized to population and then replotted. Okay, so there are some aspects that I'd like to discuss over here. One is we have grouped these countries based on the BCG minus and flu vaccination positive, and by and large, it seems to hold. But there are outliers. The outlier that was mentioned is Australia. Okay, because Australia has this is BCG minus, flu vac positive, but there are very few. So again, it reinforces the importance of other factors over here. Australia, remember, is an island. They have taken much better protection. They have been proactive, and perhaps it has helped over here. So, but for most others, it seems to hold. You have moderate ones also, and it's the moderate it's the moderate ones that are somewhat heterogeneous in nature. Let me mention two particular countries over here. UK has both BCG and flu vaccination, and the same holds true for Sweden. Now, so there is some heterogeneity in this in the coverages. But let me also mention UK and Sweden initially started off with a policy of what's herd immunity. They allowed the virus to come in the hope that people will become resistant. And once it started taking over, they've changed the policy, but it the virus got in, and by which time it was somewhat somewhat difficult to control, and which perhaps explains these high numbers. So there are outliers in this data, and we need to try and understand what are the factors that seem to play roles in this.
You are left with 5 minutes.
Yeah, sure. I'm almost done. Yeah. So the third group are the countries with low, which is Iran, India, and so on. Let me also mention that China is in the moderate category, and even though the disease started in China, mortality rates are less in China. So that's again an important part. Okay, so these were the initial the findings. Let me quickly go through it. Because it was COVID work and we were a little bit nervous about being scooped, so we have published it as a preprint, and people are more than welcome to take a look at it. The data in this preprint is for 1st June, and the preprint was published on 6th June. So it is reasonably up to date. Okay. The advantages of publishing in a preprint is you get immediate feedback. Let me quickly address two feedbacks that we have addressed. One of the feedbacks that we have addressed is that in some countries the peak has been achieved, in other countries peak is still going on, in India, for example. So how do you address it? Of course, we did not have the luxury for the peak to end, so we have done with whatever we have, but we will discuss that a little bit. Second is, how does it correlate with mortality rates? Okay. So this is just to quickly show you that US and Italy were the ones where you have very high mortality. These are the ones in the middle. Of course, China started off very early, and they have. And whereas the ones in the low category, which is both Russia and India, you can see the lag that is there. And again, there could be multiple reasons for this lag. For example, the lockdown and so on. But there is a lag in these countries. So, okay. Second is we correlated it with mortality rates, and how does that work? So in fact, it works out very well. So as you can see, with flu vaccination, more the flu vaccination, greater mortality. You have a very nice P over here, right? And with BCG coverage, again, you see very good. So in fact, it's negative over here because more the BCG rates seem to correlate, and this is post the preprint publication. Okay, so let me reinforce again that there are likely to be that there are multiple factors involved, and what we have done is we have identified flu and BCG vaccinations to be playing important roles over here, right? What of course needs to be done, and it's possible that other pathogens and vaccinations may also be important, and that again maybe some other studies need to be done. What is, of course, because the COVID-19 is ongoing in some countries, the peak has been reached. Other countries, we are in the process, and perhaps this study needs to be looked at after peaks have been achieved. But as I said, we did not have the luxury of that. We did it as the disease was ongoing over here. What I think I'd like to end with is that this is correlation data, but it needs to be experimentally validated, and perhaps a better understanding of this may reduce COVID mortality in future. So, for example, clinical trials in susceptible populations, in senior homes and all, especially whether giving BCG might be useful, should be looked into. Okay, I'd like to acknowledge people who did the work. This is Sanmoy, who did all the work, and this is Mohit from who helped us with the analysis, and Taru, of course, helped us with the presentations and so on. Let me also mention the people who encouraged us to do the work: Dipankar Ghosh from JNU, DN Rao IISc, and Shambhu Basak. All of them wanted me to write a review on immune responses, and I thought we should try and do something new, and so then that's what it was. There are other people whom we have consulted and discussed this work, which is Arvind Sahu, Ravindran, Ramre Bhatt, Uday Ranga from JNCSR, and Vrinda. Okay. This is my lab group. This is pre-lockdown. I think there will be a lot of changes in this post what has happened. So we have to regroup and get it going. I think it will take some time. I like to end by showing you a picture of the IISc main building, and these are the Gulmohars in April 2020. And I thank you all for your attention. Thank you.
So thank you very much, Professor Nandi, for a very nice talk. And I hope after your talk, many questions people had in their mind have been answered. So I had many questions which you have answered during your talk. Really wonderful presentation.
Thank you.
So this is open for now question answer session.
Can I ask a question?
Yeah.
So you're saying that there is negative correlation between flu vaccine and COVID.
That's correct.
So is it in terms of susceptibility or severe disease phenotype? So that data is not there.
So what we have got is things that is there in the public databases. So we have just looked at two aspects, which is incidences and deaths. And we see most of our data correlating with deaths, but less with incidences. Okay, so most of our thing holds with deaths, and which is why, which is what we have said.
So also, so when you say that in some countries flu vaccine is given, so again, whether actually this is applied, whether this is the whole population is given that vaccine, or yes, it's reported that yes, the vaccine is actually tried in those countries, right?
So the data for flu vaccination is for people above 65 because they are the most susceptible group, and they are given. And so it's a little unfortunate from what it appears. It appears that you give the vaccine and you might be making them more susceptible to COVID.
Yeah. So, and what do you think actually is the immunological mechanism behind it?
Okay. So yeah, so we have tried to address this. I mean, again, this is what we have said, that we don't. I mean, it might be interesting to look at levels of the ACE2 receptor, whether about non-specific immunity. But the one that we have primarily invoked is viral interference, where having flu in us or being exposed to flu protects us against other respiratory infections. So, because that's what we see in most countries. So ours is a more of a global study. So we see that several countries are like India, right? And the question is, why are they like that, right? And so what we have done is address, the role of BCG and the fact that they don't have flu vaccination, and that's where the correlations are.
So there are plenty of questions for you, Professor Nandi, on the text. Actually, people are watching on 3 different sources. So the question is over here from somebody from JNI Nova, that various news reports I have heard that African Americans are more infected with COVID-19 than Americans in the US. Why is this so?
Yes. That's a very good question. Unfortunately, you also have comorbidities. In that particular community, unfortunately, you again have higher amounts of hypertension, diabetes. Their living conditions, they are also very poor. They live together. So there are.
Hello, yeah. So there is another question from Srinivas S. The question is there any correlation between viral load and BCG vaccination?
Okay, so viral load we have not. Because again, the data has to be there for us to take a look at. But that, I think, viral load is something which is not very easy to do. Because the only way now you're testing is looking at CT values, right? And so if the viral load was high, you would have to correlate it with CT values, which is currently not being done. So I think unless that is done, we'll not be able to answer the question.
Okay, so another question is What if we take flu shots having protection against different strains, not SARS-CoV-2? Don't you think we can get protection against COVID-19 as well due to cross-antigen defense mechanism?
So, in fact, what the study says is perhaps not a good idea to take the flu shots. In fact, that's what our analysis would suggest. It's better to be a little bit dirty, get exposed to a lot of things, and that boosts your non-specific immunity. That's what it seems like.
Okay, so there is another question, and let me tell you that people around the world, they are watching this session.
What I would urge you is to go to the preprint and take a look at the preprint, because whatever I have said is pretty much out there, except for the two newer pieces of data regarding the kinetics and the mortality aspect, which we will have to include at some point. It's out there, so you can certainly read about it.
Okay. So there's another question. It is about, is there any data suggesting that BCG vaccination and seasonal viral infection lead to decreased AC2 expression?
No, so again, this is something that one needs to look into. I'm not aware of it, but you would have to take biopsies. You would have to do it. It's not all that trivial. But yes, it's an important part, and I think it needs to be done. I mean, these are some of the things in the future plans. This is something that we have listed.
Okay, so can passaging of the virus in the population be attributed to the delayed peak in the countries that are BCG flu positive?
Okay, so so one of the thoughts is, as the virus, enters and goes around, that virulence is going to be decreased. We need to see whether that really happens or not. But a lot of people feel or expect that might happen. But we'll just have to see. What we need to worry about is, some other mutant strain comes along and which takes over, which would be a major problem. So let's keep that in mind. Let me also mention that there are other factors. Both US and India saw the virus, or the first one, at pretty much about the same time. But if you see, the US response in this case could have been a lot better. India's response was much better, and so therefore we have slowed it down. So you have governmental intervention also playing a very important role over here. So these are things that which affected the spread of it. You also have, as senior centers, a lot of senior centers over there, and they are a very susceptible population. And once it enters, it goes around, right? So you have senior centers being affected, you have prisons being affected, you have slums in India being affected. So wherever people live together, the chances are much higher. So there are these aspects to it.
So Prince Gender from ISR Mohali, sir, you mentioned about countries with high flu cases have low COVID-19 cases, but in case of US, have highest flu deaths every year. So what possible explanation for high COVID-19 cases?
So because what we have done is, that's exactly the point, that we have sort of correlated it to the vaccines, because they have the maximum amount of flu vaccines that are given, and unfortunately they've been given to the susceptible population. So that's the correlation.
So Mahi Sharma, he asked, overlapping of viruses as dengue and chikungunya during monsoon can assist in increased pathogenicity of COVID-19 in India?
Again, I think it's a good. It's another question that needs to be addressed, and people have to do it. But we have not looked into it clearly, and so I will not be in a position to answer that. But that's why I mentioned that there are other diseases that one needs to look into and then try and see. But we have not done it, so we have just stuck to these two, and then try to link it to the observations that we see, and try and see if they make sense.
Okay, so This is Anuj Bhardwaj. After such an exhaustive comparison across data, do you think India has not yet encountered with the peak of COVID-19, or its adversity is controlled in India?
So, as was shown that you see, um, incidences are going to go up higher. And that's because it's entered now and it's going to go around. The question is about mortality over here, and I suspect the reason why things have been allowed to open up is because people have also realized that mortality is not that much. So at some point you need to think whether it's like most of us have had some sort of flu, some sort of infections before, and most of us are okay. But unfortunately, there are some people who are more affected. And I think people who take all these decisions have to weigh in mortality, have to weigh in economic losses, and then decide on how to go about doing it. I suspect what they did initially was to see how it would work, and they have realized that it is perhaps less, the mortality is less, and so they have tried to restart the economic policies by allowing people, by reducing lockdown measures. I suspect that is the thinking. As I said, clearly, people, are doing this with some reasoning over here. So my job was to try and understand what are the factors that could be contributing to it, and using whatever data is available, and that's what we have tried to do. Let me also mention that even while we take India as a single entity, there are some parts of India that are much more affected than other parts. And most of you are aware of that. Maharashtra, especially Maharashtra, the Bombay-Pune region are much more affected than, let's say, Kerala. So again, one must try and understand why, what are the factors over here. So clearly, there are issues with interventions, with healthcare systems, monitoring, isolation. So these are important aspects. You just cannot ignore those.
Okay, this question is from Saudi Arabia, Bashu Chen. The question is it possible to play with E protein of the virus to make it attenuated and use it as vaccine?
I'm sorry, I'm the wrong person to be asked this question. I don't know. I don't know. I don't know the answer to this question because people who are vaccine experts are in a much better position to answer that question.
Yeah, one can play with different proteins and see.
I think that is happening. So we'll have to see what finally ends up.
Exactly. So that the virus, first thing is that virus should lose its virulence. Then only you can think of using it as a vaccine.
That is where getting vaccination strategies which we'd be able to generate good neutralizing antibodies are going to be important over here. So I think, but the other important part is that that they should not, result in antibody dependent enhancement of disease. Which has happened with dengue and also. So safety of vaccines is also important. So you need to generate neutralizing antibodies, but it should not be. It should not cause immunopathology. So these aspects need to be studied before you finally put it out.
Dr. Dipankar, so is there any clear-cut report showing the role of antibodies as immune or rather infection enhancing?
No, so I thought in dengue you see that, or chikungunya.
No, for COVID, yeah, for dengue.
Not that I'm aware of. Not that I'm aware of.
Okay.
So, but I think see what often also happens. One of the questions is why is herd immunity not working all that well? So herd immunity is not working well because primarily majority of the cases, about 80% of the cases, develop a mild infection. So when you have a mild infection, you're not going to generate that much of an antibody response, and so so protection is less. So that is why the idea is that you would have a vaccine which would generate a good robust neutralizing antibody, and that would protect you, and that's why you use the vaccine approach to generate or to improve your herd immunity.
apparently, cytotoxic T cells also have to play a very critical role, actually.
It's possible, but yeah, but I think most of the vaccines have sort of relied on good antibody response and generating neutralizing antibodies to sort of to take care of it.
We have many more questions, Professor Nandi. Because of shortage of time, I'm really sorry we cannot take, and you can write to Professor Nandi.
Yes, sure.
Okay, so now we'll take only last question. This is a question from Dr. Rashmi. Is there any correlation between dengue and COVID as dengue season is approaching? Already we have addressed this question.
Yeah, I've tried to answer that.
Right. So similar question we have addressed. So thank you very much, Professor Nandi, for a very nice talk. And let us give him a big applause.
Thank you.
It was really a very good talk. So he's available on his email. So please, you can now contact him. So our next speaker is Professor K.N. Balaji from same institute, Indian Institute of Sciences, Bangalore. So we welcome you, Professor Balaji.
Thank you, Professor Javed.
And Dr. Balaji is again a very renowned scientist. He is an immunologist and has contributed very significantly in unraveling the implication of host signaling pathways and associated genetic epigenetic determinants in pathogenesis of tuberculosis. And he has been honored with the highest award of this country, Shanti Swarup Bhatnagar Award. He is also recipient of J.C. Bose National Fellowship and many other awards. So today we'll be talking about the immunological implications of host factors during mycobacterial infections, role for distinct epigenetic transaction. So we welcome you, Professor Balaji. So over to you.
Yes, thank you very much, Professor Javed. I'm trying to still figure out the uploading. Okay.
Yeah, you have done.
Can you see it?
Yes.
Okay. First of all, I would like to thank Professor Javed as well as organizing committee for giving an opportunity to present here. It was really exciting to listen to a morning session with Professor Doherty, as well as excellent talk so far. And unlike many of them, I'm just going to present about two stories, which is essentially I often jokingly call it as a basic benchtop experimental data. So the broad title today is essentially looking at immunological implications in terms of host mycobacterial interactions. And I'm going to talk about, sort of a role of distinct epigenetic factors, and specifically two of them, how they influence some of the studies what we have been looking for the past few years. Now, this slide is somewhat redundant for all of you, but having said that, let me put this way, that MTB pathogenesis is very complex. Professor Javed knows it extremely well on this. And in that entire, I would call it as a big pie, we take a small piece. And in that, we look at, how does MTB sort of exhibits survival strategies. And we study by modulating signaling pathways along with regulation of or the modulation of host epigenetic factors, which in turn regulates the host gene expression. That's as simple it can be. So epigenetic modifications, all of you know what they are, but generally they're considered, many of them are stable, many of them are reversible as well as heritable. And usually the modification which many people talk, which is heritable and stays for much longer, is DNA methylation, as well as histone methylations, as well as now many non-coding RNAs come into picture, miRNAs, link RNAs, etc., etc. Now, past at least about 5 years or so, we are looking at some of the histone modification which are interested, and of late into non-coding RNAs also. Now, in this, I'm going to talk about, we have studied a bunch of them. In this, I'm going to talk about two factors. One is called JMJD3, and another is, can you hear me?
Vacuum. इससे वो लगाओ ना, इससे तो तमाम धूल उड़ा रहे हो.
Excuse me, can you hear me? Hello?
We can hear you.
Okay, so can I continue?
Definitely continue.
Yes, so JMGD3 and SIRTUN6. So we're going to talk about two aspects of it. One is looking at so-called lipid cell formation, or they call foam cells. Another is accumulation of cholesterol. Now, the lipids, all of you know, is one of the major class of macromolecules and very, very important for the host from variety of aspects. Now, in one of the aspects is which is important from the current today talk is actually they play a very important role in terms of host pathogen interaction. For example, it's well known, for example, pathogen docking requires many a times lipid invasion or uptake or phagocytosis required. Persistence as well as replication within the cell requires, as well as escape from the cell requires lipids. So in that way, there are this is, if you look at any classic biochemistry book, you can study lipids, what all type of lipids one can come. And in that case, add it to lipid derivatives which come from metabolic pathways, and they lead to lipid signaling, and they regulate the inflammatory process very well. So that's a classic, well-known textbook material. But of late, lipids seems to have very, very important role in terms of the host mycobacterial interaction, and that alteration, if any alterations are there, would have a consequence in terms of the pathogenesis. Now, there are various studies on that. If you take, the top half, if you look at a fatty acid derivative, for example, one of the products which you often get is something called LXA4 or PGE2, which are the products of something called arachidonic pathway metabolism. And they have various immunomodulatory properties. For example, prostaglandin E2 plays a very important role in multiple ways. So, and these actually directly and indirectly regulate the cell fate as well as expression of various cytokines. For example, if you look at it here, the fatty acid, for example, induction of LXA4 induces necrosis. For example, if you have a lot of cholesterol, that regulates phagosomal maturation. So thereby it has consequences, what happens in case of infection and the progression of TB into picture. So this is where the terminology comes into that, whether there's something called host-directed therapeutics. In this sense, whether we can modulate any of the host-derived metabolic pathways or metabolic products to our help so that we can regulate the sort of a disease condition. In particular case, in case of TB, a lot of pathways are known that may have a role. A simple example, for example, here, TB, like any other infection, causes a lot of pro-inflammatory environment. For example, TNF-alpha is produced a lot, VEGF is produced a lot. And any of the molecules which can regulate this expression would have an effect on that. So, for example, these two molecules, enambrel or this, which regulate VEGF, is extensively used in case of arthritis. So, essentially, many of these drugs, whether here or here or here, all can be repurposed to regulate the pathogenesis of TB. And of course, it has various effects for this. So this is a sort of a comprehensive review it has appeared in some time back in Frontiers in Medicine. Now, what is the focus of our talk? The focus, what we have been looking at is, we're looking at host factors that are responsible for dysregulation of lipid levels upon infection of TB in macrophages as well as dendritic cells. And if there is one, so what are the molecules that are regulating that? So if we can identify those molecules, maybe we can get a therapeutic targets. They can act as therapeutic targets. And so hopefully we can regulate the TB pathology. Now, to come back to why these so-called foam cells are lipid-rich macrophages. Interestingly, in case of many diseases, but TB is sort of well known by now, that macrophages are recruited to the site of infection. We know that especially so-called TB granulomas. There are various studies on it. They are well characterized in case of mouse. They are fairly organized, which is not the case in case of human. But however, the macrophages come to the site of infection, and some of them change, and they become highly sort of rich in lipid droplets. So the lipids are sort of formed in the form of a droplets, and they turn into something called, foamy macrophages. They're called foam cells or foamy macrophages. Now, the lipid droplets, contain a lot of inflammatory mediators, and as well as, enzymes metabolizing immunomodulatory molecules like we talked about PGE2, lipoxins, etc., and eicosanoids. And also actually regulates all of intracellular signaling with this. The argument has been for some time that in addition to these properties, these lipids also provide nutrition source, and it can sort of provides or at least generates an environment which is required for mycobacteria to persist, and stay for a long time to come. The question is, so what we were interested is how exactly this happens. So we started to study about 4 or 5 years back. So here is a classic example of it. So we take, these are mouse macrophages. So you infect them with virulent strain of TB. This is a case of Esterisone RV, or a clinical virulent strain called one of the Jalma strains right here. So when you take these macrophages and stain for lipids, you can use a molecule called BODIP, which stains green color. You can see nicely, green color formation in these macrophages upon infection, and red color here is actually virulent TB. So essentially what we showed, which was shown by earlier already, that saying that, if you infect the macrophages with TB, whether you do it in vitro or you take the lung section, which is from aerosolized mice, you take the mice aerosolized with Esterisone RV, and you take the lung section, stain for this lipid staining, you get nicely formation of these lipid bodies, as you can stain with BODIP, and you can quantify by various means, by confocal, by FACS, etc., etc. So one of the ways a lipid accumulation happen in any cell is simple process. In the sense, A, the lipid synthesis gene expression might go up. B, there could be import of the lipid from outside. It can come from, from the circulation or from the medium. C, that the efflux of lipid could be stopped. Means there's no export of the lipid. So all these three can contribute to the lipid accumulation in a cell. So we asked this question that, is it possible all these molecules are expressed? Yeah, here is a one in vivo experiment, for example, here. You can look at here, all of them, that if you look at CD36 ADRP, which are all involved in a sort of a lipid synthesis genes or lipid import genes, all of them are nicely expressed in wild type mice when you infect RV, Esterisone RV, means virulent TB. But if you use a TLR2 null mice, means where there's no TLR2 receptor, which is a major receptor for Mycobacterium tuberculosis, that effect goes away. So essentially it is a TLR2 mediated event, and this gene expression goes away. So when we came for this, so we wanted to know how these gene expressions are going up and what is the response for it. So in that case, we came to one of the epigenetic factors. Now, as I said, one of the modification that regulates the gene expressions is histone modification. Now, in histone, especially Hes3 histone, there are a series of lysine residues which has been identified, which is lysine 4, lysine 27, lysine 9. They can be methylated. So whenever there is a methylation, either the gene expression can go up or gene expression can go down. So in this case, what we looked for, a removal of a repressive marker. So what happens in the case of many gene expression, the histone 3 at lysine 27 get methylated, trimethylated. That is usually considered as a repressive marker. So whenever that happens, this gene expression goes down. And it is well known in case of Mycobacterium infection. We have shown it, others have shown it, that one of the way the class 2 expression goes down in mycobacterial infection is by this histone modification, a repressive histone modification, thereby gene expression goes down. So in this case, we started looking for a molecule that actually which can remove this repressive marker. So this molecule is called JMJD3. Some of these names which I'm going to mention is interesting from the development point of view. That JMJD3 is nothing but Jumonji domain containing protein. So essentially that is actual demethylase which removes the repression, the trimethylation mark from H3K27. When it is removed, the gene expression goes down. So this is what it is. So the JMD3 comes, it removes this repression mark. When the repression mark goes, gene expression goes down. The question we asked was whether this JMJD3 expression level is going up. The answer is yes. If you take virulent strain of TB or a clinical strain, this JMD3 expression goes up either by RNA level or by protein level. And if you use a mice which are TLR2 null, this expression level doesn't go up. It means the mycobacteria has to interact with TLR2 in order to induce this demethylase, which is JMJD3. So we went on to show that JMJD3 is required for this foam cell formation. The standard approaches you all knew very well. If you take the macrophages and transfect it with this siRNA for JMJD3, the mycobacteria ability to induce this foam cell formation completely reduced. As well as the ability of this, how the foam cells are formed. I said the lipid synthesis gene expression goes up. So this gene expression induced by mycobacteria is completely goes down if you knock off JMJD3 using siRNA. So now the question is, yes, okay, we know now JMJD3 regulates this lipid formation or lipid accumulation in macrophages. Now how exactly JMJD3 works? See, one of the ways JMJD3 works is for any molecule. If you have a regulatory molecule, the regulatory molecule expression can go up. If its negative regulatory expression goes down. It's simple as that. So we know that one of the molecules which regulates the JMJD3 in a negative fashion is called MINT. So the MINT here, what we show that actually the MINT level upon mycobacterial infection goes down. You can see here nicely by Nice Western. So when the negative regulator of JMJD3 goes down, the MINT level, when JMJD3 expression goes down, the MINT level, which is a negative regulator of JMJD3, goes down. Okay, that's a good thing. Now the MINT is an interesting molecule. The MINT is actually an RNA binding protein. MINT is an interesting molecule in the sense the MINT expression is regulated by another protein, which is an RNA binding protein. So here is one. See, for example, MINT negatively regulates JMJD3. We show that MINT level is going down. The way MINT level is going down is because of another molecule, an RNA binding protein called Musashi. So what we went on to show actually is Musashi actually binds to MINT in a 3' UTR, and that downregulates eventually to JMJD3. What is the proof of this? Here is the expression. If you take macrophages infected with H30s and RB, this is and transfected with a non-targeting siRNA, it has no effect. However, if you target this Musashi siRNA, because remember Musashi regulates the expression of JMJD3 in a positive manner, the foam cell formation is reduced. So this clearly says that this story that Musashi regulates positively JMJD3 expression by regulating this MINT expression. Now having said that, so does it have any other effect? This Musashi as well as JMJD3 has any effect? Answer is correct in the sense both Musashi and JMJD3 not only regulates the lipid synthesis gene expression, there's also regulates both M1 versus M2 cytokines level in a differential manner. So this brings to this point, this is the first story is that mycobacteria regulates the expression of the lipid genes, thereby contributes the accumulation of lipids in these macrophages. They turned out to be called foamy macrophages. That require a signaling pathway, and that requires the participation of a methylase called JMJD3. Now, having said that, now the question we have is, so what happens? So it's not just lipid. What are those lipids accumulate in this? One of the lipids accumulate in case of foamy macrophages is cholesterol. Now, cholesterol is a very interesting molecule. All of you know how important it is. And the MTB, by various means, triggers the accumulation of neutral lipids as well as cholesterol. Now, it's very important for, cholesterol is very important for MTB pathogenesis, whether you do in vitro or in vivo, for its survival, dormancy, antibiotic resistance, etc., etc. So we want to know whether any epigenetic factors are playing a role in the accumulation of cholesterol in these cells. So here we went back to our story we were doing. So we went for two molecules. Now, in a typical, simplest a textbook pictorial depiction of how does epigenetic factor works, this is where it is. Here is a histone. Now, histone has very interesting molecule, and it has various lysine residues, and the lysine residues either can be methylated, acetylated, or this methylation or acetylation can be removed also. So they are called as erasers. Now, those who put methylation or acetylation, they are called writers, and those we already know methylated or acetylated as seen by a molecule called readers. So we were interested in two such molecules. One is who does the methylation to histone residue, which induces gene expression, and we also were interested in another modifier which removes the acetylation. Means it's the ratio of methylation to acetylation can regulate the gene expression. Now the methylation what we are looking at is an activatory methylation. Essentially a lysine 4 methylation at H3 with this. So we went for two molecules, something called G9A, which is a bifunctional methylase, and a deacetylase called sirtuin 6. Now sirtuin, many of you know, there are a class of them, and we focus on sirtuin 6 for a variety of reasons. So here is a classic example. If you take macrophages, whether in vivo or in vitro, you stain for Philippine staining. Here it is, which stains for cholesterol. You can see upon TB infection, you can see nice cholesterol accumulation. In case of RE infection, whether it's in vitro or in vivo. Now we ask the question whether these two molecules, whether G9A, which is a methylase, or a deacetylase sirtuin 6, play a role in cholesterol accumulation. Now, the question is very simple. We went for a very simple-minded approach. If you look at the expression of G9A and sirtuin, both are induced upon TB infection, whether in macrophages or in vivo lung samples. However, the global level of this methylation at, K9 can be monomethylated, gets activated gene, or K4, did not change, or the global level of acetylation did not change, which is not surprising. Now, is the same story is true whether you take in vitro macrophages or in vivo? Now, so we asked the question whether G9A, SIRT6 contributes to cholesterol accumulation. So we took in the macrophages, which is peritoneal macrophages or bone marrow derived macrophages. Now here it is, when you use an siRNA for G9A or SIRT6, the accumulation of cholesterol reduces a lot. The story is true similarly. If you take BMDMs, you take from wild type mice or a sirtuin 6 knockout mice, the accumulation of cholesterol reduces a lot when you use a sirtuin 6 knockout mice. But there is an issue with sirtuin 6 knockout mice. The reason is it won't survive beyond 4 weeks. So that's one of the reasons we have not been able to do in vivo experiment with sirtuin 6 knockout mice. But we have done experiment with sirtuin 6 heterozygote mice to see some effects there. Now, one of the way I mentioned earlier for lipid synthesis, it applies for cholesterol also, is that any cell can accumulate a cholesterol by 3 mechanisms. One, there could be increase in the gene expression of cholesterol synthesis gene. The influx, the importers expression might go up so that cholesterol can be taken up. Also, the exporters, which are involved in the efflux of cholesterol, can go down. By all the 3 mechanisms, you can have a cholesterol accumulation in the cell. So the story what we showed that when you infect with TB and all the cholesterol synthesis gene and influx genes are going up, when you use siRNA for G9A, it goes down very nicely. Interestingly, when you use here, when you look at here, that efflux gene cholesterol exflux from the cell goes down upon TB infection, which is ABCA1, ABCG1, which are transporter. But when it goes down, but if we interfere this SIRT6 expression during that time, the efflux gene downregulation by TB is reversed. So
you are left with 5 minutes. You are left with 5 minutes.
Sure, I'm done. Okay, thank you, Javed. So essentially what it says is the story that both G9A and SIRT6 are playing together. So the story is here also same, whether you take, so is that case how it is possible? So G9A, remember methylase, it has to come to the promoter of these genes, bind to them and methylates the histones at H3K9. So we did a standard ChIP-EC chromatin immunoprecipitation assays. You can see that G9A comes to the promoters of cholesterol synthesis gene. Whenever they come there, the activatory methylation marker goes up very nicely with this. And same story is true in SIRT6. SIRT6 comes to this efflux gene. And when it comes to efflux gene, it removes the acetylation marker. So when it removes the acetylation, the gene expression goes down. So the efflux genes are downregulated with this. And we went on to show by various in vivo experiments, whether using, G9A inhibitor or SIRT6 heterozygotes with this. And of course, at the end of the day in TB field, you have to show it has effect on CFUs. Yes, answer is true. So inhibition of G9A and SIRT6 actually regulates the, sort of down-regulates pathology a lot, and as well as it regulates CFU. So finally the story, the last but one slide, is we went on to show these two molecules, G9A and SIRT6, two epigenetic factors. One is a methylase, another is a deacetylase, play a role in cholesterol accumulation in case of TB infection. And what happens when you accumulate cholesterol, it regulates what of many things, and one of the gene expression is bunch of antioxidant gene expression goes up, and that's how it plays a role in pathogenesis. And finally, I would like to mention, just these two molecules are not alone. There are a bunch of molecules are there which are going together, all of them going together in a given site, opens up the genome, and then does this expression of the genes regulation with this. So at the end, I wish I can say that I have done all the experiments, while the majority was done by Praveen. And of course, Salik is continuing with this. Of course, I thank various funding agencies and of course, the beautiful IISc, which I am very thankful to this. So I thank once again to Professor Javed and organizers for giving me this opportunity.
Thank you very much, Professor Balaji, for this wonderful talk. And there are a lot of questions, and let us give first opportunity to the people who are viewing you through Google Meet. So if there are any questions, please, they are welcome.
Dr. Javed, I would like to ask a few questions.
Sure.
Good afternoon, Dr. Balaji. I am Pawan Gupta, working at IMTech Chandigarh.
Yes, Dr. Gupta.
So, a few questions that I have for you. What is the translatability and druggability of these targets that you have come up with, the methylase and the deacetylase?
Well, the deacetylase, which is especially in SIRT6, I have a colleague in my department who is collaborating. He is involved in a lot of molecule developments and other things. So I can't say much about that. But G9A, essentially there are some phase 1 sort of preclinical trials were done on it. But how much it has gone ahead, I'm not very sure about it till now.
So are they able to act alone and bring about changes in the bug survival in vitro and in vivo? Or are you thinking of clubbing them with the frontline drugs for the purposes of reducing the duration and the dosage of treatments?
Yes, actually, we are performed some of the experiment with the frontline drug with a sublethal dose to see whether it's more effective. There seems to be, but there are a couple of problems. One is that we are not able to get G9A knockout mice. Okay, that's number one. Now, number two, certain 6 mice, null mice won't survive beyond 4 weeks. So of course, we are using heterozygotes for that. So that's all I can tell. Of course, we have repeated some of these experiments with human DCs as well as human PBMC as per the reviewers. Remember, these are the questions. So we see a similar effect. So I am not too sure how much I have answered your question, but I think with the frontline drugs, it works nicely.
And then the cholesterol that was being referred to, is it the total cholesterol, or you have also specifically looked for the oxidized cholesterol?
No, we have not looked at the oxidized cholesterol. In fact, this was one of the suggestions with EMBO meeting. See, what we went on with a very simple-minded approach. I believe it as jokingly, I suggest my student as a carpet weaving experiment. We know the start, we know the end, so we are trying to fill it up in between. So we know that cholesterol accumulates.
Very much. I think it was a virgin territory, and a landscaping was required to begin with. It's a wonderful piece of work altogether.
Okay, but so there are many questions, and I'm going to read for you. These are text for you. So one of the question is that can host drive danger molecules and purinogenic signaling with a particular focus on host microbial defense and infection-driven can drive chronic inflammation?
Well, I think it's very difficult to answer the question, but in my gut feeling, if you take, yes. In fact, the heterogeneity in the response, what we get with many people, even with TB infection or this, if I'm not incorrect, is about microbiota, right?
No, it's not microbiota. It's host drive danger signals.
Oh, host drive danger signal. Oh,
and
okay.
Purinogenic signals.
Yeah, there are stories on purinogenic signals and TB. We had started some studies long back, but we didn't continue that. Yeah, that could be, but we have not addressed those in our studies. Yeah.
Another question is, can the mechanism be regulated by histone H4K9Me3 and K4AC poised expression?
H3K9 and?
This is H4K9Me3 and K4AC.
Yeah, actually, at the same time, we don't know. See, the way we went ahead with this was interesting. So what we said was actually certain 6 removes the acetylation marker. When the acetylation is removed at K9, the G9A comes and monomethylates them in case of synthesis gene, but dimethylates them in case of efflux genes. So what happens with that is the monomethylation and dimethylation can have different results. Monomethylation, many a times it augments the gene expression, many a time dimethylation, whether symmetric or unsymmetric, depends on what you're talking about, can inhibit gene expression. So essentially, it's when we did a sequential chip, certain, especially efflux genes, they only touch efflux genes, removes the acetylation marker, then G9A comes and further represses it so that it constantly represses, so that cholesterol don't go away.
Okay, so there is a question for you, and this is Musashi mediated mRNA translation control be effective to control COVID-19 and be a therapeutic agent for combating COVID-19?
I'm sorry, I really don't know. See, the way we got into this, this is an address to the students. I would like to say this. This was started by a student who was interested in genetics and who did some genetics on Drosophila long back. Now, all these names are derived, and all our papers derived from development. It is nothing to do with infection, nothing to do with this. So we figured out Musashi regulate JMGD3 by a paper in development journal. And I have no idea about Musashi's role in any other viral infection. I think there could be some, but I'm not aware of it.
So there is another question. This is regarding if knocking out of CHO-D genes, which is coding for cholesterol oxidase in H37RV, make it attenuated, or it can be used as a vaccine then?
Well, what we are talking is cholesterol synthesis gene from the host.
But here he's talking about bacterial.
No, I have no idea. I have no idea. Javed, you should be knowing this. I'm not aware of this.
It's going to kill the bacteria. Bacteria will not survive anymore. Okay, so another question for you is, what is the role of efflux genes in order to develop or control COVID-19 pandemic this year? I'm unable to correlate this talk with the immunological issues relating to SARS virus. This is from Srishti Singh.
Look, let me put this very clearly. I am not a virologist by expertise. I do not know the lipid accumulation that much in case of COVID-19. So it's very difficult for me to answer that question.
Okay. So did you check for expression of genes related to metabolism of the accumulated lipids?
Yes. In fact, that is where we are having some issue. Actually, when we presented this data in a EMBO meeting, in fact, one of the pioneers in this field called David Russell had a different ideas about it. So what we found that initially we found that the synthesis genes expression is going up, the import of cholesterol genes going up, of course the efflux gene expression going down. That's how cholesterol gets accumulated. But when we did an RNA-seq data as well as ATAC sequencing, what it shows is the promoters of all the genes opens up with a correspondence expression pattern on transcriptome. We can match both. We didn't see many synthesis gene expression going up, even though in vitro we can find it. But import genes were going up. So very clearly, we agreed with David Russell's view that there is a lot more import is happening with cholesterol, but efflux of the cholesterol is going down. So obviously the metabolism genes of cholesterol is regulating, but if I'm not incorrect is whether the degradation or the metabolic pathway of cholesterol downstream we have not examined.
Parul Bhatt asks you that have you observed any cholesterol accumulation in latent infection of TB?
Latent infection of TB. Look, we have done so far only the mice experiment. We went for 56 days, even beyond that. And we see the cholesterol accumulation. There's no question. But that is not a latent model.
Naveen Changundundla asks that, is bacteria regulating the foam cell formation or is host reacting to it?
No, actually, I think it's a bacteria which is regulating it, because if you take TLR2 null mice, bacteria cannot do it. It's just not TB. Even other bacteria can do that. If you engage some of the toll receptors, you can form foam cell formation, but not as great as some of the microbial species.
So,
hello.
Another, yes. Is a question from Durba?
Yes, sir.
Okay.
I'm Dr. Durba. I'm working in CBME Europe. My field is lipid-induced insulin resistance. So I have a simple question for you, because in case of the obesity, the foamy macrophages are very usually it happened in case of the women when obese patients. They usually having a lipid-laden macrophages there. So from your presentation, what I've found that the tuberculosis actually infection that actually helps in the cholesterol deposition, and so might be the tuberculosis having any connection with obesity. But there's also some papers out there that in obesity there is a lower risk of tuberculosis. So can you comment on that?
Well, you ask a very important question, because we have been looking, of course, there were few papers came, that how does these macrophages are different from atherosclerotic plaque macrophages, for example. is similar to what you asked the question. The macrophages in case of obese persons. In fact, only you can induce actually foamy macrophages. You don't require a mycobacteria for that. You can induce by adding, lipids, right? You can add, you can make them. But interestingly, the total gene expression, if you turn these macrophages into foam cells and do a transcriptome profile of many genes, cholesterol genes, other biosynthetic gene, metabolic gene, there's a distinct difference between those versus, MTB induced ones. If you do an RNA, for example, RNA-seq, you will find a distinct differences of them.
Okay.
That's number one. Number two, the general belief, of course, with the exception of 2 papers, that the bacteria requires these, lipids for its own survival, growth in granulomas or wherever the niche they are sitting there. Okay. So in that case, the people who are having lots of these lipids in various cells should be sort of more prone to TB, but I'm not very sure whether that goes that simple as that. Because remember one thing, cholesterol induces a lot of the other genes. If you just add cholesterol, the gene expression changes. And in case of TB, there's a lot more import of cholesterol into cells. And if you look at a lot of antioxidant gene expression, other things, cholesterol induces a lot of gene expression, interestingly. So it's the balance. So I'm not very sure how capable I'm able to answer your question.
Thank you, Dr. Balaji. It's a very nice presentation.
So there's a question from Hilal Bashir. Did you try to find role of this histone modification in adipocyte differentiation, as these cells are potent source of inflammatory and have good relation with M2 macrophages?
No, absolutely. We didn't do. In fact, this is one of the long-term thing we did not do. It's an interesting question. Thank you.
Okay. So there's another question for you from Dr. M.F. Khan. How can we identify unknown proteins expressed from cDNA sequence engineered in E. coli vector?
Well, I think I would skip that answer if you don't mind. Yeah.
Okay, there's a question. Anuj Bhardwaj Bandral. Is there any direct relation between cholesterol metabolism and tuberculosis progression? And what's the status of COVID-19 vis-a-vis obesity? The first one is more relevant to you.
Yeah, I think there is quite a bit of stories. If this cholesterol metabolism, as previous one before asked the question, the obesity, TB susceptibility, etc., etc. There's quite a bit of data. I'm not very clear whether it is clear-cut demarcation, whether people with high fat content actually progress towards TB versus not. Because if you see the spectrum of patient, you get all kinds of ranges. So I'm not very sure about that. So second part, I'm sorry, I don't have expertise on that.
Okay. So any more questions? So if there aren't any questions, we would like to thank Professor Balaji for really a wonderful talk. Let us give him a big applause. So it was really a very good talk. So thank you, Dr. Balaji, for this wonderful talk. So we now invite our next speaker, Dr. Pawan Gupta.
Dr. Javed,
thank you, Professor Javed.
Okay, thank you.
I hope the video is all right.
Just let Dr. Balaji leave. Yeah, now you are in. Can you upload your presentation? So Dr. Pawan Gupta is a very highly emerging young scientist of India. Let me introduce him. Right now he's working at as a senior principal scientist at Institute of Microbial Technology, Chandigarh. and his main interest is on molecular immunology, metabolic diseases, host pathogen interactions, switchable therapeutics, and all of these ultimately he tries to correlate with mycobacterium tuberculosis. Dr. Pawan Gupta has published many good papers in journals like Journal of Immunology, Biological Chemistry, and in very high impact journal like Autophagy. So, and let me tell you, he's a very silent, quiet worker. So many of young emerging immunologists of this country we really don't know. So I thought to introduce all these immunologists through this forum. So we welcome you, Dr. Pawan Gupta, and today he's going to talk about an accord of nuclear receptor in chronic inflammatory disorder. So we welcome you, Dr. Pawan Gupta.
Dr. Javed, I hope that the slides are visible.
No, your slides are not visible. Can you please again load? Quit it and again load. No, your slides are not visible yet. हाँ बोलो कौन फिर Dr. Pawan Gupta, we can't see your slides. Hello.
Yes, I'm trying it again.
आशु आप इधर से कर रहे हो आपका सारी है ना प्रेजेंटेशन
I hope slides are visible now, Dr. Javed.
No. So shall we load from here?
Maybe I'll try my laptop, but in that case, I would not be visible, but I think that is all right. Just give me a minute.
Okay. So shall we load?
Just hold on a sec.
Ashu, can you guide him?
I hope I'm audible, right? I hope
You're
I'm audible.
Yes, you're audible. हाँ तो कितने लोग आएंगे?
Can you see the slides now?
ठीक है चलो आ जाओ 1:30 बजे.
I think I'm not able to go to the option of share screen somehow.
So what will you do? We are loading from here.
Do you have my slides with you?
Yeah. Oh, we don't have slides?
I think now it will work.
See, probably you have to quit all your windows. And your slide should not be in edited mode. Open your slides and then you share. Yeah, good. Now we can see you.
Wonderful.
Wonderful. Right time you have loaded. Good.
So, a very good afternoon to you all. A short introduction to myself. I am Pawan Gupta, working as Senior Principal Scientist at MTech, a constituent laboratory of CSR at Chandigarh, India. The title of the presentation is An Accord of Nuclear Receptor in Chronic Inflammatory Disorder. The content includes the word meaning definition of druggable nuclear receptors and how they are poised to become next generation drug targets. A short introduction to chronic inflammatory disorders and placement of nuclear receptor therein, and also a brief overview of one of our research where we have posited the involvement of NR4A2, popularly known as NR1, in modulation of immunogenicity and inflammation. Now, as we all know that nuclear receptors are a member of superfamily of transcription factors, which together with their co-regulators can bind to gene promoters, the response elements, and can bring about upregulation or downregulation of genes. They can modulate processes as diverse and as varied as metabolism, development, and reproduction. Nuclear receptor can be classified into three ways. It can be classified on the basis of sequence homology or phylogenetic analysis, which reflects of evolutionary origins. They can also be classified on the basis of their dimerization ability. They can form homodimers, heterodimers, atypical heterodimers, and that has regulatory connotations to it. But by far the most acceptable classification of nuclear receptor happens to be on the basis of ligand binding. Now, this has both translational and functional connotations. On the basis of ligand binding, the nuclear receptors can be classified as the endocrine receptors, the adopted orphan receptors, and the orphan receptors. Endocrine receptors, as the name suggests, they glean their ligand from endocrine sources. And most of their cognate ligands happens to be high affinity hormonal lipids and vitamins. They could be glucocorticoids, mineralocorticoids, the sex hormones like estrogen, testosterone, progesterone, and then the vitamins like vitamin D and the retinoic acid, the physiological forms. And then we have the adopted orphan receptors. And as the name suggests, that they were initially discovered as orphans, meaning that we knew of the receptors, but the ligand information was not known. And it was only over a period of time that the ligands got deciphered, and that's how they drew the moniker of being adopted. So the orphan receptors were adopted once their ligands were known. And here their cognate ligands happens to be low affinity nutritional lipids, or dietary lipids, and metabolites. Some examples include oxysterol, bile acids, omega fatty acids, xenobiotics, and heme. And then we have the orphan receptors for which ligands are yet to be deciphered. Now, there is a very peculiar and very stringent structural organization to all the nucleoreceptors. So there are almost like 49 nucleoreceptors which find expression in mice and 48 in humans. And all of them, without exception, have a stringent structural organization to them. They have A to F domain. The A and B domain constitutes the N-terminal domain, which encompasses the activation function 1 domain. Now, this is the region which is most variable in the nucleoreceptor sequence across the family. The AF1 domain is also prone to several post-translational modification, which is a regulatory event. And then they have a C domain, which is called as a DNA binding domain. DNA binding domain has two zinc finger motifs, the P box and the D box. The P box is important for recognizing the response element, while the D box, that is important for the dimerization ability. And then there is the D domain, which is also called as the hinge domain. This is quite a flexible domain, and this flexibility is very much required between the DNA binding domain and the ligand binding domain, for the very reason that both these events should take place without offering any steric constraints on each other. There are also reports about this hinge region containing the nuclear localization signal. At times, it also contains the nuclear export signal and the 1433 binding site. And then there is a E domain, which is a very important domain for as much as it has a ligand binding pocket, which is quite a hydrophobic cleft, and it also has a AF2 domain, which is called as the activation function 2 domain. Now, through ligand binding pocket, nucleoreceptors can sense different kind of ligands. They could be agonist, antagonist, or they could be inverse agonist or selective receptor modulators for that matter. And then the AF2 domain is very much regulatory in nature. It is also called as the transactivation domain. It is important for co-regulator binding also, and at times it is important for dimerization, particularly in cases of the atypical heterodimers. And then the F domain, which is quite variable. Now, nucleoreceptors, one has to kind of appreciate and admire the multiple regulatory checkpoints that they happen to have at the level of sensing. So there are 3 regulatory checkpoints. Ligand binding happens to be one. Then it is the dimerization ability, where they can bind as a monomer or as homodimer, heterodimer, atypical heterodimer to their cognate response elements, which could be half sites, they could be direct repeats, they could be inverted repeats, or everted repeats for that matter. So one can safely assume that nucleoreceptors can be great next generation drug targets by virtue of their good sensing ability and minimal degeneracy. Nucleoreceptors are poised to become next generation drug targets, and this can be ascribed to largely 4 attributes. Firstly, that they lie deep within. they are deep-seated, unlike their counterparts in form of surface receptors or signaling molecules. They have a very narrow effector repertoire and not a broad effector repertoire. So there is less leutrotrophy. Second, as I was telling, that at the level of sensing, they have minimal degeneracy and more specificity. Also, at the level of sensing, there is less redundancy, and at the effector function level also, there is very less redundancy. Meaning that while we know of several polypharmacological ligands and several crossover ligands that can engage multiple nucleoreceptors, but those instances happens to be only few as compared to their counterparts. And many of their synthetic ligands qualify under the Lipinski rule of 5. So, them being probable potential drug targets and their synthetic ligands being future drugs is quite likely. Now, this slide actually depicts the last 10-year landscaping of different FDA-approved drugs, their cognate receptors, and one can appreciate that nucleoreceptors over the last decade have been quite successful in positioning themselves in the big category of 4 drug targets to the likes of GPCRs, the voltage-gated ion channels, the kinases, and the nucleoreceptors. If one goes by statistics, one can say that 16% of estimated FDA-approved drugs actually act through nucleoreceptors. 1/3 of the nucleoreceptors are targets of currently marketed therapeutics, and 20 of the 200 most prescribed drug targets nucleoreceptors. Now here in this table, we have tried to put the FDA-approved drugs, their cognate receptors, and different chronic inflammatory disorders. So one can see that both endocrine receptors and adopted orphan receptors are quite successfully engaged by different FDA-approved drugs. For endocrine receptors, we have the examples like GR, ER, VDR, RAR, and androgen receptor. And in case of the adopted orphan receptors, we have the PPARs, the alpha, the gamma, the obligate heterodimer, RXR, and FXR. These are only few examples. There are many more. Now, inflammation, as we all understand, happens to be a very complex biological process to any harmful stimuli or an insult for that matter, and it could be exogenous or endogenous. Exogenous examples being pathogens and irritants, and endogenous example being damaged cells. Inflammation could be immediate and short-lived, or it could be delayed and prolonged. It could be resolving, or it could be aggravating. It could be curative to a particular insult, or it could be causal to a disease condition. Speaking particularly for chronic inflammation, it is a delayed response of prolonged duration, more than a week, could be months and could be years. And here, all the 3 events are happening simultaneously: the active inflammation, the tissue destruction, and tissue repair. And the causes could range from persistent infections, chronic exposure to toxic agents, and autoimmunity. Now, chronic inflammation can be both causal to or consequence of chronic inflammatory disease, and they could be of many kinds: cancer, neurological disease, pulmonary disease, bone and joint disease, metabolic disorder, cardiovascular disease, and autoimmune disorders, and so on and so forth. Now, this slide depicts placement of nucleoreceptor in chronic inflammatory disease condition. The red fonts over here depict nucleoreceptors reported in cancer. The notable placement of nucleoreceptors include the RARs, FXR, ESR, and AR. The blue fonts depict autoimmune disorders with notable placement of nucleoreceptors, PPAR gamma, RXR alpha, and RORC. And the green font depict metabolic disorders like RARs, PPARs, RORC, HNF4 alpha, MR, ARN, and ROB2. Now, we have been involved and we have been interested in generation of nuclear receptor expression atlas in different immune sentinels, in different disease contexts, and to different cellular subsets also. We have thus far come up with an expression atlas of nuclear receptors in MTB infected macrophages and DCs. We have correlated these expressions and done extrapolation of the findings in clinical samples. We have done experiments in human MDMs and we have corroborated the findings in PBMCs obtained from TB patients. Our immediate interest is also in coming up with nuclear receptor expression atlas in different cellular subsets of macrophages, be it M1, M2, or regulatory macrophage. Yet another attempt from our lab, which got published in Immunohorizons very recently, we have come up with an expression atlas of nuclear receptor in chronically activated CD4 T helper cells in collagen-induced arthritis model of mice. And again, we have done extrapolations in clinical samples. Overall, we have been able to come up with 3 nuclear receptors, NROB2, NR4A1, and ROR alpha, which along with their cognate ligands, when used individually or in combination, are able to curtail the severity. And then we have also come up with an expression atlas of nuclear receptor in immunogenic and tolerogenic DC. Now for this, I am going to provide a brief research overview. Now DCs, as we all understand, are quite indispensable immune sentinels which are known both for their immunogenic and tolerogenic potential. The functional heterogeneity that we have among DCs determines the balance between effector T cells and the regulatory T cells. And any dysregulation can lead to autoimmunity or chronic inflammatory disease. Now here what we have done, we have used bone marrow precursors obtained from black 6 mice to generate immature DCs. Now, in order to generate the immunogenic and tolerogenic DCs, we treated immature DCs with LPS or TGF-beta. Now, BMDCs treated LPS showed increase in the maturation marker, which was not the case in case of TGF-beta treated DC. When we looked for the functional markers, we could see increase in the cognate functional markers in case of both immunogenic and tolerogenic DCs. So to this end, we were actually able to generate functionally distinct phenotype of DCs in vitro. There was another observation that prior treatment of TGF-beta abrogated all LPS-induced increase in maturation marker or the cognate functional markers. Now, having generated these tools quite successfully, we were tempted to evaluate them for the expression atlas of nuclear receptor. For the very reason that the knowledge of nuclear receptor in DCs happens to be quite rudimentary. We know only of few nuclear receptors which can modulate tolerogenic potentials, like that in case of GR, VDR, and PPAR gamma, which are known to induce tolerogenic DCs. And then we also know of LXR, RAR, and RXR, which are known to induce immunogenic DCs. So we thought of coming up with a comprehensive expression atlas, and a lot of experiments were there, several cutoffs were put in place, and we realized that 39 out of 49 nuclear receptors find expression in different cellular subsets of DCs as compared to immature DCs. Now these included 10 endocrine, 11 adopted orphans, and 18 orphan receptors. Further, we did the regurgitation of this data by different permutation and combination, and we revealed the ones which undergo discrete changes, the ones that undergo many-fold changes. And we realized that NR4A2 and PPAR gamma, they are undergoing many-fold change in expression in immunogenic versus tolerogenic DC. Now, PPAR gamma is quite established. Its mechanism is very well known. but NR4A2 was the new guy on the block. So we thought of investigating NR4A2 for its potential functional role. So in that regard, we took the immunogenic DCs and we did some gain of function and loss of function approaches by using adenovirus assisted system. And we looked for the maturation markers, the functional markers, and also the tolerogenic markers. And what we realized that NR4A2 actually dampens the immunogenic DCs, and it also imprints tolerogenic signatures on the DC phenotype. We look for different tolerogenic markers like that of GILS1, HO1, ILT3, IDO, and retinal dehydrogenase. And we realized that NR4A2 quite unequivocally is able to induce tolerogenic potential in the DC phenotype, and for sure it is dampening the immunogenic phenotype. We also performed antigen uptake and processing ability in immunogenic DCs, and we realized that NR4A2 was able to rescue the antigen uptake and processing ability. Now, we were also interested to evaluate NR4A2 function in a disease context. Now, for that matter, we employed the mouse model of multiple sclerosis, EAE. Now, we injected BMDCs overexpressing NR4A2 or LAGZ intravenously before and after the MOG immunization at the +1 and -1. We realized that mice receiving NR4A2 overexpressing BMDCs showed less clinical severity of EAE progression when compared to mice receiving LAGZ overexpressing BMDCs. Histological analysis also revealed lesser infiltration of cells when we had NR4A2 overexpressing BMDCs. We also looked into the ability of NR4A2 in modulating the T regulatory cells and effector T cells in spleen, in lymph node, and in CNS. And we realized that the tolerogenic DC regulate tolerance in part by promoting the expansion of T regulatory cells and attenuating the Th1, Th17 responses. And NR4A2 has a pivotal role to play in there. So overall, one can safely say that our findings demonstrate that NR4A2, an orphan nucleoreceptor family member, is differentially induced in immunogenic DCs, alters LPS-induced maturation while endowing the DCs with tolerogenic potential. Additionally, when we administered the BMDCs overexpressing NR4A2, it curtailed the progression of EAE by a mechanism involving expansion of T regulatory cells while limiting the abundance of effector T cells. Now, simultaneously, we were also involved in investigating the role of NR4A2 in macrophages. We realized that whether we engage TLR2, 3, or 4 by their cognate ligands, zymosan, poly-IC, or LPS, the expression of NR4A2 gets increased to different extent. And through the PI3K AKT signaling pathway, it was actually able to bring about suppression of the cytokine storm. It was preventing inflammation from going cockeyed. And not just that, there was more to it. It was actually able to upregulate the expression of a very important gene, which is very much important for wound repair and healing, arginase. It converts arginine to ornithine, polyamines, and thereby it is important for wound repair and healing, and also that it is important for macrophage polarization to the M2 phenotype. So compelling was the evidence regarding NR4A2 in different disease contexts and in different cellular subsets. So we thought of coming up with a fact sheet of NR4A2 for as much as the ligands that are there in place and important regulatory functions of NR4A2. Now, NR4A2, unlike other nucleoreceptors, have a very compromised and I would say that a very defunct ligand binding pocket. It has very few hydrophobic residues, lot of polar residues. But despite that, the number of synthetic ligands that are known for NR4A2 are quite many, almost like 13 of them. Now, of these 13, 5 happens to be somewhat polypharmacological and crossover ligand, where they can engage other family members also of NR4A, or they can also crosstalk with other receptors. But 8 of them happens to be quite specific. And when one actually exercises the Lipinski rule of 5 onto these 8 ligands, one can clearly see few potential drugs of future. Some functional facts about NR4A2. NR4A2 actually upregulates FOXP3 to the extent that FOXP3 is a direct target gene of NR4A2 in Tregs. So apart from the cellular cues wherein NR4A2 can modulate tolerogenic disease, and by virtue of that, more of T regulatory population, at molecular level also, NR4A2 and its cognate ligand can actually induce stability in the T regulatory population by...
Dr. Pawan Gupta, you're left with 5 minutes.
Sure, I'll be in time. I'll keep it short. And then, as I was telling, that arginine is a direct target gene of NR4A2 in macrophages. NR4A2 has been reported for both its anti-inflammatory role in macroglia and astrocytes and the macrophages. NR4A2 has been reported to be protective in Parkinson's disease and in sepsis. So one can safely say from this presentation that NR4A2 is an inflammation dampener and an immune regulator. It is safe to conclude from this presentation that nuclear receptors, they are poised to become next generation drug target. 16% FDA approved drugs acting through nuclear receptor. That is just the start, and it's going to increase further. Secondly, NR4A2 seems to be a very promising anti-inflammatory drug target. Now here I would very quickly show one of our unpublished data also. So we have been working on nuclear receptor expression in different T helper cell subsets, and in that arrangement, we came across a nuclear receptor by name NR1D1 or reverb alpha, which is a very important circadian regulator, and we realized that this nuclear receptor is actually able to directly gene repress GATA3, and it had ramifications in asthma disease progression. So we employed the knockout mice and compared it to the wild type mice, and we realized that yes, NR1D1 alleviates asthma by abating the master transcription factor GATA3 and actually inhibits Th2 cell development. This is some data regarding the gene regulation, where NR1D1 together with ANCOR, a co-repressor, and HTAC3 is able to bring about direct gene repression of GATA3. So with that, I would convey my sincere thanks to Dr. Javed for giving this opportunity for making this presentation. Thank you so very much. Shall there be any queries? I'll be happy to answer.
Sir, I have one question, please. Can I ask now?
Can you be a little louder?
I have one question. Can I ask now?
Please.
Yeah, I'm Hilal Bashir from CSR MTech. I have a question that these diseases which have been generated from the bone marrow, have you tried to look for this NR4A2 in case of tissue-resident dendritic cells or any kind of mucosal tissue?
That would have been great if we were able to lay hands on the ex vivo disease.
Yeah.
But because the volume of tool that we required for the kind of assays that we were performing, the real-time array, it would not have been possible.
Yeah,
so it would also have been possible by utilizing the bone marrow precursors and BMDCs is derived therefrom.
Because it would be quite interesting. Okay, sorry.
If you look into literature, there is a lot of equivalence that has been drawn for BMDCs with that of real in vivo situation. So there are a good amount of literature in that regard. But yes, I agree that extrapolations could also have been done with ex vivo DC.
Because it will be quite interesting if we go from the bone marrow derived dendrite cells to the tissues. The story differently changes in that, because in case of tissues, you will see that CD103 positive and CD11B negative DCs are T17 inducing, and CD103 positive and CD11B positive dendritic cells are actually Treg inducing. So it will be quite interesting if we correlate the expression of this nuclear receptor with the phenotype of dendritic cells actually. So
I totally agree. Ex vivo extrapolation of BMDCs and expression atlas obtained therefrom could have been more convincing. But nevertheless, if you see literature, the equivalence is very much there as far as the expression profiles are concerned. If there is a cellular experiment involved, or if you are looking for cellular immunological questions, then in that case, ex vivo DC population is quite essential. But if you are looking for molecular switches, so BMDCs in that regard has been reported to be quite an efficient and sufficient tool to address it.
Okay. Thank you.
Okay, so any other question? Okay, so
hello.
There aren't any question for Dr. Pawan, except people they have text that they highly appreciate the work and their the presentation is very good.
I would appreciate some questions, please.
Yeah, any questions from this in Google Meet participants?
Javed, this is Balaji. Can I ask a question?
Okay.
Sure.
Dr. Pawan, just something I missed. Does NR4A2 play any role in case of TB infection?
I think you asked the right question. Actually, we came up with expression atlas and we realized that NR4A family members, they undergo significant change in expression in MTB infected DCs and macrophages. And we were also able to extrapolate and corroborate that in human MDMs and in PBMCs derived from TB patients, close contacts, and healthy individuals. So NR4A seems to have a role to play as far as expression is concerned, but we have not been able to make a functional connotation of it. So the functional studies have not been done so far, and it is just not NR4A2. That data also revealed another nucleoreceptor, ROR alpha, which is also undergoing a significant change in expression. Now, whether that expression can relate to function or not, that remains to be seen. So that actually happens to be our present effort, and we are working on that. So maybe a year, 2 year time period, that thing also may get revealed.
Is there any null mice available?
Come again?
Is there any knockout mice available?
For NR4A2, we have knockout mice available. In fact, we have been studying nucleoreceptor in TB from the host pathogen interaction point of view. We have been able to come up with several receptors which are acting as pro-TB and several which are acting as anti-TB. We have been able to come up with PPAR gamma, PXR and TR4 which are acting as pro-TB, and we have been able to come up with LXR, VDR and reverb alpha which are acting as anti-TB. Now for them we have the knockout animal with us, but for NR4A2 we have to confirm the studies, we have to get the leads, proof of concept has to be established as far as the functional role is concerned, and then definitely we would go about ordering the NR4A2 knockout. For now, we are going ahead with the adenovirus assisted systems for the gain of function or for the loss of function. So for the loss of function, we do adenovirus shRNA, and for the gain of function, it is again adenovirus overexpressing NR4A2. But yes, getting a knockout would be the big deal, and we have plans for that. But we are actually waiting for some kind of functional role, which we can ascertain before we order them up in TB.
So, Dr.
Thank you
Pawan,
so
there's a
much.
question from Sabita Singh, and the question is that she wanted to know if nuclear receptor play a role in contributing to steroid resistance conditions.
I think there is some genetic polymorphism known for some of the steroid hormone receptors, which contributes to differential sensitivity to glucocorticoids. And these polymorphism and SNPs are not just known for the coding region, but they have also been established for the non-coding regions, the gene promoter. And yes, there are reports of that kind. It can contribute, but I would say that there are not many reports.
Dr. Pawan, there's a question from me regarding nuclear receptors. So, have you checked in case of human beings, especially in patient, that does they contribute towards MTB pathogenesis or clearance?
Yes. So these findings that we obtained for pro-TB NRs and anti-TB NRs, so the extrapolation of the expression was done in PBMCs that we obtained from the TB patients, the close contacts, and also the handy individuals. And the findings that we had in vitro or in the mice model, we were successful in extrapolating that into clinical samples also. In fact, we also collected samples from drug non-responders. Drug non-responders, which include both the ones which are harboring the drug-resistant strain and which are harboring the sensitive strain, but somehow not responding to the drug well. So from both the patients, we had the samples and we looked for the expression. And yes, there is a real-time evidence which suggests that. The question.
Hello.
I hope I have answered the question.
Right. So, are there any questions?
Can I ask one question?
Yeah, sure.
So, what are the natural ligands for this receptor?
Most of the ligands are endogenous in nature. These could be dietary or nutritional lipids. These could be mineralocorticoids, glucocorticoids, the endocrine hormones that we know of, the vitamins, both the essential and the non-essential ones.
Okay, so if we don't have any more questions, we would like to thank Dr. Pawan for a very nice talk, a very naive area which he's exploring. So let us give him a big applause.
Excuse me, I had one question. Hello?
Please go ahead.
Dr. Rashmi, you can ask him at Integ now.
It was written there, but you didn't realize it. Okay, anyway, I can ask afterwards. Thank you.
I think we have time. It's only lunch time. So please feel free to ask.
Okay. Okay, so I wanted to ask, what is the role of these nuclear receptors in shaping the gut microbiota in case of like inflammatory bowel disease? And if we will target these receptors, can we restore these?
Excellent question. A very bright question. In fact, just like that we reported for a crosstalk between MTB lipids and host lipid sensing nuclear receptors, right? A heterologous ligand receptor pairing, there is also a probability and possibility wherein the metabiotics and the nutribiotics of the gut microbiome can actually crosstalk with these nucleoreceptors, engage these nucleoreceptors, and can help either subvert the system for their survival or the other way around. And that is another line of work that we are planning to expand into.
Okay.
Okay, Dr. Pawan, thank you very much for a nice talk. Let us give him a big applause. It was a really very good talk. So now we break for lunch, and we'll be back again with a very interesting talk by Dr. Pradeep Sen. Right? So we meet you at 2 o'clock. Thank you very much for joining us. We'll be back after half an hour.
groups have shown that Leishmania donovani upregulates CD40 expression on dendritic cell. Other groups showed that Leishmania donovani downregulates CD40 expression on DC. And accordingly, we first wanted to confirm whether Leishmania donovani upregulates or downregulates CD40 expression on dendritic cell. To confirm this, we infected bone marrow derived disease with Leishmania domesticode for varying time points, 6 hour, 12 hour, or 24 hour, followed by LPS stimulation. And we measured the CD40 expression via flow cytometry. We found that when LPS stimulated CD40 expression on dendritic cell, the BMDC infection with LD domesticode progressively reduced LPS stimulated CD40 expression on dendritic cell. And that is not only we observed for LPS stimulation. With respect to TNF-alpha stimulated CD40 expression on DC, LD domesticode also exhibited similar inhibitory effect on CD40 expression. Next, we wanted to confirm whether LD really downregulates CD40 expression in vivo on dendritic cell. For that, we derived the disease splenocytes from uninfected versus infected mice and stimulated with LPS. And our flow cytometric analysis on the CD11C gated population, which is designated for dendritic cells, showed that LPS stimulation considerably upregulated CD40 expression on dendritic cell in case of uninfected uninfected macrophages, whereas the DC derived from infected macrophages, infected mice, showed considerable reduction on the CD40 expression on dendritic cell. And this figure suggests that LD downregulates CD40 expression on DC. Now, from next couple of slides, I'll be showing the mechanism by which CD40 expression is regulated on dendritic cell, just in case of the uninfected DC. It is not having any relevance with the LD infection. It has been shown by the several groups that a couple of the transcription factors like STAT1 alpha, NF-kappa B, SP1, they are generally regulating CD40 expression. However, we wanted to explore whether any other transcription factor can regulate CD40 expression in dendritic cell. When we analyze the CD40 promoter sequence by computational analysis, we observed that there are two potential RUNX binding site at the CD40 promoter because it matches with the conserved RUNX binding sequence pyrimidine G, pyrimidine GGT. Run-domain transcription factor that is commonly known as RUNX transcription factor family is actually a heterodimeric complex consisting of core binding factor beta and core binding factor alpha. Core binding factor beta per se do not interact with the any promoter region of the target gene. It is responsible for heterodimerization of the complex. It enhances the DNA binding affinity of the core binding factor alpha, and also it protects the complex from ubiquitin mediated degradation. In contrast, the alpha subunit, which is also known as RUNX subunit, it is meant for binding towards the DNA. And this RUNX actually is encoded by three different isoforms, RUNX1, RUNX2, and RUNX3, which are having considerable homology among themselves, and they have the conserved RUNT homology domain, which is meant for binding to this particular sequence. The role of RUNX transcription factors in regulating dendritic cell activation or maturation has been done very limited number of the studies. For example, the Bronner group has shown that RUNX regulates DC maturation, and also they have shown that RUNX impedes the CCR7 mediated DC migration. However, role of RUNX in CD40 expression is not yet known. As I already mentioned that when we analyze the promoter sequence of the CD40 promoter, we identified two putative RUNX binding site, and we first initially confirmed whether really these two binding sites engage RUNX protein for regulation of CD40 expression in DC. Accordingly, we performed electrophoretic mobility shift assay using nuclear extracts derived from LPS and TNF-alpha stimulated DC, and we observed that LPS and TNF-alpha considerably induced binding of nuclear protein to either of these probes, which individually encompass the RUNX binding site. And when we mutated these RUNX binding sequences, this LPS and TNF alpha induced binding of the nuclear protein was completely abrogated. Supershift analysis further confirmed that RUNX1 and RUNX3, but not RUNX2, bind upon LPS and TNF alpha stimulation. And chip analysis also showed the recruitment of RUNX1 and RUNX3 to the CD40 promoter in dendritic cell following LPS and TNF alpha stimulation. Next, we wanted to confirm whether both sides really bind RUNX transcription factor upon LPS stimulation in DC within the cell. And for that, we performed the in vivo footprint analysis, where we found that in contrast to untreated DC, the LPS and TNF alpha treated DC displayed the protection at both R1 site and R2 site due to the binding of some nuclear protein. And that indicated that indeed both sides are getting engaged by the RUNX transcription factor upon LPS and TNF alpha stimulation. Next, we wanted to check whether RUNX really play any role in regulation of CD40 expression in DCL. For that, we silenced RUNX1 and RUNX3 expression either individually or in combined fashion. And we found that whereas LPS and TNF alpha stimulation upregulated the CD40 expression in controlled silence DC, silencing of RUNX1 or RUNX3 generally considerably inhibited LPS and TNF alpha stimulated CD40 upregulation. And that these results collectively show that RUNX1 and RUNX3 promote CD40 expression in dendritic cell. Now, what could be the upstream signaling events that is responsible for RUNX-mediated CD40 upregulation in dendritic cell? The previous studies have shown that PI3K regulates LPS and TNF alpha induced CD40 expression in dendritic cell. And that gave us the clue that probably PI3K might be the upstream signaling mediator for RUNX-mediated CD40 upregulation in DC. Indeed, when we pretreated the DC with warfarin and lye, LPS and TNF alpha induced CD40 upregulation was considerably inhibited, and also the RUNX binding towards the CD40 promoter was also inhibited, although RUNX1 and RUNX3 expression remained unaltered. Next, we wanted to identify what is the PI3K isoform that is, or the PI3K complex is responsible for this RUNX-mediated CD40 upregulation. For that, we performed immunoprecipitation assay, and we found that P85 alpha P110 beta complex interacted with the TLR4 and TNFR1 receptor following LPS and TNF alpha stimulation. To identify the role of this PI3K complex, we silenced P110 beta expression via siRNA, and we found that silencing of P110 beta inhibited LPS and TNF alpha induced RUNX protein binding to the CD40 promoter, and also it inhibited LPS and TNF alpha induced CD40 upregulation in dendritic cell. Indeed, PI3K P110 beta expression, when it was silenced, the phosphorylation of the downstream effector AKT was also inhibited. And when we pretreated the DC with the AKT inhibitor, we found that LPS and TNF alpha induced RUNX binding to the CD40 promoter, and LPS and TNF alpha induced CD40 upregulation on DC is also inhibited. This suggests that PI3K AKT signaling drives RUNX-mediated CD40 upregulation on dendritic cell. Now, next we wanted to check what is the relevance of this PI3K AKT RUNX signaling in LD-mediated suppression of CD40 expression in DC. We've observed that when we infected the disease with LD promesticode for varying time point, that is 12-hour or 24-hour, LPS-induced RUNX binding to the CD40 promoter or TNF alpha-induced RUNX binding to the CD40 promoter was greatly inhibited. And indeed, when we isolated the DC from infected mice and stimulated with LPS as compared to the DC derived from uninfected mice, RUNX binding to the CD40 promoter was largely inhibited. This is due to the fact that LD promestigotes inhibits the phosphorylation of AKT, that is the upstream mediator of RANX, that induced by LPS and TNF alpha. Now, the question is how LD inhibits this PI3K-AKT RANX signaling in dendritic cell. It is already known that LD induces the phosphorylation of the phosphoprotein tyrosine phosphatase SHP1 in macrophages, but it is not yet known whether it can induce SHP1 activation in DC. In addition, another study has shown that SHP1 negatively regulates PI3K-AKT signaling. So we first wanted to check whether Dyschnia promestigotes can at all induce SHP1 activation in DC. We found that when diseases were infected with Dyschnia promestigote, there is a continuous activation of SHP1, and this activation of SHP1 was continued despite LPS and TNF alpha stimulation. And when we silenced SHP1 expression in dendritic cell, we found that LPS induced and TNF alpha induced AKT phosphorylation was restored in LD infected DC, and also the RANX binding to the CD40 promoter and CD40 upregulation was again restored in LD infected DC. That suggests that LD generally inhibits PI3K-AKT RANX signaling via SHP1, and that is why we should expect to see the CD40 downregulation in dendritic cell. So far, I have shown all this data with respect to LPS and TNF alpha to elucidate the mechanism of how LD can regulate CD40 expression in dendritic cell. Now onward, my subsequent slides will be depicting the role of RANX in the context of anti-leishmanial immunity, and to explore that, we focused our research on sodium antimony gluconate, because sodium antimony gluconate is regarded as the first-line drug against visceral leishmaniasis. And we have previously shown that sodium antimony gluconate upregulates CD40 expression on dendritic cell. However, whether sodium antimony gluconate really engages RANX transcription factor to upregulate CD40 expression in DC is not yet known. To confirm this, we have performed the electrophoretic mobility shift assay followed by supershift analysis, which depicted that RANX1 and RANX3, but not RANX2, binds to the CD40 promoter upon sodium antimony gluconate stimulation. And this is due to the fact that sodium antimony gluconate induces AKT phosphorylation in DC. And the same PI3K complex that I've already mentioned with respect to LPS and TNF alpha stimulation, that P85 alpha P110 beta is probably playing a role in this AKT mediated RANX binding to the CD40 promoter. This is because of the fact that when we silence P110 beta expression via siRNA, sodium antimony gluconate induced RANX binding to the CD40 promoter was largely inhibited. And this is because of the fact that it inhibited the AKT phosphorylation in dendritic cell. And the downstream effector of the PI3K, that is AKT activity, when it was blocked by the AKT inhibitor, sodium antimony gluconate induced RANX binding to the CD40 promoter is also abrogated largely. Next, we wanted to check whether RANX transcription factor plays any role in sodium antimony gluconate induced CD40 upregulation. And this, we silenced RANX1 and RANX3 expression via siRNA, and we found that sodium antimony gluconate induced CD40 upregulation was largely inhibited. Interestingly, sodium antimony gluconate continued to upregulate the Leishmania donovani infected dendritic cell, unlike the LPS and TNF alpha stimulation. This is because of the fact the sodium antimony gluconate, being a potent inhibitor of SHP1, it blocked LD induced SHP1 phosphorylation. As a result, the PI3K pathway is now active, and that will facilitate the binding of the RANX protein to the CD40 promoter, and consequently it upregulated CD40 expression in LD infected disease. Next, we wanted to check whether RANX is having any role in sodium antimony gluconate induced DC anti-leishmanial response. I apologize for this overcrowded slide, but I'm trying to explain. Here we have isolated the bone marrow derived DC from biopsy mice and treated with either PBS or sodium antimony gluconate. And in some experimental set, we silenced RUNX1 and RUNX3 expression via siRNA. And in other set, we overexpressed the CD40 expression in this RUNX1 and RUNX3 silenced disease, and then treated all these diseases with the sodium antimony gluconate. Subsequently, all these disease sets were adaptively transferred in LD infected mice, and after 48 days post-infection, we analyzed the two parameters. One is parasite burden in the liver and the spleen, and also the Th1 and Th2 profile in these infected mice. We observed that unlike the PBS treated DC, sodium antimony gluconate treated DC considerably reduced the Leishmania parasite burden in the spleen and the liver, whereas RUNX1 and RUNX3 silencing in this DC largely inhibited the anti-Leishmanial effect of sodium antimony gluconate, whereas overexpression of the CD40 in these RUNX silenced DCs restored the ability of sodium antimony gluconate induced anti-Leishmanial effect.
5 minutes are left.
Yes, sir, I'm almost done. To analyze the cellular basis for this particular immune protection, we measured the Th1 and Th2 profile. We found that when this particular disease is treated with the SAC, this particular set was adaptively transferred in the LD infected mice, it considerably increased the frequency of IFN gamma producing T cell, but reduced the IL-10 producing T cell. Whereas when we silenced the RUNX1 and RUNX3 expression in this disease by siRNA, the IFN gamma producing T cell frequency was going low, and IL-10 producing T cell frequency was going high. But when we overexpressed the CD40 expression in this RUNX silence DC, the frequency of IFN gamma producing T cell and IL-10 producing T cell was comparable to that of the sodium antimony gluconate treated DC. This particular data suggests that RUNX proteins play a critical role in conferring the DC-mediated anti-Leishmanial response, and that is true by CD40 regulation of the CD40 expression. In summary, we can say that LPS and TNF-alpha stimulation of TLR4 or TNF receptor induces the PI3K-AKT signaling pathway that drives RUNX transcription factor binding to the CD40 promoter, and consequently it upregulates CD40 expression on DC. When LD promastigote infect the dendritic cell, it induces SHP1 phosphorylation, and as a result, will be this particular pathway, and the CD40 expression is getting reduced. In contrast, sodium antimony gluconate treatment of DC blocks the LD-induced SH1 phosphorylation. As a result, this PI3K-AKT mediated RUNX binding to the CD40 promoter is restored, that is upregulating CD40 expression on LD-infected DC, leading to the type 1 T cell response, and that leads to the anti-Leishmanial immunity. And these are my group, Mr. Nausad Akhtar, Manish Mishra, Vinod Jadhav, Manisha Jadhav, Sunaina Lal, Ravinder Guzo, all of them contributed largely to this project, and Dr. Rajkumar contributed to the molecular biology part of this related to these findings. And this project is funded by DBT, CSR, and GCRF UK. Thank you so much.
So thank you, Dr. Sen, for very interesting talk. It was really very exciting that how Leishmania donovani reduces CD40 by blocking PI3K-AKT pathway, RUNX, of course, and how through synthesis via SHP1. So it was really very good. So you can see, yes, especially for youngsters, you can do good science in India as well. So you should be motivated with this young scientist, Dr. Pradeep Sen. So it's open for question answer session.
Yeah, I'm ready to answer all the questions.
Okay, so there is a question for you.
Yes, sir.
Can I have
That,
one question?
this is Sushma. She asked, can any other compound be used as inhibitor of SHP1?
Yeah. Uh, so far, SHP1, we used sodium antimony gluconate that blocks the SHP1 activity. I'm not sure about any other compound that can be used, but even the primary goal is that if you block the SHP1 activity by any drug or like that, it should prevent the inhibitory effect of Yersinia donovani on the CD40 expression, and that is why you should expect to see the anti-Leishmania immune response.
Okay. Anybody else?
Yeah, I have one question, sir. Can I ask you?
Yeah, sure.
Yeah, I'm Hilal Bashir from the same institute actually. I have a question regarding the induced expression of CD40 in whether it's in macrophages or dendritic cells, because it has been shown by many studies. You take the Christian Barthel group, and various other groups have shown that the induced expression of CD40 signaling or the constitutive expression of CD has led to the dysbiosis in mice. It has been shown, and also the fetal colitis. It has been shown that it leads to the colitis, because it has been shown that whenever there is a CD40 high expression, there is the elevation in the CD40 signaling actually, and that leads to the abolishment of the ROR gamma positive Tregs, and conclusively that leads to the colitis in the host. So is it physiologically relevant to induce the overexpression of CD40?
Here, Hilal, one thing is that generally you will have to see in what context you are going to overexpress CD40 or not. In certain disease, when you are overexpressing CD40, as you mentioned, it is detrimental because already it will lead to the pro-inflammatory response tremendously. But in case of the Leishmania infection, the primary consequence is the immune suppression. So here, I'm not talking about that you should enhance tremendously, but it should have at least some level which is close to the normal, should confer the protection.
Okay, thank you.
Could I give you the answer?
No, it's okay, sir.
Okay. Any more question? Hello? Am I audible?
Can you hear me, Dr. Sen?
Yes, speaking.
So there is a question from Anik Mitra. LPS and TNF signaling affects runs binding or runs expression as well. So if so, did you check the mechanism?
The RUNX expression is not at all altered, as I already showed. Because, if you see that what is the mechanism for the RUNX mediated CD40 regulation, it is primarily because LPS and TNF alpha, by regulating PI3K-AKT signaling, it is we have started getting some preliminary data. It is controlling the nuclear localization of RUNX transcription factor. It is facilitating RUNX transcription factor nuclear localization, and that is why it is promoting the RUNX binding to the CD40 promoter. It is not, in fact, regulating the RUNX expression. As I already showed the Western blot data, that there is no change in the RUNX transcription factor expression.
Okay. Any other question? So, Dr. Sen.
Yes, please.
Do you think modulating CD40 can affect the survival of Leishmania donovana inside the macrophages?
Yeah, actually, previous studies have shown that CD40 generally interacts with the CD40 ligand. So you will be expecting more IL-12 mediated IFN gamma production. And this IFN gamma production induces expression of ionos, and that is why you should expect to see more nitric oxide, elevated nitric oxide production within the infected macrophages, and that will lead to the killing of the parasite. And you can see that if the CD40 expression can be restored there, then it should promote the anti-lesional immunity.
So, what is your opinion about immunotherapy using CD40?
The preliminary indication is showing that CD40 should play a critical role. But currently, I cannot say at this moment in response to the therapeutic part, but it should have the potential. And it is not only restricted to the visceral leishmaniasis, any pathogenic infection where that is governed by Th1, Th2 balance, you can see that it might have the promise as a therapeutic agent.
Okay. So, any more questions? Any question from the students? Okay, so if there aren't any questions, we would like to thank Dr. Sen for very exciting talk, very interesting talk, and see the amount of work he has done. And so I would like to congratulate his Dr. Sen and his group for doing wonderful work.
Thank you, sir.
Yes, it was really wonderful work. And you see the amount of data his lab generates. And it's not easy to publish paper in Journal of Immunology from India. So he regularly publishes paper from his lab in India in Journal of Infectious Disease. And he has published in PLOS Pathogen as well. So let us give us big applause to Dr. Pradeep Sen. Okay, Dr. Pradeep Sen, thank you very much for your lecture.
Yeah.
And okay, so Professor Wilkinson, we welcome you. Professor Wilkinson, he has joined us. Hi, Professor Wilkinson. Hi, Robert, how are you? So, if you're ready, we can start with your presentation. Hi, Robert, we can't hear you. Probably your mic is muted. We can't hear you. Please check. We can't hear you. Robert, probably your mic is not working.
Javed, can you hear now?
Your mic is probably not working.
No, it's a new. Where's the other computer?
So meanwhile, let me introduce you to Professor Robert Wilkinson. So Professor Robert Wilkinson, he did his MA from Cambridge, then he did his BMCPCH, the medical degree from Oxford. He did his DTMH, then PhD, FRCP, and FM Med Science. So currently he's a senior group leader at the Francis Crick Institute, London, and welcome senior fellow in clinical science at Imperial College of London. Both posts seconded to the University of Cape Town. So he's a director at the Center for Infectious Diseases Research in Africa, and the group leader and director at Crick African Network and member Institute of Infectious Diseases and Molecular Medicine, Department of Medicine, Faculty of Health Sciences, University of Cape Town. So Professor Robert Wilkinson's interests are focused on clinical and immunological aspects of tuberculosis, particularly in the context of HIV infection. So HIV, as we know, HIV and TB are the most pressing public health problems in many countries, including Africa, and many hundreds of people still die prematurely per day because of HIV, and many due to coexisting TB. So Dr. Robert Wilkinson, he focuses on how can tuberculosis infection in HIV-infected people be more effectively prevented, and by what immune mechanism do preventive therapies decrease susceptibility to TB, and what is the cause and best management of HIV, TB, tuberculosis-associated immune reconstitution, inflammatory syndrome. So Professor Robert Wilkinson, he has published hundreds of papers, probably 300 plus, Robert. And
yeah, can you hear now? Can you hear now?
Yeah. I can hear you now.
Oh, good. Okay,
good,
so
good.
Sorry about that. I'm not
and
sure
many of these papers he has published in very high impact journal. He has published many papers in Lancet, Proceeding National Academy Sciences, US. And with this introduction, I would like to welcome Professor Wilkinson, and today he'll be talking about the immune response in tuberculosis and meningitis. So we welcome you.
Thank you, Jared. Do I press the present now button? Javed?
Yeah, you can start your presentation.
I press present now. Okay.
Yeah, right. You present now.
Oh, but I haven't got the talk, not on this computer. Oh, God. Sorry, I switched computers, Javed, and I'm just going to... You don't have the talk, do you?
No, we don't have your talk. So you switch on your slides, and then you have to just press present now, and you'll be in.
Yes, but in order to get sound, Javed, I had to change to computers. So I'm just going to change get this talk back onto the new one. We're still going to start on time. Miracles never cease to amaze, do they? Control. Sorry, I'm not familiar
tell you my association with Professor Robert Wilkinson. We know each other about 30 years back. We met when Professor Wilkinson was very young, very charming personality, very handsome person. So this is now quite long. And We worked together, and I think we published about 3-4 papers on tuberculosis. So we were working mainly on some T-cell epitopes. So during that time, we were in Hammersmith Hospital and working with Professor Uri Ivani. So Professor Uri.
Are you seeing?
Oh, great! Yeah.
Okay.
Now you break into presentation mode, and there you are. Yeah, good.
You're happy.
I'm happy.
Good. Then we're all happy. So exactly on time, despite some manic shuffling around. So Javed's given me an overlong introduction. I'm a physician scientist who I'm employed by Imperial College, but I have a laboratory at the Francis Crick Institute, and I spend most of my time in Cape Town. So that reminds me of Groucho Marx, who didn't want to be a member of a club, who wanted him as a member. I always begin my talks by acknowledging that I'm the Paymaster General and Travelling Salesman. In this year, they're somewhat static and virtual travelling salesman for a larger group of more talented people who are listed here. I might say as well that we struggled over the years to get money to research TB meningitis. It seems to be regarded as a recherche part of the tuberculosis spectrum, but in the last year or so that trend has reversed, and I'm grateful to acknowledge several funders have come to the fold. I need to give particular acknowledgements in this talk to Graham Mankeys, who is a physician scientist who has worked with me over many years in Cape Town, for the early part of the talk, for the work of Suzanne Marais, who is now a neurologist at Groote Schuur Hospital in Cape Town, and Rachel Lai, who is an MRC Career Development Fellow at Imperial College London. So the classic view of the pathogenesis of TB meningitis is that TB somehow gets into the brain, and it's conceived that that is bloodborne because the blood-brain barrier is intended to prevent infections getting into the brain, but obviously that is circumvented in some way in TB meningitis. And two mechanisms are postulated. One is that bacteria translocate the basement membrane vascular endothelial cell via a Trojan horse mechanism. In other words, there is intracellular infection, and through biopedesis against an inflamed membrane, the infection gets in that way. Or it's possible, and we've demonstrated this in other work, that the bacteria themselves could actually get into endothelium and thereby translocate the basement membrane. But whichever mechanism that might be, and however frequently that may occur, which is also an area of some debate and discussion in TB meningitis, a small focus is believed to be established. And should that focus not be eliminated by the immune response, it gives rise to inflammation.
Robert, your slides have disappeared.
Oh, I don't know why.
You are visible, your slides have disappeared.
Okay, let's just see why that might be.
I think it happened.
Still hear me though. Your screen is still visible. Resume your presentation. Resume presenting. Okay, let's go back up to there. So are you seeing them again now, Javed?
No. You're seeing probably your desktop. So you have to share your presentation. Yeah, good. You can see now.
You can see it now?
Yes. Do it full screen.
And now?
Presentation
and now?
mode. Yeah, do it. Yeah, good. Good.
It's okay now, is it? Yeah,
it's
okay.
Okay now.
So where was I up to? So a focus of infection is established, and that leads to an inflammatory influx. And if that isn't controlled, and in the vast majority of cases it's presumed not to, eventually that focus ruptures and gives rise to intracerebral spread of infection via the CSF, and can result in the clinical picture that you see on the right and that's a very destructive brain disease with obstruction to the cerebrospinal fluid flow, giving rise to hydrocephalus, tuberculomas, which are these ring-enhancing lesions. And in actual fact, in this particularly severe CT scan, it's very difficult to discern any normal brain whatsoever. So what does that look like in reality? Well, what I'm about to show you is the macroscopic pathology of TB meningitis. And this is visualized by transiting the third ventricle into the lateral ventricles into the fourth ventricle here. And that's done in a child to relieve the pressure either due to a tumor or indeed due to TB meningitis. And the left-hand panel represents the normal appearances. So what's happened here is that the endoscope has been passed into the lateral ventricles and a fenestration is being made in the floor of the third ventricle, passing into the fourth ventricle. And the endoscope goes down through that hole. And what we're seeing here in this journey to the back of the brain is the midbrain. These arachnoid viscera, the normal appearance, that spider's web appearance. And what you can see down at the bottom as well there is a normal pulsation of the basilar artery. So let's just run through that again because we've got quite nice slack timing in this meeting. So here we are. This is the normal appearances of the base of the brain. And you can see down at the bottom there, I don't know whether you can see my little finger pointer wiggling. There's the basilar artery beating normally. This is all clean and beautiful. So this is a case of TB meningitis who presented with obstructive hydrocephalus. And you can see immediately that the endoscopist is having some difficulty fenestrating the floor of the lateral ventricle. When he eventually does, you can see that that pulsation is not restored and there's a little bit of bleeding as well, and that's due to a thickening. And then we go down into the base of the brain, which is caused coalescence of the administration. Now we go with those arachnoid villi. You see a tubercle down there as well, and the normal pulsation of the basilar artery is absent. So this is the macroscopic pathology of meningeal tuberculosis, and it gives you some insight into one of the most characteristic syndromes, which is of a basal meningitis. So I first really got interested in this about 16, 17 years ago, and that was coincident with my move to Cape Town. Now, Cape Town has a very high tuberculosis incidence. About 1% of the people get TB every year. It had a very high HIV prevalence in the order of about 17% amongst adults aged 15 to 44. And from about 2004 onwards, there was highly efficient access to antiretroviral therapy. And that triple coincidence led us to observe an increased incidence of a disease, of a iatrogenic complication referred to as paradoxical TB iris. And these are the clinical circumstances when a patient is diagnosed with TB and started on TB treatment and then begins to improve, but is predicated by international guidelines then to start antiretroviral therapy. Otherwise, the risk of dying of other HIV-related conditions remains high. And in those circumstances, a variable number of patients will manifest a transient deterioration of their tuberculosis, either of existing lesions or new lesions. And that usually occurs about 1 to 4 weeks after starting antiretroviral therapy. And the critical risk factors to that are a low CD4 count and a disseminated TB and a short interval between starting TB treatment and antiretroviral therapy. So these are some clinical instances of this syndrome. And I'm particularly, for the purpose of this talk, draw your attention to the 3 panels towards the right of the slide, which show suppurative inflammation. So the pus that you see in the left middle arises from this psoas abscess and is essentially non-viable. I took this to the laboratory and said, look at the lymphocyte repertoire in there, and the laboratory staff were unable to, in fact, isolate anything viable at all. The other 2 pictures, again, this shows the most unusual manifestation of TB. This is referred to as a compartment syndrome. It's a pyogenic swelling of a forearm. So this is not the classic presentation of TB. And again, from that was expressed a very large amount of necrotic material. And I must say, when these pictures were taken in the late part of the noughties, we didn't click what the significance, what the macroscopic pathological significance of this might be. We were still thinking that this in some way must be lymphocyte mediated, because the characteristic of antiretroviral therapy is to increase the CD4 lymphocyte count, and that's what we thought must be causing it. So we carried out a number of observational and case control studies on the syndrome. And what became apparent over a number of years and through those studies, in a different number of ways of examining the condition, was that the suggestion was that the signal coming from TB iris patients was actually of the innate immune response. And I think our most important paper was the 2015 paper in Nature Comms, and as a consequence of that, we built up a model that suggested that it is actually the innate recognition of TB antigens that give rise, both through interferon signaling and the induction of the inflammasome and pyroptotic and probably now necroptotic and apoptotic cell death, but also via the classical mediated pathway of NF-kappa B activation. It gives rise to the secretion of a large number of both pro and anti-inflammatory cytokines. Now, in 2020, so-called cytokine storms have become front-page headlines, and the general public understands this. But in 2008, it was a difficult concept to get over. And I wouldn't really describe what occurs in TB iris as a cytokine storm, more a sort of occluded cytokine front, because it's not as acute as the syndrome that's seen in SARS-CoV-2 infection. But nevertheless, that there is good evidence that this is occurring and that the underlying cause is inflammasome activation. So around the same time, we began to observe that the most severe form of this syndrome was that which occurred in the central nervous system. So TB is the cause of a very common cause of meningitis in sub-Saharan Africa. And we began to observe cases of immune reconstitution inflammatory syndrome in the central nervous system and indeed in the spine. And those cases were developing despite corticosteroid therapy, and indeed 13 to 25% of patients die of it. So our first study was essentially observational. What we did was sample at the point of TBM diagnosis. We sampled again at the day that patients started antiretroviral therapy. And we sampled again at day 28, irrespective of whether they did or did not develop the syndrome. And the first striking observation, and that ties in with our prior observations that we were seeing what appeared to be a suppurative inflammation, was that looking at the left-hand panel, you see that in cases that did not develop TBM iris, there was neutrophilia at baseline that resolved on anti-TB therapy, and when antiretroviral therapy was started, it made no difference, and the patients continued with an uncomplicated course. By contrast, in cases when there was the development of TBM iris, there was again evidence of neutrophilia at baseline, which resolved during TB treatment. But upon starting antiretroviral therapy, there was a marked increase in cerebrospinal fluid polymorph count, which gradually resolved over the course of the next 3 weeks. So we wanted to bring an unbiased technique to the analysis of this condition, and the technique that we've used, and which a lot of the rest of the presentation depends, is the technique of transcriptomic profiling in human tuberculosis. And I, together with a colleague, Anna Gara, from what was then the National Institute of Medical Research, now the Francis Crick Institute.
Let me just hide lower panel on the screen.
बोथ तो उनके साइड से हमारे साइड से भी ये अराविड है ये
Sorry, there's quite a lot of background noise, but I'll try to carry on through it. what was shown was that active tuberculosis has a peripheral blood transcriptomic signature dominated by neutrophils and type 1 and type 2 interferons. That transcriptomic signature tends to relate to disease extent and resolves during successful treatment. The differentiation of active from latent TB and other conditions might be aided by transcriptomic profiling, and that was particularly present in children, which is otherwise a difficult diagnostic domain in tuberculosis. And more recently, we've applied this to the ancient problem of latent tuberculosis and found evidence that transcriptomic signatures may predict progression of or detect subclinical tuberculosis. So we applied this to the peripheral blood of these patients who were undergoing immune restorative antiretroviral therapy during the course of treatment for TB meningitis. So the first panel that you'll see here is actually the transcriptome before that antiretroviral therapy had been established. And to our surprise, there was a clustering that indicated a distinct pattern, even ab initio, in patients who went on to develop TB and iris. The p-values are not very strong because it's a small number of patients, but what you see on the right is a predominance of innate immune signaling and not the acquired immune response. So inflammasome activation, IL-8 signaling liable to lead to recruitment of neutrophils, and a strong signal that activation of innate immunity was important in this. At the time of TB and iris onset, that pattern became far more prominent, and so we see inflammasome activation, phagosome formation, and the role of pattern recognition coming up as very strongly overrepresented pathways. The lower panel shows technical validation by RT-PCR of some of the genes that we'd seen in the microarray analysis. And the right two panels show activation in TB and iris within the CSF of the inflammasome-related caspase-1, but not the non-inflammasome-related caspase-3. Looking at protein mediators in the same CSF of the same patients, what we saw again was a very strong pattern of either neutrophil chemoattractants or neutrophil mediators such as CXCL8, lipocalin-2, human neutrophil peptides 1 to 3, and this S100, A8, A9 proteins. We then compared TB meningitis with cryptococcal meningitis, which is another form of opportunistic and severe meningitis in HIV-infected patients, and began to discern quite distinct patterns of, in TB meningitis, IL-1 beta, matrix metalloproteinase 1, matrix metalloproteinase 3, interferon gamma, and CXCL10 production that were not characteristic of cryptococcal disease, which conversely was characterized by a greater IL-17 signal. And those preliminary findings comparing TB meningitis with cryptococcal meningitis has led us on to a multi-omic comparison arising from a diagnostic study that we conducted, and to actually begin to try to dissect transcriptomic patterns in the cerebrospinal fluid itself, which has been a technical challenge. But the study that I'm going to base the rest of the talk on involved 25 controls from whom there was no evidence of neurological infection, 8 with viral meningitis, 24 with cryptococcal meningitis, and 27 with TB meningitis. And looking at first of all, at the transcriptome, so the method of deriving the CSF transcriptome took us perhaps the best part of 2 years to derive, because it's not as straightforward a material to work with as peripheral blood. But what you see in terms of the CSF transcriptome in definite TB meningitis compared to cryptococcal meningitis is overrepresentation of type 1 and type 2 interferon signaling, IL-1 signaling. There is now an annotated pathway called M. tuberculosis infection, and so we felt quite pleased to have hit that one, and inflammasome activation. And by comparison, cryptococcal meningitis tended to be characterized by a signature that was more dominated by a pattern actually of metabolic derangement in terms of GABA uptake and degradation and blocking of NMDA receptors by glutamate, and a synthesis of glycolipids. To confirm our findings with respect to the limited proteomic analysis we'd hitherto undertaken, we actually undertook what is referred to as aptamer-based SomaScan profiling, which is 5,400 analytes in the CSF. And in order to gain confidence that the findings that we were observing from that technique, which are relative, not absolute units, we looked immediately at interferon and IL-1 beta, and those showed patterns that were consistent with the Luminex data that we'd observed in the same materials in a more limited study. So we're fairly confident that this technique renders good information. So looking now at what is differentially abundant in terms of TBM by comparison with cryptococcal meningitis, what we see is and there's a lot of power in this study, which accounts for the FDR-corrected p-value, is inflammatory signaling. Again, we've hit this pattern called tuberculosis, which is, as I say, reassuring. Hypoxia-inducing factor 1 signaling, neurotropin signaling, and necroptosis, a strong signal of necrotic cell death. There were lesser numbers of differentially abundant proteins specific to cryptococcal meningitis, but again, what you see here is quite a distinct and different pattern. Yes, innate immune response is there, and neutrophil degranulation and apoptosis, but also this metabolic pattern, sugar metabolism and glycolysis, that is distinct from TB meningitis. So we went on to re-examine the transcriptomic findings in TB meningitis specifically in a third study, and that's of children with TB meningitis. And in this case, we had the advantage of being able to sample both the ventricular, the lumbar CSF, as well as the blood. And this clustergram shows the ventricular CSF by comparison with other infections. And again, what we saw in the context of TB meningitis, and which was novel, was a transcriptomic pattern that suggested that there was patterns of metabolic derangement set up by inflammation that persist beyond the initial phase of inflammatory response. And those were GABA degradation, NMDA receptor binding, and glutamate release. And that tied in with observations that have been made by one of our colleagues, Reinout van Krevel from Radboud and in Indonesia, that a low level of CSF tryptophan characterizes TB meningitis. So we measured that again in these samples and reproduced that finding, and then we looked at the metabolites involved in the degradation of tryptophan. Kynurenine, for example, is a moderately immunosuppressive metabolite. Quinolinic acid, by contrast, is an inflammatory one. And the ratio of quinolinic acid to kynurenic acid is believed to be important in terms of the resolution of infection. The other thing that we've noticed in this data set is, aside from the inflammatory cytokines, which are shown in the top left, is that, again, as a consequence of that inflammation, even after inflammation has resolved, that TB meningitis causes significant brain injury. And that's evidenced by the production or the detection in CSF at greater levels than controls viral meningitis or cryptococcal meningitis of this glial fibrillary acidic protein, UCHL1. And then on the right-hand panel, LARC2, which is associated with both Parkinson's disease and with Alzheimer's disease, tau and alpha-synuclein. So I hope I've shown you that the life-saving therapy for HIV-TB is not infrequently complicated by this iatrogenic inflammatory syndrome, TB iris, and that what underlines that syndrome, notwithstanding that the most characteristic effect of antiretroviral therapy is to expand the CD4 pool, is actually an innate response dominated by inflammasome activation with downstream inflammatory consequences. I've shown you that TBN iris in the brain is severe, and it's contributed also to by inflammasome formation and neutrophil activation. That's a very distinct pattern from the inflammation of cryptococcal meningitis, and that TB meningitis is also characterized by metabolic abnormalities and evidence of neurodegeneration which can persist and perhaps contribute to the disability that is observed in TB meningitis. TB meningitis causes the death of about 40% of HIV-infected patients that contract it, but the disability that may result as well is extremely important. The translational consequences that we're investigating by randomized controlled trial, in fact by 2 randomized controlled trials going forward, are more effective and specific anti-inflammatories. It would be very nice to inhibit the inflammasome, but there's nothing ready or nothing at the time of designing these trials that was ready for prime time. There is evidence that aspirin may have a useful both anti-thrombotic and anti-inflammatory effect in TB meningitis, and we're involved in trials of that. And the metabolic findings, although early, suggest that perhaps metabolic interventions may also help to reduce some of the brain damage that characterizes this unpleasant condition. So I thank you for your time, and we'll be happy to take questions. Javid, I guess I
Thank you. Thank you very much, Professor Wilkinson, for a nice holistic view about TB meningitis. So before people start asking questions, I would like to ask a question. In many of your data, it has been observed that there is quite overlap in case of pro-inflammatory molecules with TB meningitis patients and the healthy individuals. So what does these individual indicates? That these individuals... These individuals, they show almost, some of the individuals, they show almost like data equivalent to healthy individuals. Your voice is not, you're not audible, Robert.
Yeah, sorry, Javid. Can you hear me now?
Yeah.
Yeah, sorry, the program, the whole browser crashed for about 30 seconds, fortunately, just at the end of the talk. So I didn't hear the first part of your question. Do you want to ask it again?
Right, the question was that in your data, the responses concerning pro-inflammatory responses, most of the data within patient and healthy individual, it was overlapping. So how are you going to comment on that?
Yeah, also this is a factor that this is something that we've observed for, the best part of 25 to 27 years in human tuberculosis. And I think it's a big theme in tuberculosis research in terms of its immunology going forward. This is heterogeneity. You rarely see very consistent patterns. And the fact that no 2 cases of TB meningitis are ever the same in terms of their clinical presentation or their management, I think backs that up. I think you get very heterogeneous responses to the presence of TB in the brain, and that's reflected at both the macroscopic and microscopic levels. Some children, for example, get obstructive hydrocephalus and a predominance of granuloma formation. HIV-infected people tend to get this severe leptomeningitis made worse by inflammation, and that can cause pseudo-obstruction. So the response varies by age, by HIV status, even by gender to some extent. So I think that heterogeneity is just something that we have to approach by adequate power.
Okay, so now it's open for question answers.
Andrea Cooper, question to Rob. Does the innate response... the question disappeared, Andrea. Perhaps you better ask it.
Okay, so... Okay, Andrea, go ahead.
I'm just wondering, so you were talking about the innate response being is the innate response a consequence of the aberrant T cell response as a result of antiretroviral treatment, or is it clearly T cell independent? And why then does the antiretroviral treatment promote it?
Yeah, I mean, so we thought about this, and I don't have proof for what I'm about to say, but you can observe the syndrome of iris as little as 2 to 3 days after starting antiretroviral therapy. And I think that leads us to think what antiretroviral therapy actually does. What it does is it prevents HIV replication. And so what you're presented with is potentially a very large intracellular cytoplasmic RNA load. And what our present suspicion is that is detected by RNA receptors that are designed to trigger innate inflammatory responses. Now, I think during the course then of the next few days and weeks, Andrea, you do get restoration of CD4 T-cell responses, and you do see associated T-cell expansions in this syndrome. And I think that those amplify the inflammation, but I think the trigger is definitely the death or the loss of viability of HIV with a consequent increase in free RNA within the cytoplasm of antigen-presenting and other myeloid cells.
Thank you. That sounds like a reasonable explanation.
But addressing that experimentally in humans, Andrea, is a challenge.
I can imagine. Thank you.
So there's another question from you from Hilal Bashir. He asked, have you tried to find correlation between kynurenine metabolite and Treg induction, as there are many studies related to this?
Yes, I know. And the straight answer is no. We've not looked much at so those metabolic changes are documented in the CSF, and we haven't done a large amount of work on lymphocyte activation or lymphocyte phenotyping within that CSF. I think that is an interesting thought, but it's not work that we've done so far. I might say that an early hypothesis that went around about the cause of iris was defective Treg induction. And we did look at that, and we saw no evidence of differences in either Treg numbers or to the extent that it's possible to measure it in Treg function between people who did and did not get iris, although those were peripheral blood-based analyses. And as I said, the degree to which we can measure function is limited. We infer it by the presence of simultaneous surface markers.
So there's another question for you. This is by Aditi Singh, and she asks that will latent TB without HIV infection background increase the susceptibility of developing TB meningitis in an individual? How TB meningitis is different from classical neurodegenerative disease?
Yeah, okay, so there's two parts to that question. So TB meningitis is the most at-risk group, and overrepresented by about fivefold is children. And I think that what we envisage there is that the TB meningitis arises as a consequence of primary and early dissemination, that bloodborne TB makes its way into the central nervous system and causes the syndrome. And I think we recognise that that's one of the best actions or best documented actions of BCG, or at least neonatal BCG, in preventing that disseminated form of disease. So I think in adults, the presence of latent TB infection per se is obviously a lifelong moderate risk factor for the subsequent development of TB. If you're HIV infected, the risk increases, and the risk of disseminated forms of disease increases in HIV infected people. But merely being TST positive has no predictive value of whether you will get TB meningitis. Now, the second question, sorry, could you just say it again, because I've gone on for so long in the answer to the first one that I've forgotten what the second question was. Javed's on the phone. Could you just ask the second part of the question again, because honestly, I'm not playing games, I've just forgotten it. we seem to have gone. Can anyone hear me?
Yeah, we can hear you very well. So,
yeah, sorry, Javed. I just the second question. What else? It slipped my mind.
The second question was how TB meningitis is different from classical neurodegenerative disease.
Well, I think the answer to that, to some extent, is self-evident. TB meningitis starts as an acute intracerebral inflammatory disorder. In fact, I don't think there are many, apart from bacterial meningitis, and intracerebral inflammatory disorders that are more furious. But what we're seeing is that the long-term consequence of that, as a consequence of inflammation that's set up, and despite the relatively effective killing of bacilli and the resolution of inflammation, that a pattern of brain injury seems to be set up, which in patients who go on to become disabled, and we've best shown this in children, for data that I didn't have time to show for reasons of time, it's associated with these markers of neurodegeneration. So our hypothesis is obvious. It's obvious that inflammation sets up the damage, but that brain damage via both metabolic and ultimately neurodegenerative mechanisms may persist and continue. And it's finding adjunctive treatments for that and stopping the resultant disability and mortality that occur that's a big challenge in this disease.
There's a question from Arti Chauhan. And,
yes?
No, sorry, this is from Tantre Shafia. And she wants to ask a question that is, tuberculosis bacteria always release a protein, PTPA, protein tyrosine phosphatase. If not, then it can't inhibit the VATPase pump.
Right. I don't dispute that, Javed. I'm just not sure what the answer to the, or whether the question is, what the question inherent in that is. I'm quite prepared to believe the statement, but I'm not sure.
Okay, so there is another question from Anuj Bandral. This is regarding do M72/AS0IE vaccine for latent TB have same efficacy in case of persons with HIV? Can this vaccine show efficacy in other TB patients?
Yeah, and that's a nice and interesting question, which is relatively easy to answer because the only clinical trials evidence for vaccine protection of M72-AS01E is against pulmonary tuberculosis in HIV-infected adults. We are very interested, so this is wearing a different hat as an investigator in that trial. We're very interested in the possibility that it might have a protective benefit in HIV-infected people and indeed against disseminated forms of TB, but that there's no present evidence that allows us to make that conclusion. I think experimentally it's also quite difficult to ascertain, and this is a problem that occurred in, for example, the earlier trials done by our colleagues at SAPI, that when you do a vaccine trial for TB, you watch your patients really carefully, and at the very first sign of disease, you treat them. So establishing that a new vaccine would protect against a very nasty and disseminated form of TB like TB meningitis is actually quite difficult because you do not want the disease in your trial participants to progress to such an extent unobserved that they end up with a very severe manifestation like TB meningitis. I think that kind of data could probably only arise from what's referred to as phase 4, and that's actually post-registration, post-deployment, and post-marketing surveillance. But I think it's an interesting question, and thanks for asking it.
So there is a question by Ashanka. Can similar approaches, that is whatever you use, be used for meningitis caused by Neisseria meningitis?
In terms of therapeutic approaches or in terms of immune modulation or obviously meningococcus causes very severe disease as well. Its specific antibacterial therapy, of course, is different. There is limited evidence in the developed world that adjunctive steroids are beneficial, but in lower and middle income countries, the conclusion is that adjunctive steroids in other forms of bacterial meningitis, such as pneumococcal meningitis and meningococcal disease, are in general not felt to be beneficial. I'm not aware of studies that have looked at neurological sequelae in meningococcal disease that have detected the same patterns of metabolic and/or brain damage. Obviously, that might be of interest.
So when there is now, this is I think questions there are probably over here. There are many appreciation for you. People like this talk very much. And can I ask you questions? Still, we have 5 minutes, so let me take this opportunity.
Yes, of course. You can ask about
right.
anything that you want that you assume that I've got competence to comment on.
Right. So that during TB meningitis, there's a brain injury. During TB meningitis, there is brain injury.
Yes.
So the molecules, especially which are causative, which are present, or ultimately they are shed in brain. This may be because of brain injury, brain molecules, or MTB molecules. Can you monitor? Can you detect these in blood as well, or these molecules can be used as a diagnostic for injury in brain or the severity of meningitis?
Yes, that's an interesting. We've not actually looked in blood to see if they actually spill over. That's an interesting question to know if that biomarker was present peripherally, whether that would predict a poor prognosis. I think there's a hint of it from the peripheral blood transcriptome. But I think, that's an interesting question, and it's not one that we've explored actually, whether you can prognostically know who's going to have a poor outcome. I mean, there are clinical scales that give you an idea who's going to do well and who's going to do badly with TB meningitis, and the one that's best validated is the British Medical Research Council staging scheme into stages 1, 2, and 3. And if you're in stage 3, then that tends to be associated with a poor outcome, particularly if you're HIV infected. Another major risk factor for death and very poor outcome in TB meningitis is drug-resistant tuberculosis, which is very, very difficult to treat.
What is the situation of M1 macrophages during TB meningitis, M1 M2 ratio?
Well, Jarvin, again, I have to confess that I haven't specifically looked for them, but looking at the patterns of transcriptomic disturbance that we see, one would strongly suspect that they're seen because there is a substantial inflammatory signature, but we've not immunophenotyped them specifically in terms of cytokine production. That's something that we're intending to do going forward in the 2 clinical trials. There's surprisingly little work being done on the immunology, at least in the contemporary area, on TB meningitis, or in fact on any form of TB at the disease side. In humans, obviously.
Okay, so any more questions? There is one more question posted, and if you can handle that, what renders it difficult to develop vaccine for HIV? Is it a high mutation or invasion?
That one might be beyond my pay grade, Javid. But I mean, I'm interested in tuberculosis and HIV interaction, but I'm definitely not a HIV vaccinologist. Our clinical site was involved in the latest HV702 trial, which was very disappointing because we thought that was the rational approach. We thought that there was a combination of vaccines there that would induce an immune response similar to that which was seen in the RV144 trial in Thailand that led to 30-odd% efficacy, and we observed nothing at all. I think it's one of those problems that one would have to describe as wicked. I think the consensus of opinion is that you need both a cellular, both CD8 and cytotoxic response, but most of all, the money seems to be on broadly neutralizing antibodies. But obviously HIV vaccines have a distance to go, and I hope that we can be somewhat quicker in developing one against SARS-CoV-2.
Right, and do you have any future program and looking into correlation between TB and COVID-19?
Yes, we do. But I think that's probably, we're worried that TB might predispose to severe COVID-19, and conversely that COVID-19 might set up an immunological pattern that would lead to the greater reactivation and progression of TB. But I think it's beyond the scope of this talk to detail much on that at the moment.
Okay, so if there aren't any questions, let us give a big applause to Dr. Professor Wilkinson for a nice overview about the meningitis. So thank you very much, Robert. And sometimes we'll invite you over here at IIT Ropar. It's foothills of Himalayas, just 40 kilometers ahead of Chandigarh.
Yes, I do apologise, Javed. I've got to leave now because,
okay.
Surprise, I've
Thank you very much.
Meeting about COVID-19.
Okay, good. Good luck. Good luck with COVID-19. So let us introduce now another Professor Wilkinson, and this time it is Dr. Catalin Wilkinson. So Dr. Catalin, she did her PhD from Lyotos University, Budapest, and then currently she's working as a principal laboratory research scientist at Francis Crick Institute in London. And her interest is on tuberculosis and immune response. And today she'll be speaking on effect of antiretroviral treatment on the immune response to tuberculosis in HIV co-infected persons. So we welcome you, Dr. Catalin.
Thank you for that introduction. I'm going to put my slide up. Can you see my slides?
No, we can see. Yeah, it's coming.
Okay, I'm going to try and put it on full screen.
Good.
Hopefully,
yeah. Still not full screen. Your slides are not visible. Yes, it's full screen. Good. So you're going to start your presentation. Catalin, we can't hear you. I think your mic is not working.
okay, can you hear me now?
Yes, we can hear you now.
Perfect, and hopefully still full screen.
Yeah, good.
Okay, all right. Let's hope there'll be no more problems.
No more problems.
Thank you for this opportunity to present my work today, and hopefully there'll be no more breaks in the communication. And I'm going to talk to you about my main research interest, which is the effect of antiretroviral treatment on the immune response to TB in HIV co-infected people. And I don't need to introduce the problem of TB to this audience, but I just want to emphasize that apart from the global burden of TB, which is a major problem in sub-Saharan Africa, there's also the superimposition of HIV-associated TB, as you can see here in purple, the countries which are most strongly affected by the dual infection of HIV and TB. And this is illustrated here in this township on the outskirts of Cape Town called Khayelitsha, which is only half a 30-minute drive from the center of Cape Town where our lab is. And the TB incidence in this township is 1% a year, with 4,000 cases every year, over 4,000 cases a year, 60% of which is HIV associated. So the risk of TB in HIV co-infected people is increased at all stages of HIV infection, and they are up to 30 times more likely to develop TB compared to HIV uninfected people who have a 5 to 10% chance to develop TB in their lifetime. HIV infected people have a 10% chance of developing TB every year. And antiretroviral treatment is the most effective way to reduce the risk of TB in these HIV co-infected people, with antiretroviral treatment resulting in a reduced risk, which is between 50 and 90% at the individual level. So this is why it's important to understand the effect of antiretroviral treatment on the immune response, because it doesn't have anything to do with the Mycobacterium tuberculosis itself. It has an effect on the immune response that can help us to find protective and pathogenic mechanisms that we can then use to turn around and come up with something that's protective in TB. So this is illustrated on this graph with the CD4 count related change in TB risk before and after starting antiretroviral treatment. You can see with falling CD4 counts, the risk of TB increases. As soon as they start antiretroviral treatment, the risk of TB decreases together with increasing CD4 counts. And the majority of people will undergo what I call protective immune reconstitution. And the subset will undergo the pathogenic immune reconstitution that you heard about in the previous talk, which is the TB-associated immune reconstitution inflammatory syndrome, one of which is TB meningitis iris. I'm not going to talk about TB iris. I'm going to tell you more about the protective immune reconstitution, where the hypothesis is that longitudinal analysis of this highly susceptible group of HIV-infected people with low CD4 counts undergoing immune reconstitution as a result of antiretroviral treatment and therefore become less susceptible to TB. And this contributes to our understanding of protective mechanisms in human TB. So we aim to understand this protective mechanism by identifying correlates of reduced TB risk in the form of cellular, soluble, and transcriptomic biomarkers. So based on this hypothesis, we did a preliminary study many, many years ago where we longitudinally followed a small number of HIV-infected adults for a year on antiretroviral treatment. And what we described is that in the context of overall improved TB-specific T-cell responses, it was the central memory, early differentiated phenotype, rather than the late differentiated T-cells that best correlated with decreased susceptibility. And later on, through the years, the importance of early differentiated T-cells in protection against TB were confirmed in a number of animal models, as well as various vaccine studies. So we carried on with this hypothesis to try and identify in more detail what is going on. And so we performed a second similar study with the aim to understand how MTB antigen-specific T-cells recover during early antiretroviral treatment, as well as look at soluble and transcriptomic markers. So the main difference is that in this study, we mainly looked at bulk CD4 T-cell responses, where here we went more deeply. So for the study, we recruited 50 HIV-infected people starting antiretroviral treatment in Khayelitsha and followed them at 1, 3, and 6 months of antiretroviral treatment because we really wanted to focus on the first half of the year to see what happens. And so we collected blood for PBMC separation. We performed quantiferin and ELISpot for to ascertain MTB sensitization. We collected plasma and also blood in tempus tubes for RNA extraction. And we also recruited an HIV uninfected control group from the same township who were only sampled once. And so this graph here summarizes the quantiferin and ELISpot responses over time. And we deemed everybody sensitized to Mycobacterium tuberculosis by at least one positive response in at least one assay during the 6 months of follow-up. So everybody experienced CD4 T cell reconstitution in the study and viral load suppression. There's one outlier here, but there's always an outlier just to spoil the trend. Obviously, we didn't have samples from every patient to do all the different analyses. So for flow cytometry, we had available samples because we need quite a lot of cell numbers for intracellular cytokine staining. And so we had 23 PBMC, 23 patients with adequate numbers of PBMCs from all time points to be able to perform this set of analysis. All of them were MTB sensitized, none developed TB during the 6 months of follow-up. And we also had 28 HIV uninfected patients, people with enough PBMC stored for FACS analysis. So here you can see the memory profile of total CD4 T cells during the first 6 months of antiretroviral treatment using CD45RA and CD27 to phenotype them as naive, early differentiated, late differentiated, and terminal differentiated. And what we saw here is that the memory profile normalized within the first 6 months with an expansion of naive subsets, an expansion, relative expansion of the early differentiated subset, and proportional fall of the late differentiated subset within the first 6 months. CD4 T cell activation was ascertained by HLA-DR expression on CD4 T cells, and we saw that this decreased, but it remained high compared to the HIV uninfected people. And it was in correlation with CD4 count. So the higher the activation level on the CD4 T cells, the lower the CD4 count at baseline, as well as 6 months. Here you can see in blue the 6 months where the higher CD4 count meant lower activation level. But in those who didn't really have an increased CD4 count to the higher levels, they still had relatively higher levels of activation. So looking at the MTB antigen-specific CD4 T cell response at baseline, we used MTB whole cell lysate to try and get as many antigen-specific T cells as possible, and we deemed cytokine-responsive cell, every single cell that responded to MTB whole cell lysate, either by IL-2 secretion, TNF secretion, or interferon gamma secretion. So these were the 3 cytokines that we looked at in this analysis. So cytokine positive cell means response to in any cytokine detection. We had a very stringent cutoff, which was twice the background. And we were quite surprised to see that half of the people that we had PBMC from at baseline had no detectable MTB-specific response based on our stringent cutoff. So, for example, these people didn't have twice the background response, so they were deemed as non-responders. It's also known that low frequency of T cell responses can be more difficult to visualize by intracellular cytokine staining compared to the ELISpot, for example. But nevertheless, so we grouped the people into those who were deemed to have no detectable response using intracellular cytokine staining and those who had a detectable response. They had no difference in CD4 count at the baseline. And those who had no detectable response at baseline, we saw no detectable response during the 6 months of antiretroviral treatment. And then we looked in more detail at their T cells, and we saw that this is possibly due to heightened turnover in these patients, because when you look at HLA-DR expression on their CD4 T cells, so those who had no detectable response at baseline, they also had higher expression of HLA-DR compared to those who had detectable response. They were more late differentiated, so more advanced in their differentiation state compared to those who had a detectable response. and the gain in CD4 T cells compared to baseline at 6 months was lower. So indicating low gain, increased memory differentiation, higher activation, possibly indicating heightened turnover, leading to lack of detectability on intracellular cytokine staining assays. So in those who had a detectable MTB-specific CD4 T cell response at baseline, we saw no change in frequency over time, but we saw a significant increase in absolute numbers during the first 6 months of antiretroviral treatment, and this is obviously due to the CD4 T cell reconstitution. And HLA-DR expression on the surface of these MTB antigen-specific CD4 T cells significantly fell, and this was quite a sharp fall of a median of 1.84, almost twofold at 6 months of antiretroviral treatment. And it's important to point out that the MTB-specific CD4 T cell activation mirrored that of the total CD4 T cell activation, both at baseline and 6 months. Functional profile was analyzed looking at TNF, IL-2, and interferon gamma intercellular cytokine staining, and we saw a significant increase in the proportion of cells expressing all 3 cytokines. So polyfunctional CD4 T cells significantly expanded during the first 6 months of antiretroviral treatment, as well as cells positive for IL-2 and TNF, so indicating that these antigen-specific CD4 T cells regain their ability to produce IL-2. This is also illustrated here using these black arcs. So at baseline compared to 6 months, you can see that IL-2 secreting cells proportionally increased. So in summary, the absolute number of MTB antigen-specific CD4 T cells significantly expanded during the first 6 months of antiretroviral treatment, together with polyfunctional capacity and a concomitant decrease in activation profile. So to put this in context, we showed in a previous study that the activation profile of these MTB antigen-specific CD4 T cells, defined as interferon gamma secreting CD4 T cells in the study, reflects disease activity irrespective of HIV status. So here, when we looked at latent TB and active TB patients in both HIV uninfected and infected populations, we saw significant expansion of HLA-DR, KR67, and CD38 in those who had active TB, but HLA-DR expression was further elevated in those who had active TB and HIV infection. So considering that, we can conclude that the antiretroviral treatment-induced decrease in immune activation involves decreased MTB antigen-specific T cell activation, and that's how it contributes to reduce susceptibility to TB. And so if you envisage MTB infection as a spectrum of immune responses, where infection is eliminated without priming antigen-specific T cells by the innate immune response, to infection being cleared by the acquired response, infection being controlled with bacterial persisting in a non-replicating form, which is quiescent infection, to active infection, where the patient don't actually present at the clinic yet, but induced sputum can identify them as sputum positive at the subclinical level, to the top of the spectrum, where they present at the clinic with clinical disease, all related to bacterial load. And we know that the effect of HIV is to push people up on the spectrum. The effect of antiretroviral treatment is to push people down on the spectrum, except in those who develop TB iris, obviously. We know that giving them isoniazid pushes them down on the spectrum. So it would be interesting to speculate that antigen-specific CD4 T cell activation could be added as a benchmarking assay to people to identify them where they are on the spectrum, and if they have high levels of antigen-specific T cell activation, then they should be started on RPT prophylaxis. If they don't have high enough T cell activation, then they should not be given chemoprophylaxis, which obviously would save a lot of RPT being given to people who don't necessarily need it. And we did recently receive funding to develop such a quick flow cytometry assay, and then COVID-19 intervened, but hopefully we will still be able to carry on and do that. So we were also interested in looking at a change in soluble plasma biomarkers during antiretroviral treatment. So when we performed the quantiferin assay, we kept the supernatants, and we undertook bioplex, multiplex, milliplex assays and various ELISAs. And so here I'm just presenting a subset of these results in 30 analytes that was performed in 30 individuals who had available data at various time points, so we could perform a paired analysis. And we looked at antigen minus nil as well as nil alone. And this analysis was done at the CRIC by the biostatisticians. So they did 2 unsupervised clustering analysis, hierarchical clustering and k-means clustering, as well as principal components analysis. And the subset of patients who underwent this analysis also showed a decrease in viral load, as well as an increase in CD4 T cells. So this slide shows the change in analytes compared to day 0 at 1 month, 3 months, and 6 months. And what you can see is the 0 is here, obviously indicating no change. To the right is things that increase, to the left is things that decrease compared to day 0. And the red is the significance threshold. So everything that goes beyond the red is what survives correction for multiple comparisons. And you can see there's a lot of variation during this first 6 months. But the next slide, this one here, is a summary of all the change that survives multiple comparison over time. And in other words, it's the consistent change in analytes compared to day 0. And what comes out is R1 alpha and R1 beta antigen minus nil, as well as MCP1 nil, significantly decreasing over the first 6 months, and IP10 antigen minus nil increasing. And not surprisingly, there is a correlation between R1 alpha and R1 beta. And obviously, we can explain how R1 alpha and R1 beta and MCP1 decrease over antiretroviral treatment, indicating reduced immune activation as a result of antiretroviral treatment and reduced viral load. But I was quite surprised about the IP10 increasing. Bear in mind, this is antigen minus nil, so this is antigen-induced IP10. So I looked at the CD4 T cells that express the receptor for IP10, which is CXCR3, and that showed a significant increase over time during the first 6 months of antiretroviral treatment. So these are CD4 T cells being positive for CXCR3. So this is a chemokine receptor that's highly expressed on effector T cells. It plays a very important role in T cell trafficking and recruitment of effector T cells from lymphoid tissue to peripheral sites. It plays a role in memory T cell differentiation and function throughout the activation and differentiation pathway. And it's increased in line with memory T cell expansion. So looking at it that way, it's really not that surprising. And to me, overall, this data points towards a broadly improved capacity to respond to new infections through better responsiveness to antigen. So looking at the PCA analysis, what we can see is that the day note is essentially driven, here the red dots are day note analytes, driven by the R1 alpha and R1 beta antigen minus nil results that decrease over time. And the blue, which is essentially the 6 months analyte, are driven by IP10 antigen minus nil, which increases over time. So this kind of illustrates it quite nicely how a change in soluble markers during antiretroviral treatment shows reduced immune activation. And to further establish that this is the case, I did another Luminex analysis. This was a thermo 65-plex using plasma. So this was not supernatant from quantiferin tubes. This was just plasma collected at the time of blood collection. And obviously, I didn't want to put everything 65 analytes up here. It would be even more tiny and invisible to see. But essentially, everything that was detectable fell from day naught to 6 months. I couldn't find a single thing increasing. And so I just summarized a few things like TNF superfamily members and markers that are part of the interferon gamma response, including IP10 here. So this is just plain unstimulated IP10, as well as other things like ITAC, which decreased, that's CXCL11, and it's interferon-inducible T-cell chemoattractant, which is here, as well as MIG, which is monokine induced by interferon gamma, or CXCL9. So everything falls in line overall, indicating a decreased HIV-induced immune activation and inflammation during the first 6 months of antiretroviral treatment. And finally, we also did RNA sequencing analysis in a small number of samples. Unfortunately, our RNA, the sample quality was not great after the many years having been collected in Tempus tubes by the time we extracted it. So we had good enough quality to do RNA-seq analysis on 6 day node samples and 6 month samples, which were not paired. And we saw, again, this is whole blood RNA, that this could be the case for HIV immune activation, in inflammation, decreased T-cell activation, which we saw in overall bulk CD4 T-cells, as well as MTB antigen-specific CD4 T-cells, together with the improved T-cell homeostasis with increased numbers of and thereby a reduced susceptibility to TB. So I would like to acknowledge everyone who contributed to this study. Catherine, who helped me with flow cytometry, Rachel, who helped me with RNA sequencing, Nishta, whose PhD was the basis of all these statisticians, Deborah, Chris, and Gavin, and the advanced sequencing facility at the CRIC. And thank you for your attention.
Thank you very much, Dr. Catherine Wilkinson. So now it is open for question and answer.
Javed, may I ask a question?
Yeah, sure.
So thank you, Catherine. That was fantastic. Tour de force. Andrea Cooper here. Hi. I was interested in the IP10 in the plasma was dropping during treatment, but the antigen-specific driving, the only thing that would come up was the IP10. So I'm wondering if that, what you think the source of the IP10 in the antigen-specific response is, and why, and what that's sort of suggesting to us.
Yeah, so I was surprised about that because obviously there's a lot of data pointing to some papers even indicating it as a biomarker for TB. I didn't really think about the source, the cellular source of IP10. I was just thinking more in the broader context that so we had performed a previous analysis using quantiferin supernatants in a different cohort where we tried to look at can you prevent TB in people who are taking IPT, and so we had baseline samples in those patients and we compared them to those who went on to get TB. And we couldn't predict that, possibly because of the low numbers, but we could differentiate between prevalent TB and no TB. And in those predictive models, we had interferon gamma and IL-2 together with IP10. So it was in the context of gamma and IL-2 together. So I guess that is more of a T-cell source, whereas in this case, it's very likely not. I don't know. Would you have any?
I think I think that was pretty informative, wasn't it? It suggests that the IP10 is not being produced unless there's a stimulus.
Exactly.
And as the stimulus drops, it's going away, but it's required for maintenance of some kind of protective response.
So that's why I concluded that it's not really, in this context, is not specific to TB. It just happens to be the TB antigen that we see in context with. But I think we can conclude that it's overall better responsiveness to antigen.
Yeah.
also looking at the CXCR3 expression on T cells in general.
Yeah, fantastic work. Thank you.
Thank you.
Okay, any other question? So there's a question. Yeah, please go ahead.
Yeah, so basically those patients who are on ART, antiretroviral, so that means you must be reducing the inflammation as such, right? So is it that some of the new cells which were not recruited in the response earlier, now they have started recruiting, or maybe some of these cells actually are the convertants?
So that's also very likely what happens, that during antiretroviral treatment, cells that are sequestered in lymph nodes come back into the circulation, and that has been shown before. So yes, definitely you see expanding populations that were not present in the peripheral blood before.
Thank you.
So there is a question for you, Catalin.
Yep.
This is from Clayton Fernando. The question is, why you analyze only CD4 cells, not CD8 cells? Also in CD4, you have seen only HLA-DR. Is there any specific reasons you can please brief that?
Yes, so we did look at CD8 as well. And in general, we were really not able to find a lot of CD8 responses, possibly because we used MTB whole cell lysate, and yeah, we didn't really see much change in CD8 responses. And yes, we could have looked at other activation markers, but we had a limit on the number of markers we could use. We already had up to 14. So based on the previous results where HLA-DR seemed to be most prominent in HIV-infected patients, we just decided to stick with that one.
There's a question from Anjali Patel. Sometimes due to increased CD4 count, it can lead to hyperactivated and exaggerated immune response or the death of patient within 3 months. So why this therapy becomes severe in this type of cases?
Sorry, I didn't quite get that question.
The question is that what happens sometimes is an sufficient increase in CD4 T cell counts, and the patient get hyperactivated and exaggerated immune response it becomes the cause of death of patient within 3 months. So what is the reason?
So that's the TB iris. So that's the pathogenic immune reconstitution. Yes. So we didn't have any of the individuals in this cohort exhibiting any TB iris syndromes, mainly because they were not TB patients. So these were healthy individuals. So in other words, they didn't have TB. They were just sensitized and started on antiretroviral treatment. So this wasn't a TB patient cohort.
Somnath Pai questions, what recommendation can be provided to PL-HIV on HRT to prevent TB?
So it's IPT. We've shown that people taking antiretroviral treatment can be safely given IPT, and it does reduce their chance of developing TB.
So the question from Anuj Pardral, how HIV-associated TB differs from normal TB? Is this medication-based difference or anything specific?
So that's more of a clinical question in terms of manifestations, and I don't think I'm qualified to answer that question, not being a clinician.
Okay.
But in general, you often see more disseminated TB in HIV, in the context of HIV. But no, I really shouldn't try to answer that question.
So there's another question. Was it a combination of agents that was used in ART? If yes, can you please name the drugs used?
Okay. So they had a standard antiretroviral treatment that was available at the time of recruitment in South Africa, and that was between 2015, 14, 15. I'm not sure what the standard drug regimen was at that time, but they all had the same regimen.
Okay. So another question is there any possible way to use combined form of BCG and ARD to treat TB HIV?
BCG is given to all South Africans at birth, so they don't, it's not part of the treatment regime.
Okay, so you cannot give it BCG twice, isn't it?
Well, that's a different question. They don't normally give BCG as a treatment agent in South Africa.
What is the experience in case of memory response in TB patients who get, or HIV patients who get infected with TB?
So the antigen-specific responses tend to be more terminally differentiated in TB patients, and those, so there haven't really been large enough studies to look at that and properly, but we did look in HIV-infected patients who had pericardial TB, and we found that compared to those who didn't have pericardial TB, the differentiation of antigen-specific T cells was far more advanced in those with pericardial TB. So we concluded that HIV co-infection drives the differentiation of memory T cells towards a more differentiated phenotype, which also leads to more turnover and more death of these antigen-specific T cells. So that is one of the differences.
But this death, antigen-induced death of T cells, is it irrespective of the antigen specificity or like epitope specificity, or it is specific to one particular epitope of mycobacterial tuberculosis? Or the question is, the death is specific to particular epitope.
So that, yeah, that's an interesting point, because all the different studies use different stimulants. Some people use PPD, some, in this study, for example, we used MTB whole cell lysate, some people use various peptides. It's really quite difficult to come to an overall conclusion. There hasn't been a detailed enough study comparing that, how epitope specificity drives T cell differentiation, and is it specific to certain TB epitopes? Yeah, that's quite a complex study to answer that question.
Right.
You have to do a lot of comparisons.
Okay.
In large
so
numbers.
For you, this is, can RTD1 theta defensin-based therapy class of AMPs be a future mode of therapy against HIV plus TB patients?
Wow, I cannot answer that question.
Okay, so let us thank Dr. Katalin Wilkinson for a very interesting talk, and let us give a big applause.
Thank you.
So thank you, Katalin, very much, and we highly appreciate that you joined us. And there are about 1,500 people watching this online program.
Thank you very much.
So now we welcome. Yesterday till evening, there were about 2,700 people who registered for this event. and it's a good number of people, 300 people they are watching. So
Yes.
now the next speaker is Professor Andrea Cooper. So another very exciting talk. And so Dr. Andrea Cooper is, after receiving her PhD from London School of Tropical Hygiene and Medicine, she moved to National Institute of Health, Bethesda, and then she moved to Colorado State University and worked with a very pioneer person, Ian Ahmed. And later on, she became a faculty at Trudeau Institute. And currently, she'll be talking about T cells in trouble, cells that can function in tuberculosis. And she's a faculty now at the University of Leicester. So we welcome you, Andrea.
Okay, thank you very much, Javed. I'm delighted to be here. Can you see my slides okay? And can you hear me?
Very much. We can see your slides.
Excellent. And so I was fine. I wasn't nervous until you mentioned how many people were watching. So I'm just delighted to be able to talk to so many people, but also a little bit nervous about the same fact. So it's just a great opportunity for me to follow on from two excellent presentations talking about T cells and TB. And I think we may be able, I'm principally working in animal models and understanding how T cells work in microbacterial infection in animal models. And so that's the kind of focus of my work. And I think we can put in some interesting comments about how T cells behave in a microbacterial infection. So I'm very simplistic in my understanding of TB, and it's a very confusing disease. And so I use this symbol here. This is the epidemiological triad for TB. And we know that 90 to 95% of people who are infected do not develop disease. And that's just an amazing situation. We have this disease that's covers the world. Many people are infected, but most people don't get disease. So why is that? And we know that there's changes in the bacteria can influence progression. We know that the environment and the situation that people find themselves living in can drive this TB. And of course, the host themselves drives susceptibility in a variety of ways. And we know that it's a combination of all of these features which will allow tuberculosis to occur. And that's this picture here of a lung that's full of lesions. Now, the simple thing that we know about TB, or the simplest thing we think we know, is that the bacteria lives within infected cells, infected phagocytes, and that we think antigen-specific T cells come along and instruct these infected phagocytes to stop the growth of the bacteria. And so if that's the case, we know quite a lot about this. Why have we not been successful in driving a vaccine that stops all disease occurring? And you can see from the previous talks, Ruth, there's the complexity of the disease and the ability of the T cells to mediate protection is variable depending on the status of the host. So is this model too simple? And what are the critical variables that we need to think about? I think in the first instance, one of the first things we see, we always see mycobacterium tuberculosis associated with phagocytes, and we see a very complex accumulation of cells. And here we can just see the bacteria sitting in an accumulation of cells. And this is actually a lung tissue here. And it's obviously not, it doesn't look like a normal lung tissue because we're full of cells that shouldn't be there. So we know, for instance, in this first instance, the interaction, in this immunofluorescence picture here, we've got red T cells, green bacteria, and then blue macrophages. So in this situation, how do those T cells really overcome the barriers associated with the macrophages that are infected? So in a simplistic manner, if we look at this, we've got a situation where the effector T cells may be present in the body and may be functional, but how do they contact the infected phagocyte when there are so many other phagocytes or cells around that are interfering with that communication? And so that's the first block for this to stop this occurring. And we also know, of course, that the bacteria themselves have a lot of elaborate mechanisms on their surface, a lot of elaborate molecules that can drive differential accumulations of cells, which may provide a lot of cells arriving at the site of infection, which then provide a nice place for the bacteria to grow, but then those cells are not then that responsive to the antigen-specific T cells. So there's a lot of manipulation by the bacteria. We need to know about what else happens as well. So in addition to an inflammatory mononuclear inflammation, we can also, in the context of the person and the environment and the ability of the individual to mount an immune response, we can imagine that this mononuclear inflammation may be compromised by the arrival of pyogranulomatous inflammation, and that's what's shown here, as we saw in the TB iris. When you've got a high purulent type of inflammatory response, then here, I don't know if it's clear enough for you to see, but in this situation, there's very few bacteria in this mononuclear inflammation. When you've got the pyogranulomatous inflammation, the bacteria are just everywhere. This is an airway here. So you can see that what happens if you just have something that upsets the nature of the inflammatory response to the degree that it allows a pyogranulomatous inflammation to occur, then what happens then is this antigen-specific T cell, it's unable then to interact with the infected phagocyte. And in fact, the infected phagocytes are going to be disrupted and you're going to get extracellular bacteria. And in that situation, there's very little that this very effective immune response, which we know controls disease in most individuals, is unable to occur. And so then this is a product of both the nature of the innate interaction of the bacteria with the host, and then the ability of the host to mediate an effective immune response. And so that's summed up in this slide here. And this is my sort of the way I think about tuberculosis, and why I think the way I think about how the T cells and the cells can be very similar within all individuals. Obviously, there's variability, but the total number of antigen-specific T cells may be similar in individuals, but the effect of those T cells is very greatly modulated by the environment that they find themselves in at the inflammatory site. And so in this instance, in this instance, we have a very good effective T cell interacting with an infected macrophage, causing the macrophage to kill the bacteria, and that's a good outcome. And that's in our 90 to 95% of people who don't get disease. You may get inflammatory macrophages coming in that are not responsive to these T cells, and they then allow bacteria to grow, and then you can start to develop a more primary progressive disease. And then as this occurs, these T cells are still present, but if you get some pyogranulomatous inflammation, then that interaction cannot occur, and then these bacteria again will start to grow and outgrow the lesion, and then develop. And then we can move on to the situation where you've got perhaps a caseous lesion where the bacteria are free living. There's a lot of purulent inflammatory response. And in that situation, there's no way that these cells can actually mediate anything. And we know that what happens in these situations is that the lesion gets walled off. And so there's a balance. The immune response has a balance between mediating protection and then walling off and sort of living with the bacteria. And I think we find that in our very broad number of people who are infected, but don't develop disease. And so this is the way I think about the same level of T cell response, but different immunity. And I think that's very important when we think about T cell responses in TB. And of course, that different immunity is dependent on the host, the bacteria itself, and the environment that they find themselves in. So this is one of my favorite pictures. We took this a long time ago, but it's a really telling picture. You've got the blue is the macrophages, and there are F480 positive macrophages there. The green, these are live lesional sites where the green is the production of toxic radicals by the macrophages. And then the red in these pictures here are CD4 and CD8 T cells. And what you can see is that, and in this situation, the lesion is only, this is a wild type lesion. These cells are able to make nitric oxide. And you can see there's very few red cells, and this is a wild type animal. The number of T cells getting into the lesion and surviving in the lesions is pretty low, and that's what we see quite often. However, what we see here is if we remove the gene that allows these macrophages to make the nitric oxide, then you can see the lymphocytes are really able to pour in and be part of the lesional site and start to mediate their functions. So this is a very telling situation. In the wild type situation, there are factors which impact the ability of the T cells to penetrate the lesion, survive in the lesion, and mediate their function. So if we have that, keep that in our minds, and we'll think about the nature of the T cells. And I'm not going to speak specifically about effector memories or late developing cells or anything in particular. I want us to just think about a type, a cell that's just called T cell 1 and a cell that's called T cell 2. And if we just postulate that the T cell 2 is less sensitive to this inflammatory environment and the T cell 1 is more sensitive to the inflammatory environment, we can start to think about things that might impact the ability. And in fact, do we actually have these two kinds of cells? And if we do, then can we modulate them by vaccination? And that's what that's sort of what I'm going to talk about today. So, and then I think this is critical. If we need to develop effective vaccines, we do need to know what kind of T cell survives and functions in the inflamed site, not just circulates around in the blood. And then we need to be able to say, can we impact the functionality of T cells by vaccination? And those are two different questions. And so I'm going to address the first question first. And this is just some work that we've done over a number of years. And I'm just going to show you a sort of a little bit of a schematic. What we knew was that animals, mice that lacked IL-27 receptor, which is expressed on a lot of cells, but is also expressed on T cells specifically and is upregulated when they become activated. If you have cells that, what we wanted to know is, we knew if then an animal lacked IL-27 receptor entirely, then that animal was more able to reduce the bacterial burden after infection than a wild type animal, suggesting that IL-27 receptor was detrimental to control of TB. And in some work we did with the Wilkinsons, we saw that in human populations, expression of IL-27 was in fact associated with active disease. But in this situation here, what we wanted to do was really refine our question and ask, is it, if IL-27 receptor is expressed on T cells, is that then the mediator of the improved, is it IL-27 receptor on the T cells that is actually mediating the reduced ability to control infection? And so what we have here is bone marrow chimera models where we take T cell mice that don't have any T cells, and then we give them T cells that have IL-27 receptor. Sorry, we give them bone marrow, which is either IL-27 receptor deficient or B6 deficient, and we give them in a 75-25% ratio, and then we allow those mice to reconstitute. What we have in the end is a mouse that lacks T cells, but is reconstituted with T cells either with or without the cytokine receptor. And so in that situation, almost every cell in the body has IL-27 receptor except the T cells. And so then we wanted then to ask the question, if we do that and then infect those mice, and we deliver an aerosol infection using, it's a low-dose aerosol infection to the lung, somewhat mimicking a natural infection. And what we find then is that mice that lack the IL-27 receptor on their T cells are actually better at killing Mycobacterium tuberculosis, both at day 60 and then as we go forward to day 120. And this is important. What we're looking for here is a T cell that functions long term, not just initially, but long term, because we know it's very difficult to stop the infection with TB. What we want to do is maintain long-term control in a very refined and small lesion and not allow the bacteria to grow. And so we can see here, both at day 60 and day 120, mice lacking IL-27 receptor on their T cells are better able to control TB. So what does that mean in terms of their T cells? What are their T cells doing? And so we asked a similar question, but in this context, we used bone marrow chimeras, and we transferred in wild-type T cells that were expressed a marker, CD45.1, and into the same mice, we transferred bone marrow that had IL-27 receptor deficient bone marrow that had CD45.2. We put those dually into the same mouse, and this is critical. So now we've got a situation where the T cells, I've got T cells that are marked with one marker that are wild-type and T cells that are marked with another marker that are deficient. And we ask the question, when we go into those animals and ask the question, how do those cells survive equally in an animal during infection, we find some really striking differences. And so if we're just looking at here, this is just the flow cytometry, we're looking at the 45.1 and 45.2 cells. And if we just ask the question and look at day 20, day 70, and day 120, those are the days of infection. If we look at CD4 deficient T cells, the CD45.1 and the CD45.2 cells, if they're CD4 negative, if they're just any other cell in the body, then they retain a 1-to-1 ratio. But as we go up and we look at CD4 cells that are activated, and this is in the lung, if they're activated T cells in the lung, but they're not TCR transgenic positive, sorry, if they're not tetramer positive, so they're not specific for one of the major antigens, we can see that the ratio has started to alter. And in fact, the cells that lack the IL-27 receptor, are T cells in the lung that lack the IL-27 receptor, are more fit to survive and persist within that inflammatory lesion that is detrimental to T cell survival, suggesting that we've got a cell type which is better able to persist. And if we look at then our ESAT6 specific, the ESAT6 is an antigen on the Mycobacterium tuberculosis, which is very immunodominant. And what we see here is that the tetramer marked cells, even at day 20, they're really accumulating and persisting much more strongly if they lack the IL-27 receptor. And that profoundly occurs through day 70 and at day 120. So in fact, the only cells that are surviving in that context in the lung pretty much are the cells that lack the IL-27 receptor. And the CD4 T cells that are antigen specific that do express that receptor are very much in the minority, suggesting that, again, antigen-specific cells are very much more likely to be eliminated from these inflammatory lesions if they are IL-27 receptor positive. So then we ask the question, what does that expression do to the nature of the T cells? And so these are the cells from the day 96 post-infection, and we're looking at, and this is exactly the same environment. You can see that these cells are, in terms of IL-2 production and interferon gamma production, interferon gamma is about the same. So they've all been driven to express about the same level of interferon gamma, perhaps a little bit less intensely in the IL-27 receptor expression. But they actually then produce a lot more IL-2. And so what we're doing is allowing those cells probably to be self-locally replicating within the lesion, producing IL-2 and making a better response in the site. And so that was what we think then. We've identified a phenotype of a T cell that's then protective. And just thinking about, going back to this idea about where are the T cells and are they better able to accumulate in the lesion? If we just look at the lesional site of mice that lack IL-27 receptor in their lungs in terms of infection, what we did was a very simple sort of co-localization study where we just determined where MTB antigen was. We looked, and the blue there is macrophages again, and then we've got the T cells in red. And we just basically drew a perimeter around where the mycobacterial antigen was and asked the question, how much red pixel values were in these lesions? And you can see in the B6 animals, or the wild type animals, then the cells were, there's not a lot of red associated with the mycobacterial antigen. And you can see here, there's a sort of a large proportion of cells that are basically, you're not getting a lot of T cells getting close into the antigen. In the absence of the IL-27 receptor, where those T cells are then better able to survive and persist within the lesion, you can see that in fact there's a lot more red associated with mycobacterial antigen. And then what this really makes us think then is that these T cells are better able to mediate this function. They're better able to coordinate with the infected macrophages and cause them to control the disease much more effectively. Now, this work has been reported, and I sort of was quite swift as we went through it, but I wanted to get the idea that these T cells are, there are different kinds of T cells, and they basically, their ability to get into the site and interact with the infected macrophage is probably a very important element of their ability to function. And that reference there is the one that will allow you to look at that data in more detail. So I think we've suggested that we've got these, and there's of course a lot of other work in the field, and other people have been looking at this subject, and we know that there's a key element is the ability of the T cell to actually get into the parenchymal site and migrate within the lesion, and that's a critical function of these T cells. So, but in terms of their ability to function, we think we've got then our type 1 cells that we begin to think about. In a bunch of different studies, and also our study, we see that these cells are in fact PD-1 high, CD69 high, CXCR3 high, VLA4 high. They're parenchymally located, they're protective, and they produce IL-2. And then in these terminally differentiated cells, and this is what again Kathleen was talking about, these cells are very much more highly expressed in the people with active disease. These cells are then KLRG1 high, TBET very high, so they've been driven to a very high level. They make a lot of interferon. But in our studies and in other studies, these cells seem to stay within the vascular locations, and they don't tend to get into parenchymal sites. And whether they're not getting in or they're more susceptible to the compromised situation within the lesional sites, that's a question to study. So what we've got then is these cells that are more terminally differentiated, they're more sensitive to the inflammatory environment and less protective. And then these cells that are less differentiated are the ones that we like and the ones that we think will be productive and protective. So sharing that and keeping that in mind, can we actually impact functionality of T cells by vaccination? And so thinking about that, we knew that with the mouse model, we have the tools and the ability to be really very definitive in our experiments. And so what we wanted to do was just take the concept of TCR ligation and the strength of signal that you get between TCR and its the antigen and the presenting molecule, and we know that that's very highly specific and very sensitive. And there's quantitative and qualitative differences in the strength of that TCR reaction, where they can actually influence events far downstream of TCR recognition, including response following antigen and the functional aspects of the memory cells, such as T cell migration into tissues. So we thought, well, what we'll do, use our major antigen in the mouse model. So the ESAT6 molecule is early secreted antigenic target. It's 6 kilodaltons in size. It's highly expressed in all virulent strains of MTB. It's a major target molecule for the memory T cell response in mice and in humans. And its a CD4 T cell response is particularly strong in C57 black 6 mice, and it's only in its one presenting molecule. And so we only have one epitope, and this is the critical point. The ESAT6 epitope is the first 20 amino acids in this molecule. So we've got this here. Here is the sequence of amino acids in that protein, and this is the binding site. And we did this work with John Sidney and Alex Setti at Immune Epitope Database. We basically took peptides that were just shifted along the register, and we asked the question: do they bind into the presenting molecule, and how effectively are they ejecting the full length sequence from that molecule? And you can see that we've got a range of peptides here, all of which sort of go into the binding site and have different abilities to then eject the full length molecule. So they've got different affinities, and we wanted to know whether that would then impact the ability of these peptides to drive T cell responses. And just this is a sort of a lot of data sets that we've combined into one little note here. And what you can see here, these are just these are this is a signal, a flow cytometry signal coming from TCR transgenic T cells that have got the receptor for the ESAT6, and they express a molecule that glows fluorescently when they get a TCR signal. And so this red line here is the negative control. This positive control here is where all of the cells have been stimulated. And then we've got full-length peptide, shows that a good proportion of the cells get stimulated if you put full-length peptide in. We've got peptide AO2, which is down here. It's very similar. It drives a good, strong response, just the same as the full-length peptide. We've got AO1, which I can learn to understand a subset of the cells quite strongly. And then we've got peptide AO6, which doesn't drive many of the cells very well, but there is a reproducible response. And we also have done some studies just looking at the cells in vitro and found that the AO1 is very good at driving cytokine production in TCR transgenic T cells in vitro. It's not so good at driving activation. AO2 drives a lot of activation, but less cytokine production. And AO6 really doesn't drive the TCR transgenic T cells in vitro to do very much. And so that's a lot of data. We looked at lots of things in vitro, looking at the TCR transgenics, but we were concerned that would not be so very clear in terms of what was going on in vivo. But we took that in vitro data to allow us to give us a set of peptides, 3 peptides that we would use alongside of the full-length peptide to ask the question, what happens to T cells during vaccination? And we have a vaccine model which is basically it's basically just the peptide is the only thing in there that can drive T cells. It's got monophospholipid A, which is TLR4 stimulant, trehalose dicornamycalate, which drives the CARD pathway, and then dimethyl dioxyammonium bromide, which has lipophilic properties, acts as an antigen depot, and allows the vaccine to. We make an emulsion with these products and add in peptide. And when we do that with the full-length ESAT peptide, we published this quite a long time ago, that can give us 1 log of protection at day 20 post-infection, suggesting that we're inducing T cells that are effective at reducing bacterial burden very early on. And you can just see here, so naive mice have a 10 to the 6 in their lungs. If you vaccinate with this vaccine, with the ESAT6 full-length peptide, you get 10 to the 5 bacteria. If you use a non-specific peptide such as OVA, you don't get protection. And the sham, where there's no peptide, you don't get protection. So what happens when you do that in vivo? So we give the vaccination, and what we've got up here is just details. We basically take TCR transgenic T cells, put them into mice, and then give them the vaccination, and ask the question, what do those T cells look like? And you can see the different peptides. We've got the sham here, we've got the ESAT, we've got the AO1, we've got AO2, and AO6. And those were their very three different. This one was supposed not to stimulate any cells. This was good at stimulating activation, and this one was good at stimulating cytokine production. And then our ESAT, we know, is a protective cytokine, protective peptide. And what we find is when we deliver these and look at the cells day 4 after, this is day 2 and day 4 after vaccination, you can see that, and day 7, that we've got the ESAT6 is driving a very strong CD25 upregulation. It's also got a strong CD69 upregulation. You see the sham doesn't really upregulate CD69, showing that there's no TCR ligation. ESAT6, AO1, AO2, and AO6 all drive the cells to upregulate CD69. They also then, it drops down at day 4 and day 7, but CD25 upregulates very strongly in the ESAT and the AO6, and then variably in the other peptides. And I think that the key message from this is that we've got very different outcomes depending on the nature of the peptide that we're delivering. And you can see then that one of the key things there is that AO2 is very slow to express CD25 on its surface, but then everything resolves back down to day 7. Things are relaxed and get back to normal. So the question is, what's happening to those T cells? And so what we did as well, looking at those T cells at day 2 and 4, what are the instructions they're getting? And what are they changing in the nature of their response to the vaccine? And you can see then, if we just look at the sham, there's very little response day 2 and day 4 in terms of the expression of cytokines or transcription factors. But you can see ESAT6 is driving, it's difficult to see, but you can see day 2 and day 4, there's quite a strong upregulation of specific responses. And A1, AO2, and AO6 are quite different in the nature of that response. You can see at day 4 that it's basically AO6 has got a delayed upregulation of all of these markers. And I'm not going to talk specifically about any one of these things, but you can. And the key message to take away is that these different peptides, although they're very, very close to the original peptide, are having very different effects on the TCR transgenic T cells that are in there in the very early response. And just to remind you that the AO1 is not a great binder into the presenting molecule. AO2 is a good binder into the presenting molecule, and AO6 is a variable or intermediate binder. So what happens then if we just take, and then we're going away from TCR transgenic cells now, and we're just going to look at tetramer positive cells. So this is a much more natural then environment. The mice are vaccinated, they are exposed to the aerosol infection, and then for 15 days we're going to look at the the tetramer positive cells in the lungs and the lymph nodes. And you can see very first off that the ESAT6, AO2, and AO6 are very good at accumulating a large number of tetramer positive cells in the lung very soon after infection, day 7, and that's very good in terms of protection. We know that the sooner the T cells get there, the better in terms of bacterial control it is. And day 15 of infection, you can see that AO1 has caught up. The shams are still being very laggardly and not doing anything because we don't really see any T cell responses in the lungs for about 15 days. Only in vaccination responses are we able to see. And you can see, despite those differences in the response, the cells get in there and they can mediate protection. If we look at the phenotype of those cells, you can see again that day 15, those cells are actually looking different again, even though they're all present. The tetramer positive cells are getting a strong KLRG1 signal quite early on, which is not present in the other ones. And you can see here that there's variability in the expression. They've all got less PD1, and they've got a little bit more KLRG1. They're not profound differences, but we also see then differences in the cytokine expression. So AO6, which was our late arrival, is actually much better at producing interferon, a higher frequency of interferon gamma producing cells. IL-2 is not dramatically different. IL-17 is very strong in the AO2 induction, and then TNF is stronger again in the AO6. And so whilst this data is a little bit different from what we saw in the vaccination data, the key element is that these peptides are driving the cells to behave differently. So they've all been vaccinated previously. When they see the antigen, they're behaving differently in vivo when the antigen arrives. So what does that mean in terms of protection? So this is a day 20 post-infection bacterial burden study, and you can see very clearly here that we've got our protection is a little bit higher infection model here. We've got high level of protection in our ESAT6, but in fact, it's not dissimilar. All of them seem to have quite a good protective effect, even though they've got these different functions. And in fact, the AO6 was actually late to arrive. It got there in time to reduce the bacterial burden effectively. And just to remind you that these have very different ability to drive the T cells and to bind into the MHC. So why is that interesting? And why does this variety matter? So we've got all this variety here. They're doing all different things at day 15. They've got different transcription factors being expressed, and yet they're all doing about the same thing. Is it really important? Well, I wanted to come back to this model where we've got our inflammatory site and thinking about our T cells that survive. And so day 20, things are all, probably there's not a lot of lesional sites, the T cells are all able to function. But what we do know, by day 90, we've got an inflammatory site which is a lot of macrophages, a lot of nitric oxide production, a lot of drawing away of nutrients from the T cells. So we wanted to see, are those T cells still functioning? And if you look at day 90 here, you can see that all of the full length, the AO2 and the AO6 has been able to maintain the level of bacterial control. And what AO1 is doing exceptionally, which we've never seen before, it seems to be able to be reducing the bacterial burden over time more profoundly than the other ones, suggesting that those T cells are then mediating, they're staying there longer, they're mediating their function, and they're actually reducing bacterial burden. So just in this final few slides, I'm going to just discuss what looks different in the AO1 vaccinated mice. And what you can see, if we just look at them at the lung and the lymph node, so basically you're just looking at, the purple is interferon gamma, TNF, IL-2, IL-17, and IL-10. You can see that whilst the predominant T cell phenotype in these, all the other vaccinated animals is largely interferon gamma driving responses, that's not the majority in the AO1 stimulated cells. And this is at day 40, so this is halfway through the infection pathway. Suggesting that, again, it's this, but we know polyfunctional cells are important, and it seems like here we're really maintaining a strong polyfunctional phenotype in the lymph node where antigen stimulation is occurring, and also at the lung in the site of the infection. And this is probably an important feature. And this is all on tetramer positive cells. And then we wanted also to look at the nature of those cells in terms of their expression of PD1 and KLRG1, which are associated with, KLRG1 particularly is associated with inability to get into the site and not be able to function within the site. And PD1 is a marker that suggests that there's T cell activation occurring. And we find that PD1 is actually get into the lesional sites quite strongly. And so what you can see here, we're actually maintaining a PD1, KLRG1 negative population more strongly. Again, looking at this large section here, you can see that these populations are around a third of the population in the ESAT and the lung infection, but they're more than they're getting on for 2/3 here in the AO1 vaccinated animal. So again, I haven't got any definitive data. What is the protective mechanism going on here? But you can see that we've really generated a different phenotype of cell as a result of the vaccination early on, and which is really exciting in terms of vaccine development. So just looking at the transcription factors, they're a little bit, they follow a similar pattern in that we've got a differential expression, and RO gamma T is actually one of the stronger expression patterns, perhaps suggesting that these cells remain a little bit less differentiated than the other T cells in the other lesions. So coming back to one of the features, it may not be T cell dependent, this better survival. It may be that the environment is different. And so we've been also examining the environment in terms of the nature of cells that are accumulating. And you can see that in this situation, again, we've got a very large proportion of the cells that are neutrophils here, not knowing whether that's then important. but we've also got then a less proportion of different cells. So whilst that doesn't fit with our hypothesis, it does reflect the fact that the environment that's induced by the different T cells is probably influential in the outcome of the infection. So just to summarize all of this, we think that we've got different antigen-specific T cells, and I think that's pretty much that's well accepted in the field, and it's well accepted in the human situation too. These T cells are then circulating, and they can move around and they can function. And it seems like the less differentiated, the cells that are making IL-2, they're IL-7 receptor high as well. They're also making interferon gamma, but they're not making as much interferon gamma, and they also keep a low T-bet, a mid-level T-bet expression, which is a transcription factor for interferon gamma production. And we think those cells are more able to proliferate, they're more able to penetrate the granuloma, more resistant to the environment, and therefore are protective. Those ones that are more differentiated are less able to penetrate the lesion. Once they get into the lesion, they're very susceptible to the toxic environment in the lesion. They need, they're metabolically active, they need a lot of resource, and they don't have a lot of resource available to them. And the key thing that we've found in our studies just that I've just talked to you about is that the priming of the event can lead us to have a preponderance of those cells that are more likely to function. And I think this is critical in our understanding of how we then drive vaccine-induced cells. And so, yes, priming T cells with a peptide, which is not optimal for the antigen that we'll see upon challenge, alters long-term outcome. And we can see this in other studies where antigen 85, for instance, is less predominantly expressed in the lesional sites, and the antigen 85 specific T cells last longer than ESAT6 specific T cells. And in fact, in humans, excuse me, the ESAT6 specific T cells are more activated and more liable for loss. And that's a nice piece of work. That's another piece of work by Kevin Erdl and Tom Schrieber. So I come to the end of my talk now. I'd just like to thank Egidio Torrado, who did the work in the IL-27 receptor knockout mice. I'd like to thank John Pearl, who's my work colleague and also husband, and he also manages the animal infection models. Mingfeng Liao and Mariana Razande both worked on all of the peptide work models. And these are my two best projects, the children who have been listening to the talk as we've been, now that we can give these talks at home. And of course, all of the funding, NIH in the US, American Lung Association, Trudeau Institute, the Bill and Melinda Gates Foundation, the Royal Society in the UK, and the University of Leicester, which has been a great resource for me as I've moved here. And just to let you know that we are the Leicester TB Research Group, and if you'd like to talk to us, we're very happy to communicate with you. So I'll finish there.
So thank you very much for a very nice talk, and it's open for question answers.
And can you see me?
Yes. Yes.
Can I ask one?
Yes.
Yeah. So do you have any ideas basically how actually these cells are able to penetrate in the granuloma and then be in contact with the macrophages? So some chemokine receptors, they are expressing.
Yes. Yes, and so there's quite a lot of work with that. Shabana Kadir took that work with her and went and pushed that forward. She's been looking at B-cell follicles and the role of CXCL13 and CXCL 9, 10, and 11. And so the CXCR3 and CXCR5 seem to drive that accumulation. And it's critical. And it's not just getting out of the vasculature and into the parenchyma, it's also then moving through the parenchyma into the granuloma to site. And so when in that data set that we were looking at from the early studies, the PD-1 cells seem to accumulate alongside the B cells, whereas the KLRG1 cells, when they do get in, seem to go into the macrophage sites. So it's an interesting question still. And I think there's quite a lot of work now going on in where do T cells locate in the lesions, and it's a critical function.
Yeah. So the other question was on the peptide mutants that you basically the peptide which are recognizing TCR with high affinity. So in the mycobacterium as such, in the natural infection, do you also see emergence of those mutants, or basically these are the synthetic, like the altered peptide ligands?
Yes, these are altered peptide ligands that we're generating in order to generate T cells that when they come in and they see the full length signal, they'll get the full length signal, but they'll have been driven differently at the beginning. So the idea is that we're not thinking about the altered ligand being expressed during infection, but it's the fact that the T cell will experience a different interaction once it sees that response. And I think there's other work. There's two other. The Anderson group were looking at epitopes of ESAT6 which were less strong, and they see the similar outcome. And again, the antigen 85 work and comparing to ESAT6 also shows the same thing. So it seems to be a broadly applicable idea.
Thank you.
So there is a question from Parul Bhatt that is there any IL-27 receptor inhibitor? Did you see the same impact as knockout of IL-27 receptor? If yes, can it be used as in therapy?
Yes, that's a good thought. And in fact, I'm not sure if I was working with a drug company with that, and we didn't take it further. But it is a thought. But whether you would be able to interfere at the site of the lesion is another question. It is. I don't think it's been pursued in TB per se.
So what about these peptides? Do you think they can be taken ahead as vaccines?
Well, no, because so what we're doing here is a proof of concept. In that driving the T cells themselves. So the problem is then how would you do that in a human population and broadly? What are the peptides that would drive that response? So I think what we need to think about is perhaps driving the T cells themselves less strongly. So if we can define, the work that we're doing currently at the moment in Leicester is trying to define what it is. What is the critical element in terms of the driving? Is it, if it's not the TCR ligation, can we do it by adjuvant flexing? So what is the feature of the T cell during that vaccine profile that then links into its ability to persist and maintain that long-term protection? And then we can say, if we then develop vaccines that are delivered and we've got a correlate of the likelihood of that T cell being protective long-term. And those are big questions that we're still trying to answer.
Have you seen long-term protection? If long-term, then how long?
Well, that was the day 90. That was the. Oh, you mean in terms of long-term between vaccination and challenge?
Right.
Yeah, we haven't flexed that part of the experimental model yet because we're really focusing on cells that function long-term after infection rather than long-term after vaccination. So we've only gone out to 30 days. Well, sometimes we go 30, 60 days. But we don't take it further than that.
So suppose if this peptide works and you would like to take it ahead as a vaccine, which adjuvant would you think will be quite suitable for human use?
Yeah, so I don't know, right? So we've been actually looking at just using alum vaccine in animal models. And of course, you don't get a real change in bacterial burden, but you do get a change in the health of the animal when they get infected. So that may then, in turn, alter the ability of the animal to be protected, or it may alter the ability of the host to mediate an effective immune response. If they're feeling well, they're less likely to have stress-induced inhibition of their immune response. And that's a lot of hand waving because I'm really very good at sticking with the T cells in vivo with a very defined model. I'm hoping others will translate the information that we get into an effector pathway.
Have you seen that whether these peptides, they show permissive binding or are they promiscuous in nature? They work irrespective of HLA diversity in human?
I don't know. I haven't looked in human. We've chose those peptides simply based on their ability to bind to the B6 IAB molecule. That's where we've looked at their binding capacity. And we know there's no other binding presenting molecule in the B6 mice. So it's really very much a tool to ask the question, what does the T cell look like that's able to function later on, rather than a tool to say this is how we would vaccinate. So we're looking for then, what is that T cell, what is the phenotype of that T cell and how does it function? There's another question from Dr. Bashir.
Yeah.
Where he's asking for, apart from a protective role in TB, does IL-27 receptor deficiency affect STAT1 mediated Th1 polarization against other pathogens? So absence of IL-27 receptor deficiency is very interesting. Different pathogens have different outcomes. So if you don't have that regulation of Th1 responsiveness in toxoplasma infections, for instance, you get a very different outcome. So it's very much a regulator of type 1 immunopathology, which is critical. And then he's asking about the CD153 T cells, and I haven't actually looked at CD153 T cells in this model. So I just have to own up to that fact.
Okay, so if there aren't any more questions, we would like to thank Dr. Andrea Cooper for very nice talk, a wonderful talk. So let us give her a big applause. Thank you very much, Dr. Andrea Cooper.
Thank you very much. It's been a fantastic meeting. I'm looking forward to the rest of the talks. Thank you, Javad. See you soon, hopefully.
Yeah, once this lockdown is over, so we'll invite you over here.
That'll be fantastic.
Right.
All right. I will disappear.
So we'll now invite our next speaker, another very interesting talk. Our next speaker is Dr. Ellen Foxman. And she studied her medicine and immunology at Stanford, Harvard, and Yale. And currently she's a faculty, she's assistant professor at Yale University, the Laboratory of Medicine and Immunology. And her laboratory focuses on understanding the natural mechanisms that protect the airway from respiratory viruses and how these mechanisms can be leveraged to improve diagnostics and therapeutics. And today she'll be talking about host response-based diagnostics for respiratory virus infection and application to COVID-19. So it'll be really very interesting talk. So we welcome you, Dr. Ellen.
Thank you. Thank you. It's a pleasure to participate. I really appreciate the invitation. I'm going to share my screen. So let's see. Just let me do this here. All right, good. I hope you can you see the presentation?
Yeah.
Okay, great. All right. Well, thank you again for having me. It's really a pleasure to participate in an international community. I guess one advantage of COVID-19 is at least we're all discovering new ways to communicate, and that it's not as doesn't have to be barriers of international flights and everything to be able to participate and share information this way. So thank you very much to the organizers for inviting me. Today I'm going to talk about a pretty early stage project. SARS-CoV-2, the virus that causes COVID-19, came to our region in the northeastern United States around March of 2019. And most of the data I'm going to show today is actually since March. So it's very new, and it's a continually evolving story. But it also plays off of some work we've been doing for the past couple of years. So this is a bit more, this is a bit of a translational talk. Our lab also studies the basic science of innate immune recognition of viruses in the airway as well. So today I'm going to talk about using the host response as a way of detecting the presence of a pathogen, and in this case, specifically applications to COVID-19. Just by way of disclosure, I'm an inventor on 2 patents related to biomarkers. So today I'll talk about studying early responses to viral infections and SARS-CoV-2 in particular. The basic diagnostic test that we use here at Yale, the concept of host response-based testing in general and how it applies to pandemics and how we can leverage this, as well as our future directions. So actually, when preparing this talk, I looked back at a talk that I gave only in November of last year, and this is my intro slide at that time, which is, I have some updates for this now. So in the US alone, acute respiratory viral infections are extremely common, as they are around the world, and we have more than one infection per person. Now, of course, every year, but that includes anything from common cold to serious illness. But serious illness is quite a big impact even before this year, with about 2 million hospitalizations every year. And of course, even back in November, it seems that we were worried about emerging infections, because it seems that every few years we have been getting something. And this is actually the original SARS coronavirus in 2003 that's depicted here. But of course, these images mean a lot different things to us now that we have a global pandemic of a second coronavirus. Of course, now the impact is being felt everywhere in terms of we have a new emerging virus. We've already had over 7 million cases around the world, and we're still in the midst of a lot of challenges. Different countries, different regions are at different stages of the pandemic, and it's rapidly evolving. What do we need to do in order to contain this, and of course to prevent future occurrences like this? So my lab, this is just a screenshot from our lab website. My lab in general studies this tissue depicted by here, which is the respiratory mucosa. These are epithelial cells that form the lining of the airway. We always think of cells of the immune system, leukocytes, as causing cytokine responses and immune responses to contain pathogens. But actually, the very first stage of this process is often mediated by the stromal cells of a tissue. In this case, the respiratory epithelium gets infected first by a virus. That's the main site of viral replication. And so actually, like all cells in the body, these cells have innate immune sensors within them intracellularly to detect the presence of viral nucleic acid and initiate very robust innate immune responses. Huge amounts of cytokine and chemokine production come from these cells. So we're very interested in general in how setting up the initial inflammatory response in one way versus another way sort of biases the outcome and changes the course of disease. You can imagine if you have a very rapid innate immune response in the first few infected cells, you might completely stop viral replication and end up with a mild or asymptomatic infection instead of letting the disease progress to the lung. And in general, we think of the upper respiratory tract as a gatekeeper. So there's a lot of pathogens that enter our upper respiratory tract that never get to our lungs. And this includes a lot of viruses that come and go from the nasal mucosa without ever causing serious illness, as well as even bacteria that may colonize the upper respiratory tract and be completely asymptomatic. But if they cause it, they can also cause a bacterial pneumonia in certain situations. So in general, we're interested in genetic and environmental factors that can tip the balance of how well this gatekeeper function works. And we're very aware for this current virus, SARS-CoV-2, that it's estimated at least 80% or so of infections are mild or asymptomatic, but some people can get very, very ill. And yet we don't know exactly why that is. And so probably each step of the antiviral defense process contributes to that. And we're looking at the very early stage, this gatekeeping function. So how can you study this? Well, one way we study it in the lab, which I'm not going to talk about today, is using organoid models of the airway mucosa and just infecting cells and looking how the different innate immune responses unfold. But in this case, what I'm going to talk about today is actually on the respiratory swab, that same nasopharyngeal swab that we use to actually detect the virus and look for virus in infected people. You can also find host mRNAs and proteins that are representative of whatever the innate immune system is doing right then. And so we're interested in this very early response stage of the disease before it's progressed to the lung, and also much later in disease you get this hyperinflammation phase. But we're really looking up here and asking what's the best strategies for diagnosis And can we change the course of illness at an early stage? So today I'm going to focus on diagnosis. We don't really have the answer to number 2 yet, although we're working on that. So I'm going to give a brief overview. I know different places, different regions are at different stages of this pandemic and have sort of done different things in terms of how people are pursuing diagnostics for the virus. So I can just tell you what we are doing here at Yale, which is representative of our region. So the basic diagnostic tests that are available for patients right now are twofold. The first one asks the question, does the patient have the infection right now? And that's basically a PCR test for the viral genome. There's other ways of testing for the presence of a virus that are developed for other viruses, but for this virus right now, this is the best way, which is basically looking at specific regions of the viral genome to see if they're present in the laboratory swab. And this is a highly specific test. It may or may not be 100% sensitive in that it depends on the sample collection as well as the phase of disease, what the level is going to be in the upper respiratory tract, but it's very specific. The other question, of course, is did the patient have infection? Is there testing to see does that patient have viral specific antibodies in their blood? And we're actually not using that for any in most cases, this is our go-to test, the PCR test. The serological test, it's unclear what the meaning of it is in terms of actions you can take and recommendations you can make to the patient. So we are mostly focusing on PCR as our screening test to ask, is this person infectious? Can they work as a healthcare worker? Can they, go back to a facility that they were from after they've recovered and things like that? So, but we still have more challenges, right? We have more challenges facing us, as I mentioned, which is we need to be able to screen more broadly. This is showing a temperature screen at the airport, maybe not the most sensitive or specific test for this virus. We need to know. One thing the PCR test does not seem to do is distinguish between PCR positivity and someone who's actually infectious. So it looks like from a study in South Korea that some people who've recovered can have a positive PCR test for a long time, but they don't actually spread the virus to their contacts. So we don't really have a test yet to distinguish that situation. And then finally, how do we find these potentially pandemic viruses before they find us? How do we look ahead and see what we can detect in our patient population that's causing disease that might be under our radar so we can be more prepared the next time? So now I'm going to talk in general about the concept of host response-based testing. I mean, we're all familiar with the idea that when you get an infection, the body has responses, and like a fever is a very basic one. So we know that fever is a sign of infection. That's a host response to the infection that we just use as an indicator that somebody has an infection. But of course, fever is not very specific. There's other things that can cause fever besides an infection. There's a lot of blood tests, the white blood cell count, all these things that can indicate an infection. And in the last several years, there's been much more technology available to look at the whole transcriptome, the whole proteome, specific cytokines and things like that to get much more granular about what the host response is than just measuring the body temperature. Those studies indicate that it's possible to sort of query the body's own diagnosis about what's going on. And this is just a small example here of if someone's coughing, that could be for any reason. It could be something they just ate. It could be an allergy. It could be a viral infection. It could be another type of infection. But if you can look at the pattern of molecules in the blood or even in the nasopharynx, as we have done, you can get a lot more detail about the type of infection the body thinks it's fighting by looking at the innate immune response. Because the patterns recognized by the innate immune system are different depending on the pathogen class. If it's a virus, that's one type of pattern recognition pathway we're going down. If it's a bacteria, if it's a fungal infection, other types of infection, we get different pathways. So this is just I know we have a lot of immunologists here, but not everybody thinks of epithelial cells as immune cells. So I just thought I would briefly give an intro slide about this concept, which is that our airway, our conducting airways are lined. Actually, our entire respiratory tree is lined with epithelial cells from the nasal passages to the distal lung. And like all cells of the body, these cells have innate immune sensors and can detect when they're infected by a virus. And that, so the viral enters the cytoplasm, starts to replicate. And this is actually a picture from Field's virology showing replication of a single-stranded RNA virus and how that generates double-stranded RNA in the cell cytoplasm during viral replication. And this double-stranded RNA is a pattern that's recognized by innate immune sensors like RIG-I and MDA5 that will then lead to a signaling cascade leading to the secretion of interferons and induction of about at least 300 different interferon-stimulated genes that have different infections that can either directly block viral replication or recruit cells of the immune system to the site to fight the viral infection. And so a couple of years ago, we wanted to ask, well, are there any innate immune molecules like interferon-stimulated genes that are really highly induced within nasal epithelial cells when a virus is replicating there? And can we use some of those very highly induced genes or proteins as biomarkers of viral infection? And so a simple experiment was to just take the primary nasal epithelial cells in vitro, transfect them with a small molecule ligand made by one of our collaborators, Anna Pyle at Yale, and see if this, what type of innate immune response this triggers off in the nasal epithelial cells, and which genes and proteins are most highly induced. And this shows RNA-seq of mock versus transfected cells. It's a volcano plot, but the take-home message here is just that there are some very highly expressed genes and proteins that are turned on by something that mimics viral RNA in general in the nasal epithelial cell cytoplasm. And almost all respiratory viruses are RNA viruses. So we thought if we could focus on these, we might be able to find pan-viral biomarkers of viral infection. And in fact, that did seem to bear out back in 2018 when we did this study. So basically, this is just showing one piece of a figure from the paper, but it's published, so you can look at it more if you're interested. But one molecule we looked at is CXCL10, which we just heard about in the last talk as well. This is a chemokine that recruits T cells to a site of infection. And we found that if you find this in the nasopharynx, of course you can find it anywhere in the body in response to any number of insults. But if you find it in the nasopharynx, it's a pretty good indicator of viral infection in the nasopharynx. And this shows mRNA. This is also true of protein. Basically, this is shows huge induction, 10 to the 5th fold induction of CXCL10 in the presence of a viral infection. These horizontal, this indicates the CXCL10 level. This indicates the presence of a virus this is all in patient samples. And what was also really cool is that we did 2 different studies at 2 different times of year when different viruses were circulating. And basically, this type of biomarker strategy seemed to pick up any virus that our patients had, even adenovirus, which is actually a DNA virus. So DNA viruses also trigger the interferon response via a different pathway. So the question is, so we've been studying this for a while in different ways and how we can use it. And when COVID-19 came along, our questions were, does the same pan-viral biomarker detect COVID-19? Because each virus may be different, may be differentially able to inhibit innate immune responses. So we want to know, first of all, does it pick up this virus? We know it picks up seasonal coronaviruses. And how can we use this to help us learn more about the virus and even fight the pandemic? So this is actually Reddy Chumarla, a postdoc in my lab, who's from India actually, and he has been the spearhead person on the data I'm about to show you. And I also want to acknowledge we have a whole working group at Yale who helped get the PCR tests up and running very soon, extremely fast after our FDA allowed it. So I'll talk more about them at the end. But basically, this is the situation in our region of the northeastern U.S. So green is COVID-19 cases. The first case in the U.S. was on the West Coast in January. But then in New York, which is very, very close to Yale, and some of our patients are from New York, the first case was on March 2nd. And then in Connecticut, our state, the first case was on March 6th. And because the FDA only allowed us to set up our own test at Yale they allowed that starting February 29th, our testing, actually, we got it up and running by the 13th of March, thanks to Marie Landry, shown here, the director of our virology lab. But we still hadn't been testing these first couple weeks of March. So we thought, can we use this biomarker approach to help us quickly screen to see if we missed any cases? And so basically we use CXCL10 in this case. We could have used several different proteins, as you'll see if you look at our paper, but in this case, we use CXCL10 to screen all the patients during that time window who had been symptomatic, and they had been tested for our respiratory viruses, other viruses on the panel that our hospital has, which includes about 15 viruses, but they had tested negative. So these were people who their doctor thought they might have an infection, but they tested negative for the viruses we were testing for. And we thought those people if they have a viral infection biomarker, they're pretty good candidates for people who might have had SARS-CoV-2. So out of the 376 people in that category, about 8% of them or so were positive on the CXCL10 ELISA for seeming to have an active viral infection. And then we tested these people by PCR, and it turns out that 4 of these 33 people actually did have SARS-CoV-2, and one was very surprising. It was an infant. So, because it was a research study, we actually tested all these 343 people as well, and they were all negative. So this was pretty interesting, and luckily at Yale, there's a lot of interesting people with different areas of expertise to collaborate with. So we wanted to learn more about these cases, and our collaborators in the Gruba lab, who also work with this website NextStream, which I encourage you to check out if you haven't seen it before, were able to map those 4 cases on a phylogenetic tree of all the cases reported so far. And interestingly, first of all, when they sequenced them, they found that they were all different. Even though these 4 cases all occurred within a few days in our region, none of them were exactly the same as the other, which means they were 4 independent introductions. Another thing that was pretty interesting, and 3 of them were related to North American strains. 1 was related to a strain from Western Europe. So that was interesting, and it kind of went along with a study that had previously been published by the Grubal lab, showing that a lot of our early cases in our region were from domestic transmission rather than international. So that study's already published here. So that was pretty interesting to find these cases and then to get some insight into where this virus was coming from into our region. It also gave us the idea, looking at this graph here, that maybe, we are thinking this was 376 people that their doctors suspected they had a viral infection. If we had one issue right now is testing capacity and expanding testing capacity to include more people. But the PCR test is pretty complicated, requires 3 PCRs, RNA extraction, a lot of more complex procedures than an ELISA or immunoassay, which is pretty straightforward. And what we realized in here is, look, if we had only tested these 8% of people with the PCR test, we still would have found all the cases. And potentially it suggests a way you could conserve PCR tests, because you wouldn't have to waste PCR testing basically on people with a very low likelihood of having the infection, active infection at that time. And so that is basically something that we wanted to further explore, because that really might help places around the world as they're trying to leverage and expand their existing testing capacity to test more people, to use this kind of approach as a pre-screen. And so so far we've just done one other day where we're working on getting a few more, increasing the N here so that we can have better data. But this was a different day in March, a single day in early March, about a week after what I just talked about, where we had 144 people that were tested with the PCR test for SARS-CoV-2. We screened them with the biomarker, and in this case, 35 of them were positive. And if we ask who had COVID-19, there were 17 people on that day, 16 of which were positive for the biomarker and one of whom was negative. And so this calculates out to be a negative predictive value, about 99%. And this one case, we really wanted to see, why is that one case not biomarker positive? We really wanted to understand that better. And it turns out this one case had a very low viral load. So we then wanted to look a little bit more about biological variables and how they affect the innate immune response in the upper respiratory tract. And so we looked at 59 patients so far. We hope to really increase this number by a lot. And what we saw is that most of the people tested were in the older age groups, as you might expect, and most of the people in the older age groups were more ill than the younger people that happened to be in our cohort. Like most of them were admitted to the hospital, for example. And if you look at these people, the CXCL10 level in the nasopharynx does track with the viral load, as you would expect, because viral replication is the trigger for CXCL10 production. Interestingly, we found an inverse correlation between age and innate immunity, and now we're doing a study with Tom Murray, another collaborator at Yale, to see whether that is, if you look at a lot more patients of diverse ages, is that true? Is there a more robust innate immune response in the upper respiratory tract of younger people, which would be very interesting if true, but there's other explanations too. We did not see a correlation between age and viral load. That would be one other explanation. There's a correlation between age and viral load, but we did not see that in this sample set. So finally, in the last couple minutes, I just want to talk about an additional project we have related to the same theme. So some of the issues I talked about before can be potentially addressed with biomarker-based pre-screening, but we really need to know a few things first, like what's the expression of biomarker over time during illness? Are these biomarker low people? Are they actually infectious or not? And things like that. And then how can we use this approach also to look for novel viral infections? And that's something we actually had started back in 2017. So this is a student, public health student who was in my lab, Amelia Hanron. She actually graduated already, but she was working on using the same approach I just talked about to screen people who might have unexpected viral infections by looking at, this was from 1 week in January, we did the same thing we just described, where we looked at people who were suspected to have a viral infection. They tested negative on our panel, and then we screened them for CXCL10, and we found a percentage of them actually had a high level of CXCL10 in their nasopharynx. And interestingly, back then we were not screening for seasonal coronaviruses on our panel. But when Amelia did this, she found that about half of these were just seasonal coronaviruses. So we were missing seasonal coronaviruses in our patients. And as a result, we've now added those 4 seasonal coronaviruses to our panel. But as a proof of concept, this also shows that this method was good at picking up undiagnosed viral infections. So now we're also doing that to, we also have half these samples where we don't know the virus, but we're sort of viral immunologists. We do viral culture, we do EM, we do all the metagenomics and all the techniques you would use for viral discovery. So we're now doing that on these other samples to see what viruses might there be that are circulating in our patients unbeknownst to us. And this is sort of a general strategy that we want to use to look for undiagnosed infections. And we would be happy to talk to anyone who might be listening who's interested in international collaboration with this project, because of course, different viruses are going to be circulating and different pathogens are going to be circulating in different regions of the world. So finally, just to sum up, we, in the Foxman lab, in general, we study host-virus interactions in the respiratory tract. We also study airway repair post-infection, but I didn't really talk about that today. But the specific way this could help us fight COVID-19 is to understand early innate immune responses and how they alter the outcome of disease, as well as biomarker-based approaches to simplify and improve testing algorithms. And I talked about some of the applications for potentially pre-screening people to leverage existing PCR testing capacity and direct it towards people most likely to be infected. And finally, I talked about our undiagnosed viruses project, which has been going on for a while, and as a sort of response to try to find the next pandemic before it finds us. So, of course, most important slide, I want to acknowledge all the many people who help with this project. These are my lab members listed here. I mentioned Reddy Chimarla, who's been sort of the point person, been side by side with me every day since this pandemic began, testing these samples and trying to make a difference here, as well as Amelia Hanran, who did his initial work on the Undiagnosed Viruses Project, and other lab members who've also been very supportive and helpful. I want to acknowledge Marie Landry and our clinical lab for their collaboration, our bioinformaticist, Dejian Zhao, the Gruba lab, who I mentioned helped with the metagenomics, as well as the lab working group, which includes Saad Omer, Nate Gruba, Albert Ko, Akiko Iwasaki, Marie Landry, and myself. And our first meeting was actually on March 2nd, at which time there's only 45,000 global cases. So things have really evolved, and we really hope that we can get ahead of this virus and engage in the rapid pace of scientific discovery that's going on to help us all combat this virus. So thank you very much for your attention, and I would be happy to take any questions at this time.
So it was really a very educative talk, and with the data, and you see the enthusiasm, dedication of a person like Alan Foxman, it appears that soon the days are not far away when we can find some strategy to control COVID-19 infection. So now this is open for question and answer. So I've got a question for you, that what do you think about the nasal immunity in case of COVID-19?
We're very interested in that. I mean, that's a little bit even more in the line of our usual research, as this is a little bit more translational than what our lab usually does. But it's very clear that that early innate immune response, just modulating it a tiny bit, can have a huge impact on the outcome of infection. And in fact, people who are genetically deficient in RIG-I or MDA5, those receptors for sensing intracellular viral RNA in the epithelial cells, those individuals are highly susceptible to serious respiratory infection as children. So, and furthermore, certain environmental factors alter that innate immune recognition step and can really be risk factors for serious illness. So that's something we're very interested in, to look at how that early innate immune recognition and the pattern, this talk was just showing one molecule, CXCL10, but there's a huge pattern of molecules that become expressed during viral infection, and it's not the same for everybody. It depends on your genetics. It depends on environmental factors at the time of infection. And that can really influence whether that virus gets cleared asymptomatically, causes a moderate infection, or goes down the lung and leads to that whole cascade of events leading to even mortality, So we're very interested in understanding that process. We think of it like a marble at the top of a mountain. If you nudge that marble, this is the very first step where the virus starts to replicate. If you nudge that marble in one direction, the marble rolls down the mountain in one way. If you nudge it in a different direction, the marble rolls down a different way, and you get a totally different outcome. So that's what we're also doing right now. We weren't able to get the data quite as fast because it's a little more complicated looking at the whole transcriptome instead of one biomarker, but trying to understand what's the initial response in people who then go on to have different outcomes. And something we're very intrigued by is the difference between kids and adults, because children seem to be relatively protected from lung manifestations of the disease. I mean, there are some children who get serious disease, but it's very rare. And so that's one thing we're doing now is looking at these early stage responses in children compared to older adults. And that's a group that has very different outcomes. And we're hoping that will provide some insight into why that is. I mean, there are also some older adults who have mild or asymptomatic infection as well. And so that's another way to get at it. And so I really think that we will find some differences.
Yeah.
Can I ask a question? Can I ask?
Yes, sure.
Yeah, of course, yeah.
Okay, sure. So what do you think is the source of this CXCL10? So what is this?
Well, we know that the respiratory epithelial cells, we grow these respiratory epithelial cells, the primary human airway cells in our lab and infect them. And they produce a ton of CXCL10. They really do produce quite a bit when in the early stages of viral replication. So certainly that is one source. I would be extremely surprised if that is not a major source. But as well, once you get leukocyte infiltration, that could be a second source as well. But I can tell you, even in our in vitro experiment, to infect with a virus, we studied, even before this COVID-19 phenomena epidemic, we study rhinovirus, influenza, those sorts of viruses. If you infect and then you just look in the supernatant or the basolateral media of these cultures, you'll find a huge amount of chemokines being produced by the epithelium to attract cells to the area. Mm-hmm.
So is it the direct virus recognition or basically the product, basically interferon produced by these epithelial cells, and then they basically recognize the receptor, the interferon receptor? And
yeah, it's both. So the pattern recognition receptor, MDA5 or RIG-I, turns on NF-kappa B and IRF transcription factors. Actually, you can find binding sites for both those transcription factors in the promoter of CXCL10. So it directly turns those on, and the interferon secretion and interferon binding to its receptor and that signaling is a positive feedback mechanism. But even within 30 minutes of exposing these cells to a ligand for these pattern recognition receptors, you can detect CXCL10 in the media. So it's not, you don't need that. The positive feedback surely amplifies it. But even that initial signaling pathway turns these responses on. And if you look at the transcription factors downstream of interferon receptor and downstream of these pattern recognition receptors, they have almost the same, they're almost the same, right? They're IRFs, they have almost the same binding site in the DNA. So it's really, the interferon's more of an amplification, positive feedback part of it, but it starts before that. So. In fact, we find much higher levels of these cytokines than we do of interferon itself, the various interferons.
So there is a storm of questions for you. I don't know whether we can handle them. There's a question from Sunil Arora. What factors govern to decide whether a patient will be infecting other individual? Is it viral load or something other?
We are very interested in that. We want to test these samples. What would be great is, I'm certain viral load is a good proxy for it. I mean, if somebody has a very high viral load, and especially if they're symptomatic and coughing and sneezing, just common sense says, yeah, that person is probably more infectious than someone with a barely detectable viral load who's completely asymptomatic. You know, that's just sort of an intuition as being in this field for a long time. But what we would really like to do is see, can we actually get a biomarker of infectivity? Because people, one thing we're having in our hospital is people just staying a very long time because we don't want them to go back to a nursing home, let's say, or a long-term care facility or a rehab facility when they're infectious, right? Like that would be very bad. So people have been being retested and retested and retested. And if even they have a barely detectable viral load, they're still not going to that rehab facility. But it would be very nice to have a better test to be able to distinguish that if these people truly aren't infectious. Because there was a study in South Korea where they had followed a lot of people who were, they call them re-positives. They recovered, and then 6 weeks later, they're still virus positive. And they followed they followed about 250 people and 790 of their close contacts. And none of those contacts got sick from those people, which suggests that they actually were not infectious. And so that's something that's very important to find a better way, just in terms of managing the pandemic. We need to distinguish those states, infectious and non-infectious.
So there's a question that is an interesting idea using innate immune-based biomarker for detection of virus. But I have seen lots of variability in data because these various factors like ethnicity, gender, and age, it can make impact.
Absolutely. But I will say in our 2018 study, we did not filter anyone out. We had infant to 90 years old, 100-year-old patient. We had immunosuppressed cancer patient and healthy people who had a cold, anybody who came to our hospital who got the virology panel, and everybody had an innate immune response in their nasopharynx. Almost everybody. You can look at the data in the paper. Maybe looking at one biomarker isn't enough, like just CXCL10, but if we looked at 3 interferon-stimulated gene, a fingerprint of 3 interferon-stimulated genes that we found in our in vitro studies, the accuracy was 97%. No matter if you're talking about an infant, no matter if you're talking about someone immunosuppressed, no matter. So I believe it's absolutely true. I'm sure gender, age, ethnicity, state of health, things you recently breathed in, all of those things impact innate immune response. But there are certain innate immune responses to viral infection that are absent in health that go up many orders of magnitude in disease. So there is a big signal to noise ratio there, at least for symptomatic patients. So when you're getting into more subtle things like pre-symptomatic, post-symptomatic, subtle differences in levels, I am sure you will find differences, like we even found for age. But when you're just talking about, is this thing below a threshold or above a threshold, something that goes up 10 to the 5th fold in disease and is absent in health, I think you can have some universal biomarkers.
The question from Anjali Patil: why in some cases corona become asymptomatic and directly leads to death? And why high rate of corona infection and increased mortality observed in people with diabetes?
Oh, that's 2 different questions. The diabetes one, I don't know the answer. I mean, I guess in a general sense, comorbidities or other disease states that make the health state more fragile, you would expect people to have worse outcomes. However, not all comorbidities are equal for this disease. Some don't seem to matter that much, and some seem to make a big difference, like even body mass index, or being like having a very high body mass index and being overweight seems to be a big risk factor, for example, whereas it wouldn't be a risk factor for other infections. And then so I don't know the answer yet about diabetes. And what was the first part of that question? I can't recall.
Okay,
why is it sometimes asymptomatic and sometimes leads to mortality? That's another one. It's still, I mean, that's getting pieced together. It's getting pieced together by people looking at immune response in the blood, immune response in the lung. There's not as much known about immune response in the upper respiratory tract. We're hoping to help fill that gap with some of our studies, but it's a relatively new virus and we don't know the answer yet.
So there's a question from Lena Sapra. What are the underlying mechanisms that are responsible for lymphopenia in COVID-19 patients?
Yeah, that's very interesting. So that's a later stage of the disease. So I showed that figure with the upper respiratory, the lung, and then the hyperinflammatory state in the third phase of people who get seriously ill. And it does seem, it is true that there's this lymphopenia and destruction of lymphocytes. I guess I'm not an expert on what the mechanism of that is. That direct killing or a cytokine mediated response, the reason why you see that lymphopenia, but that does seem to be holding true in different regions and different studies. Oh, I can't hear right now. I cannot hear you.
Sorry.
Oh yeah, there we go.
The question is, Gurpreet Kaur, she asks, with regards to translational application, what is your take on type 1 interferons in treatment of COVID-19 patients?
Well, that's a very interesting question. I mean, I get because there has been some data suggesting that, you know, as I showed those three phases of the disease, in that later phase where you have this sort of hyperactivation of the inflammatory response, it's very damaging to the lungs. And there's some suggestion that interferon can fuel that stage of the disease. So my thought is it probably matters a tremendous amount when the interferon therapy is initiated. And like before or during that first phase, you could imagine it being beneficial, especially a local, you know, like a airway type of stimulating the interferon response. But towards the later stages, you could imagine it going either way. And I think a lot of the studies are showing that, like it's not very clear cut that it's beneficial. Interferon. Obviously, a lot of the people who've been treated so far are people who already have pretty serious disease, not asymptomatic people who are just getting the disease. That's much harder to find those people, you know, so.
Well, another question is from Sanpreet, saying the question is, does the expression of S and N proteins decline upon the recovery of patients?
So that's looking at the viral proteins rather than the PCR. So in general, in diagnostic clinical pathology, viral antigen tests are a lot less sensitive than PCR. I mean, probably most of the people on this call have performed PCR and know that it's, even if you have just a few molecules of your target, you can amplify it so much, like 2 to the 40th in your 40 cycles of PCR, that it's extremely sensitive. Whereas viral antigen tests it's harder to make them as sensitive and specific. So I don't think that there are any approved, at least not in this country, like a FDA or EUA approved viral antigen tests at the moment. But so we're not in generally measuring the S and N proteins, but you would imagine that would be, it could be a good correlate of the presence of like the whole virion, the whole viral particle, rather than just viral RNA. But in general, because these types of tests are generally so much less sensitive than PCR, I don't know if that's going to have a good negative predictive value. You know, maybe positive predictive value if you find that you have the virus, but if you don't find that, what does that mean? That's the question.
So the question is there any biomarker to distinguish between asymptomatic and symptomatic patients?
Well, you don't need a biomarker for that, right? Then if you're asymptomatic, you're asymptomatic, right? But I guess it would be more interesting to distinguish asymptomatic subjects from healthy subjects that don't have the virus that are also asymptomatic. That would be very useful. But in terms of, I see there's also a question in the chat about susceptibility. Like, is there a biomarker to determine whether you're susceptible to getting very ill? And actually, I just read a paper about, it might have been a preprint about a few genetic factors that have already been found that seem to be predictors of susceptibility. And I'm sure we'll be learning more about that. One of the great things about this conference that you're hosting, and the great thing about this pandemic, is it's really rapidly increased the rate of data sharing and use of preprints. So the amount of data coming out is just incredible. Of course, preprints haven't been peer reviewed, so that's the flip side of it. But even we put up a preprint already. If you want to read about this talk I just gave, we already put up a preprint about it, just because if it can help people during this pandemic, getting that information out there. So I did read a paper. It might have been a preprint about genetic susceptibility, and you can probably search for that on medRxiv.
So, Alan, we follow your papers, and I would like to ask a question in connection with your paper, which you published in 2015 in PNAS, which is temperature dependent defense against common cold virus, and you showed that it limits the replication at warm temperature. So, what about COVID-19?
Well, there's a preprint about that, not from our group. Someone else looked at that. So what we found, that's an example of the marble rolling down the hill. So what we found, just by doing the experiment at different temperatures and looking at viral replication and innate immunity, what we found is for rhinovirus, the common cold virus, the interferon response and the signaling downstream of the intracellular pattern recognition receptors I talked about, MDA5 and RIG-I, is very suppressed by slightly cooler airway epithelial temperature. And that has been shown for other viruses now that cause arthritis and other peripheral diseases, that the triggering of the interferon response is much lower, several orders of magnitude at the lower temperature. And there is a preprint on either medRxiv or bioRxiv that looked at temperature dependence of the interferon response to COVID-19. And it seemed from the preprint that the interferon response was more robust at 37 degrees compared to 33 degrees Celsius, similar to what we found for common cold virus.
Okay, so any other question? There are many questions, overlapping questions. Many of them you have covered in your talk. So I would like to now thank Dr. Ellen Foxman for a very nice talk, very stimulating talk. So let us give a big applause to her. A wonderful talk.
Thank you very much for having me. It's a very interesting conference.
We will come up with some biomarker quite specific for SARS-CoV-2. So thank you very much, Alan, for giving this wonderful talk. Okay, so our next speaker is Dr. Mangala. And Dr. Mangalam, he's trying to enter. Hello, Dr. Mangalam. So next talk is by Dr. Ashutosh Mangalam. Dr. Ashutosh Mangalam, he did his PhD from Sanjay Gandhi Postgraduate Institute of Medical Sciences from India. And then he moved to the United States, and currently he is working as assistant professor of pathology at the University of Iowa Healthcare. And his area of interest is immunology and gut microbiome. And he'll be talking about the gut feeling, breaking down the role of gut microbiota in autoimmune diseases. So we are still waiting for the speaker. So the next speaker is finding some difficulty in joining us. So meanwhile, we have to wait for some time for the speaker, or anybody volunteer can give a talk. Andrea, would you like to give another talk? So let us wait for a moment. So these talks have been quite interesting. You know, people talking about COVID-19, and in the morning we heard about Professor Peter Doherty giving wonderful interaction with Dr. Peter Doherty, and he gave us good. He gave us highlights of COVID-19 situation and what is the response and what may be the strategies to control it. And then very good talk by Dr. Dipankar Nandi, and he also gave a very good overview about COVID-19 and role of infection. He talked about in perspective with epidemiological studies. Now, Ellen, she talked about COVID-19 as well, and especially on biomarkers. Appears to be that in study CXCL10 may be quite interesting biomarker. Then there were some good talks on tuberculosis. Dr. Balaji talked about immunological responses and the host factors during microbacterial infections. So he talked about role for distinct epigenetic transactions. Then it was Dr. Pavan Gupta. He talked about orphan receptors. So these receptors are quite important as far as tuberculosis is concerned, and Dr. Pavan Gupta is finding very good correlation with these markers. There were a lot of questions and very interesting questions, and the participants they are asking are really very good. Dr. Pradeep Sen, he talked about very important role of RUNX protein in regulating antimicrobial immunity. Professor Robert Wilkinson, and he talked about immune response to tuberculosis, meningitis, and it was really very interesting to know that how tuber meningitis it can affect the person's immunity and what damage it can do to the person's brain, and what are the factors which are responsible for it. And then Catherine Wilkinson, she talked about antiretroviral treatment and immune response to tuberculosis in HIV, TB infected persons. So overall, it appeared that the talks were quite interesting, and then later on by Dr. Andrew Cooper, he talked about T cells and how T cells then responds into granulocytes, and gave a very good overview about her work to these peptides. So,
Dr. Javed, can I ask something?
Yes, who is this? Can you introduce yourself?
Uma Kanga from New Delhi.
Right.
I just wanted to know if anybody, those who are participants attending this symposium, is anyone doing a work on biomarkers like what Dr. Foxman just presented? Any studies from India? Anybody can share an experience over here?
So actually, a majority of people, they are participating through our website or through YouTube. So I can pose
Okay.
the question that anybody, if anybody working in
What's the Indian experience?
okay, so anybody working on biomarkers for COVID-19, if anybody is working, can let us know. Right.
Thank you.
So still, Dr. Ashutosh Mangalam, he is not succeeding. Somehow there is some problem. Why he is not succeeding in entering? Okay, so so meanwhile, if people are interested, we can talk about the immune system. Okay, can you guys see my presentation? Somehow my presentation is not being viewed. So let us wait for a while for our speaker. I think my team they are trying. We are trying. Just have a patience. The problem is that he is not even logging in. Okay, let us wait for a moment. And I think meanwhile, if you wish, we can talk, since there are many immunologists in this forum, we can talk about some immunology. Right. Especially, let us talk about these T cells. We have been hearing a lot about T cells. These T cells, they have been basis of various markers. A lot of T cells they're getting discovered these days. And we know very well that T cells, they are the most important cells of the body. These T cells, they get birth in bone marrow, and then they mature and get educated in thymus. Very interesting cell. Birth in bone marrow, maturation and education in thymus, but then they migrate out of the thymus, and then they move into blood. And they move into blood as naive T cells. And there, then comes the training phase, which is the primary phase of T cells. And this phase is antigen dependent. And this occurs in lymphoid tissues to become memory and effector cells. So from naive, now they become memory or effector cells. And they are supposed to take care of different pathogens. So in lymph node, the antigen enters through the afferent duct, and then the cells and antibodies to these antigen exit through afferent duct. So it's a beautiful architecture of lymph node. So in case of lymph node, if you see that there's a medulla where the plasma cells they reside, and then there's a cortex where APC and B cells they prefer to live in. And interestingly, in between medulla and cortex, there's another region which is called as paracortex region, where the T cells they reside. So it's a beautiful architecture of a lymph node, and you can find where exactly the cells they are located. And through histopathology, you can stain and see these sites are quite distinct. So if you get injury or you infect at one particular portion in your body, for example, you get a cut and then the bacteria, it enters into your body. Ultimately, different cells, specifically, dendritic cells, they reach to this site of infection. They carry bacteria or antigen, and then they reach to lymph node. And where they go to the site where the T cells, they get settled. And now they prime. So the site of priming is quite different than the site where the T cells, they are supposed to react. And T cells, if you see, this is a wonderful population. T cells we have, you can distinguish T cell on the basis of yes. Yeah. Yes, Dr. Mangalam. What happened? So, is it not possible for you to log in?
No, I think I tried my computer, I tried my phone, I tried a different computer, but it just, I don't know what is happening. It's just not letting me. It's saying that our manager will let you in. And
Okay guys, अरे एक मिनट जरा आशु ये देखना जरा ये मेरा क्यों नहीं आ रहा यार ये
Another cheese, so you can make a possible release.
Oh yeah. This guy, what's with the
Australia? So थोड़ा busy रहता है 20 सेकंड.
हाँ.
हाँ ये सही है.
ये presentation नहीं हो रहा है ये presentation नहीं है ये presentation हो रही है
presentation नहीं है सर.
ये मेरे screen पे presentation है उसमें नहीं हो रहा है.
ये नहीं सर आपका presentation हुआ है screen sharing हो रखी है.
Okay, screen sharing.
Okay,
so right. So can you guys see my presentation?
No, sir.
Can you see my presentation? Radhe, can you see my presentation?
No, sir, we can't see your presentation.
You can't see my presentation?
No, sir.
Can you see now?
No, sir, not yet. Yes, sir. Now it is visible.
Okay, so I thought let us talk about very basic immunology because I see that a lot of students, they are watching us from this. So I was talking about that we have T cells, and if you see T cells, we have alpha beta T cells, we have gamma delta T cells, and if you see these T cells now, they are naive effector and memory cells. So we have been listening about these. So what exactly naive T cells are? This is especially I'm talking about talking to students. The naive T cells are T cells which have never encountered antigen in their life. So these are virgin T cells. And then once they encounter antigen and they become effector cells, so effector cells, then they become memory cells. So majority of the effector cells, they die, and what exactly, how they become memory, there are many theories about it. So there are many markers which people believe that it can differentiate between naive effector and memory cells. But most renowned markers which are considered, these are CD44, CD62L, CD27, and CD127. And in case of human, CD45, RB, RV, RA, and RO. So the cells, when they are alive, they're going to express very low level of CD44. But the effect, once they become effector, they start expressing very high level of CD44. In case of naive, CD62L is high, and but in case of effector cells, this is low. Similarly, if you see CD27, it is low in case of naive cells, but this is highly expressed in cells. So based on phenotypical markers, one can know whether it's a naive cell or it is effector or it is turning into memory cells. Okay, so this is very interesting that once the bacteria is engulfed by macrophage, so ultimately what happens? So there are many ways through which the macrophages, they phagocytose bacteria. So this bacteria, it can be phagocytosed through receptor-mediated phagocytosis can occur. Or it can also phagocytose through various mechanisms. So once the bacteria is phagocytosed, it goes into the endocytic vesicles. So it forms these vesicles. And now it gets fused with lysosomes, which has a lot of enzymes in it. So when there's a fusion, when there's a lysosome-phagosome fusion, the bacteria is degraded and these peptides, bacteria is chopped down into peptides, and these peptides ultimately they get associated with MSC molecules. So once they get associated with MSC molecules, the peptides, they are transported on the surface of the antigen-presenting cell. So once it is presented on the surface of antigen-presenting cell, then the remaining job is done by the T cells. So T cells, what they do, they recognize this MSC complex with peptide. And the first signal is through TCR. The TCR, it binds, and CD4, it also binds to MSC molecule. And once this signal is delivered, it leads to the expression of IL-2 receptor. So once IL-2 receptor is expressed on T cell, then it induces the expression of CD40L. It binds to CD40 on antigen-presenting cell, and then this signal, it leads to the induction and expression of B7 molecules, the CD80-86. And this signal of interaction between CD80-86, CD86 and CD28, it then leads to the secretion of IL-2. IL-2 binds to IL-2 receptor, and then it induces cell division. So IL-2, it works in autocrine fashion on T cells. It's also, earlier it was called as a T cell growth factor. This T cell growth factor, it works on T cells in autocrine fashion. It can also work on paracrine fashion on other T cells, and it induces cell division. So T cells now, they proliferate. Besides, this also leads to secretion of interferon gamma. It acts now on antigen-presenting cell, and these antigen-presenting cells, they get activated because of interferon gamma. So interferon gamma is responsible for induction and expression of MSC class 1, class 2 molecules, and also for induction and secretion of pro-inflammatory cytokines, IL-1, 6, and TNF-alpha, which works on T cells, and this is how the T cells get activated. So I think I've covered almost entire time which we devoted to Dr. Mangalam, and let us see if our next speaker is ready. So our next speaker is Alfred Bothwell. So our previous speaker is ready with us. So welcome, Professor Alfred Bothwell. So we are having a lot of we are running well in advance with time. We are not running short of time. So we welcome you, and if you're ready, we can start your presentation. Professor Bothwell. Good. So we can see your slides. So let me introduce you to Professor Alfred Bothwell. Professor Bothwell, he's working as a professor of immunobiology at Yale School of Medicine, Yale University. And his main area of interest is immunology and molecular oncology. And today, he'll be talking about the tick of 2 promotes tumorigenesis and metastatic progression of colorectal cancer in mice. So we welcome you, Professor Bothwell. Good. We can see your slides, and if you can please make them in a presentation mode. Professor Bothwell, can you make it into a presentation mode? So Dr. Bothwell is also characterizing the development of function of regulatory T cells and the mechanism therein that affect the cancer, autoimmunity, inflammation, transplantation, and recruitment into vascular sites. He is doing extensive research, and his lab is focusing on regulatory T cells mechanism and its effects in various diseases, including inflammatory diseases. But he also works on peroxisome induced proliferation of activatory receptor. Previously, Dr. Bothman, it was okay. Yes. Just make it full screen. Yeah, good. So we welcome you, Professor Bothman, and please, you can start your presentation. बाद में करेंगे Hello, Dr. Ashutosh.
Yeah, no, I think just let me know what time.
Yeah, we can do it at 8:30 our time.
8:30 means 10. Okay.
Right?
Okay.
Okay, fine. ये देखो क्या दिक्कत आ रही है इनका हेल्प होगा डॉक्टर बॉथमैन का लग गया था वो कर दिया था उन्होंने सब कुछ ठीक कर दिया था अब और प्रेजेंटेशन है उनका हाँ जी प्रेजेंटेशन है उनका मैं देखता हूँ सर मैं देखता हूँ अभी यही सॉल्व कर रहा हूँ Professor Bothwell, can you try again, please? Hello. I'm really sorry, guys. Dr. Bothwell is finding some difficulty.
Hello. He's not in here, Ashu sir. Yes, I have emailed him. Let's see what he replies. Okay, Ashu sir, Professor Bothwell has joined. You present from your side. He will speak.
So, Professor, both while you can start presentation, we have loaded from here, from our side, and you have to tell us for next slide, so you can keep on changing. So, welcome you. You please unmute your mic.
Okay,
Yeah.
can you hear me now?
Yeah, thank you very much. Welcome here.
Okay, let's see here. Let me go to the next slide and see what's there. Okay, so I'll have to tell you to turn the slides. I changed the slides a little bit, so we'll just go back to the first slide. Okay, well, okay, I hope this works. Okay, so today I'm going to tell you about the bulk of the presentation will be about Dickoff-2, how it promotes tumorigenesis and metastatic progression of colorectal cancer in mice. This is the exact title of a manuscript that is now under review. But this is a very complex topic, and it's I'm going to preface my remarks by giving you several background slides as to how we got into this. This was a line of investigation that began about a decade ago, and we were our current understanding is so well developed compared to what it was when this picture was taken. This is from the summit of Mont Blanc, and we were very much above the clouds, but it was a very obscure topic. We understood very little of what I'm going to tell you today. So I'm going to give you about maybe 4 minutes or so of some introductory material on how we got into this line of investigation, initially studying intestinal tumorigenesis in the APC-Min mouse. This is a developmental model of intestinal tumorigenesis. We began these studies to study in depth the role of tumorigenesis. At that time, we had no idea that this would lead us into metastasis. So I'm going to give you some introductory comments on the next slide. Next slide, please. I need to explain that this is a presentation about Wnt signaling, and on the left-hand side of the screen, you can see that Wnt signaling actually refers to a group of Wnt agonists, which there are 19 gene products, which are very much involved in differentiation in mammalian cells of almost every cell type. It interacts with a receptor on many different cells. LRP56 is the receptor, and there are many sites for Wnt proteins and frizzles. So this interaction then can result in the activation of the Wnt destruction complex, in which APC is a primary regulator, and it's a negative regulator. But when there's a Wnt agonist, there is then a destruction of this complex and release of beta-catenin into the nucleus, and that's referred to as the canonical pathway. Everything that I'm going to tell you today refers to the non-canonical pathway, because for everything we have studied, there is no involvement of beta-catenin. So Wnt can interact with the receptor and transcriptionally activate certain pathways. I will tell you about the Wnt antagonist. The best studied one is DKK1 or Dickoff-1. It is a competitive inhibitor of Wnt. It can bind the same sites on Wnt, LRP5 or 6, and inhibit the interaction of the ligand. And that then affects the function of the canonical pathway. But again, what I will tell you today is about DKK1 or DKK2, and it all is non-canonical pathway. Next slide, please. Next slide, please. Okay. So the model that we're studying initially was the APC-Min mouse of intestinal tumorigenesis. And the intestinal epithelium is replenished about every 5 or 6 days by proliferation and extensive differentiation of stem cells from the base of the crypt. And as they go up, they differentiate into various cell types and are then shed from the top of the villus. This is a very complex biologic system in which cells are differentiating and proliferating very rapidly. And that's why I think it's so sensitive to the formation of colorectal cancer. It's a very complex biological microenvironment, and there are many cell types. And to probe this more deeply, we isolated individual cell types and did extensive RT-PCR analysis to study the expression of Wnt ligands and Wnt antagonist ligands. And we made the surprising observation that the antagonist DKK1 was expressed at very high levels on regulatory T cells. So we then began studies of DKK1 on lymphocyte populations. And so we made recombinant protein and studied how it affected lymphocyte populations, both in mouse and human. So next slide, please. Next slide, please. This is an alter. I may have to sort of adjust the talk here. Our recombinant protein we studied in vitro, and we found that it was a very potent activator of type 2 immunity. And so the surprise was that we studied, because it was type 2, we studied type 2 immune responses to house dust mite-induced asthma or cutaneous leishmaniasis, major infection of BALB/c mice. And surprisingly, these environmental antigens primarily interacted with platelets. And that was true either in mice or in humans. The platelet activation then activated P-selectin, and then P-selectin interacted with its ligand, P-selectin glycoprotein ligand, and if it was on CD4 cells, there was a physical association of these of platelets, activated platelets, with, in this case, CD4 cells, and there was activation of P38 signaling and CMAF, or SGK1 and GATA3, to produce a type 2 cytokines. And so that was the mechanism of activating a type 2 response. The activated platelets could also interact quite rapidly with neutrophils, and again, there was a physical association, and that then promoted their homing to the target tissues. So this was the mechanism of interactions with lymphocyte populations. Next slide, please. Next slide, please. Ooh. Can you go back to the slides? Oh. Please return to the slides. I don't know how to return to the slides. Hello? I seem to be, I've lost control here. Please return to the slides. Oh. Hello? Please return to the slides. Oh, that's too bad. Please return to the slides.
Yeah, Professor Bothwell, we have fine.
I'll just wait until you bring the slides back. Can you hear me?
So can you please turn on the...
Can I please do what? I will open up this and see if you can see it. Can you see this?
Yes, sir. Yes, sir. We can see that.
Can you see that?
Yes, sir. Yes, sir. We can see your presentation.
Yeah, please go ahead with it. It's okay.
Can you see these slides?
Yes, yes.
Okay, you can
Yeah,
see
yeah.
this.
Good, good.
Oh, okay, good. Excellent. Okay, so, okay, gosh, sorry about all the confusion. So let me know if you see all of this information, and I'll use this cursor to show. You can hear it? Okay. So there are stem cells at the base of the crypt, and they differentiate and replenish the entire intestine in a matter of about 5 days. And so I think I said before that we're interested in the process that regulates transcription or proliferation versus differentiation of these cells. They result in intestinal epithelial cells forming these adenomas, but it's a very complex environment. And so we found that DKK1 was highly elevated in the intestinal epithelium. And so we studied that. To house dust mite-induced asthma. And in the serum of mice, there is a significant induction of serum DKK1 in the serum. If you look in wild-type B6 mice, you see the induction of neutrophils in the bowel fluid or in the lungs, and in the eosinophils, you see significant expression, increase in cellularity. And if you have a mouse strain that is very hypomorphic for DKK1 expression, that is blunted. It's almost shut off. And the same is also true if you have a STAT6 knockout mouse, that it shuts off the response of neutrophils and eosinophils. And if you take cells from mediastinal lymph nodes, you can show that the expression of type 2 cytokines is similarly blunted from mediastinal lymph nodes for IL-4, 5, and 10. I went through this before we stopped, and it just showed what we learned from our 2 models of type 2 immunity. We started by finding the high expression of DKK in regulatory T cells, and we showed that it was critical for the function of regulatory T cells by a standard inflammatory bowel disease model. If you put in effector T cells, you see a depression of body weight. But if you add in regulatory T cells from wild-type mice, they inhibit that weight loss. But if you put in Tregs from the hypomorphic mouse strain, they have no in vivo function. That's quantitated there. But that showed that DKK in Tregs was critical to their function. And if we then took mice cells from those mice and looked at CD4 T cells that are FOXP3 positive, they were also DKK positive, and that was due to a high percentage in vivo of mice express DKK on their regulatory T cells. And if you look at them again by fluorescence microscopy, you find that there is a cell surface form of DKK1 on the regulatory T cells, and that's lacking on the effector T cell populations. So that's due to a hydrophobic patch at the immunoterminus of DKK1. So DKK1 regulates regulatory T cells. but that has broad downstream consequences for myeloid-derived suppressor cells. So here we again looked at our APC-min mice, and they have lots of IL-4 and IL-4 receptor, but when you cross APC-min mice with STAT6 knockouts, there is a dramatic reduction, again, of intestinal polyps. So how does that affect, what's the mechanism? And so we found, then we looked at myeloid-derived suppressor cells in APC-min STAT-deficient mice. There are two normal MDSC populations in the spleens of APC-min mice. These are wild-type mice, and then in APC, there's an intermediate population of MDSCs. And if you have a look at STAT6 mice crossed with APC-min, there is essentially almost an elimination of that intermediate population. Below is just a quantitation of those results. The net result of that is that there is, if you look at CD4 and CD8 cells, there are CD8 cells in the APC mice have very little expression of interferon gamma, but in the knockout mice, there is activation of CD8 T cells in the spleen, and that activates the expression of granzyme and perforin and tunnel staining, and that results in basically the reduction of tumors in these mice. So DKK1 does many things to these mice. Over here on the right is what I've just described, where DKK1 can be activated on platelets and blunt a type 1 immune response. Joan Massaguet has shown that it affects myeloid-derived suppressor cell expansion. He's also shown that it affects the metastatic properties of breast cancer cells to go to either bone or lung. We have results that it blunts an NK response. And then in Kikuchi's lab in Japan, there is the co-expression of DKK1 and its high-affinity ligand, CCAP4 or cytoskeleton activated 4, connect that to the PI3 kinase pathway, which likely stimulates then cell proliferation. So DKK1 is very complicated. It can do many different things in many different microenvironments. So we then began studies about 6 years ago to study similar effects on DKK2 with similar models. So again, down here at the base of the crypts, there are the stem cells, and they go through very complex processes of proliferation and differentiation, resulting in the formation of cells as they go up the crypts. And so we believe that DKK2 is a major regulator of this process. And I'm going to show you how that happens. But I'll return now here to make some analogies with human cells. 80% or more of all human colorectal cancer involves mutation of APC that can generate adenomas. And then there's accumulation of mutations in KRAS, P53, and SMAD4 that can result in carcinomas. But in humans, that pathway of APC to RAS to P53 and SMAD4 takes years. So the APC can give adenomas, and they differentiate to full-blown carcinomas. So what I'm going to tell you is how we can understand this mechanistically by dissecting this in the analysis of organoids. We have all combinations of mutant organoids in these genes. But I'll preface this by showing that this is a public database from cbioportal.com. This shows the log of expression of RNA in various tumors. This is the basal level of expression in normal tissue, and you can see on average there may be roughly a 30-fold, but in fact quite heterogeneous, elevation expression in DKK2 in almost all cancers. So we believe what we're finding is true for certainly cancers we're studying, but it's probably generally relevant. So in APC in mice, Clever's lab showed a while ago that there was increased expression of DKK2 RNA. Another paper in 2009 showed that in human CRC patients, the adenomas had elevations in expression of DKK2, and in full-blown cancer, there was a further increase, 50 to 100-fold increase in CRC cancer. So we've used a standard model of colitis-induced tumors in which you inject a carcinogen, azoxymethane, and then follow that by 3 cycles of an irritant, so sodium dodecyl sulfate, given for 5 days, and then water, and 3 cycles, and then we analyze after all that occurs. And similar to what I just showed you with human data, our model shows a progressive elevation of DKK2 RNA expression, and if you look at the in the crypts, at the end of this experiment, there's elevated DKK in the base of the crypts. Well, we then began by crossing Villain-Cree mice, these are B6 mice, with DKK2 floxed mice, and we showed that there was about a 60 to 70% reduction in the number of colonic tumors as a consequence of this cross. And visually, you can see here in the wild-type mice, there are colonic tumors, and there are far fewer here that are present in the DKK2 knockout mice. We were a little surprised when we got these results. If we compared the polyps of these intestinal tumors, there was almost no difference in terms of protein expression for a proliferation marker. But we were then concerned with what might happen from stem cells, so we crossed a stem cell marker-driven Cree, LGR5 Cree, which is tamoxifen-inducible, with DKK2 floxed mice. And we got virtually the same data when we looked at the wild-type mice versus the knockout mice, about, again, a 70% reduction, and you can visually see the reduction in polyps when you look at the knockout mice. Now we're doing expression studies of RNA, but we've also now incorporated, in this case, normal colonic organoids that are cultured. And we showed that recombinant DKK2, when added to these organoids, causes the transcriptional induction of DKK2. So there's a quite dramatic induction of DKK2 transcription. And so we studied the time course of this response, and it also then induces the stem cell marker, LGR5. And they look more like spheroids or stem cells when they're treated with DKK2 compared to the starting organoids. We've looked at these by RNA, and that has led to actually a fundamental increase in our understanding. But these are the markers then that we could look at by quantitative RT-PCR. So we looked at markers of stemness, proliferation, and differentiation. And you can see here analysis of wild-type normal expression, wild-type tumor, knockout normal, and knockout tumor. So here are 3 different stem cell markers. And if you go from the wild-type tumor to the knockout tumor, there's a reduction in stem cell markers when you knock it out. Therefore, DKK2, when it's present, can promote the stemness. There was basically almost no change in the expression of proliferation markers. And in contrast, differentiation markers had the opposite pattern. That is, the wild-type tumor expression of differentiation markers was increased in the knockout tumor, suggesting that DKK2 inhibited differentiation. So it inhibits differentiation but promotes stemness. Well, all of our RNA-seq data was then plugged into ingenuity pathway analysis, and we got really what was a remarkable finding, that the upstream involvement of hepatocyte nuclear factor 4A was clearly in this pathway, at least as a consequence of IPA analysis. If we looked at the tumors with an antibody to HNF4 alpha 1, the normal colon had expression here at the top of the crypts. As early adenomas formed and tumors formed, that expression was reduced, and at late stages it was almost gone. So we then went back to our Villain-Creed DKK2 knockout mice, and we showed that in fact the wild type tumors expressed almost HNF4 alpha 1, none, whereas was present in the knockouts. At higher magnification, you see very low expression in the tumor and higher expression in the knockouts. Well, how does this happen? Well, we needed to look at organoids to sort this out. So we have a collection of virtually all combinations of APC, KRAS, and P53 mutant organoids. And when we looked at APC mutant organoids, they had very low or almost undetectable levels of APC expression. But when we have AKP mutant organoids, they express significant levels of DKK2. So this just shows a time course of those organoids. And also there was higher expression of LGR5, especially at day 6. So we then adopted a CRISPR strategy to knock out DKK2 in these organoids. And so this is a wild type organoid. This is the AKP organoid. And if we put in a scrambled vector, it has no effect on the level of DKK2 expression. But if we use a knockout for DKK2, you can see this knockout reduces the expression of DKK2. And then in one of the cloned organoid lines, it's also reduced. There's also a reduction on LGR5. And if we look at 3 different independent clones of DKK2 knockouts, and these were all verified by sequence targeted analysis, there was a similar reduction of LGR5. Well, we kind of knew we had made an important observation here when we looked at this is a summary of the RNA expression analysis of these cloned organoid in which there's reduced expression of DKK2, LGR5, no effect on alpha-1 or alpha-7 isoform expression, and an increase in the differentiation marker. If we look at confocal analysis of these organoids, this shows the DAPI stain. There is very little diffuse expression of HNF4 alpha 1 and in the AKP organoids, but in the knockout AKP organoids, it is now returned. Both of these organoids have a tandem duplication tomato fluorescent tag, and so we can see this is allows us to calibrate expression levels. And over here on the right, you can see that AKP, AKPS organoids are now here 3 different clones of DKK2 knockout show increased quantitation expression of HNF4 alpha, and that's quantitated here, showing a significant, as you can see visually, expression of HNF4 alpha. Well, what's the explanation for that? Well, in 2012, a fantastic paper from Chalapa et al. Showed that activated SPARC kinase can cause the phosphorylation of 3 tyrosine residues on HNF4 alpha. And they're identified here in a sequential manner. 4 years later, this same group showed that there was degradation of HNF4 alpha as a consequence of this phosphorylation by the proteasome. And so that's the mechanism by which the HNF4 alpha disappears from cells or these organoids. So we showed in vitro that there was induction of SPARC phosphorylation. And now if we take these organoids, you see the wild-type organoids here are not really staining much at high magnification now of HNF4 alpha, but the knockouts show nuclear expression of HNF4 alpha. If we take these cells and fax them, we show, compared to controls, phospho-SARC expression and also LGR5 expression here in the fax plot. If we compare the wild-type AKP organoids with the knockout organoids, there is a reduction in the mean fluorescence intensity, and there is a reduction of the SARC population, the LGR5 population, when you go from the wild-type to the knockout organoids. Well, we then decided to look whether there was an effect on metastasis. And we've used now a standard assay in which these cloned organoids were injected into mice. And you can see here now that when we inject the organoids into the spleens of wild-type mice, wild-type meaning AKP mice, wild-type mice, there is metastasis to the liver, and you can see here the fluorescence signal by live imaging. And there's no signal really detectable by imaging in the knockout. If you take out these livers, you can see significant expression of metastasized tumors, whereas in the knockout tumor, there are some cells here, but they're very few. So this is quantitation of the fluorescence, and this is then the survival curves. Basically, the knockout, almost all mice survived out to 4 months. By contrast, the ones that had the wild-type AKP organoids showed they were all dead by about 3 months, beginning at about 2 months. So this shows that these changes in the organoids had a dramatic effect on the ability in this particular metastasis assay to actually metastasize to the liver. Those cells that I just described from the wild type and knockout were used to study by single cell RNA-seq analysis. And this is a UMAP plot showing the different cell types that we could detect in those isolated tumors. And this basically confirmed the individual cell types and their expression of, their altered expression of different markers. And due to lack of time, I won't go through this. This is probably hard for you to see on your screen. But we also took these same cells and we did analysis by ATAC-seq to look at epigenetic changes that may be accompanying these organoids that we've been now analyzing. And our hypothesis was that the hepatocyte nuclear factor 4 alpha 1 may be a very important director of the gene expression patterns that we could detect and define by ATAC-seq. And so this again is probably hard to see, but those the data are shown here of the expression differentiation markers, but we can now then look at markers of differentiation and proliferation and stemness at the promoters of these genes for the genes identified here. And we can then correlate here with the little red bars indicate where there is binding shown by ChIP-seq of HNF4 alpha to these promoter regions. And so this has been shown. And then if we look at by ATAC-seq analysis, we can look at the expression or how that affects the targeting of these proteins to the promoters. And so again, this is very consistent with what we would expect now for hepatocyte nuclear factor being a very important regulator of the generation of cells that can become metastatic. So down here, for instance, there's an analysis of the LGR5 promoter and the regions where HNF4 alpha can bind in the in this promoter. And here's another example of BMP4 and the HNF4 sites and how they are affected in the knockout. So our model here is that there are stem cells, and DKK2 protein can autoregulate itself. There's a feed-forward loop, and that can result, when it gets to certain levels, to activate SPARC. And SPARC phosphorylation at 416 then can result in phosphorylation of hepatocyte nuclear factor 4 alpha 1. That's normally a negative regulator of LGR5, but when it's eliminated by proteasomal degradation, there's elevation of LGR5, and that results in cells that are then more prone to be metastatic. And so in a primary CRC tumor, such green cells can then go through the portal vein to the liver and then form metastatic liver colonies. We can then hypothesize that reduction of DKK2 function can protect the host from colon cancer. So we believe that both of these proteins are very important immunoregulators. I've never seen such multifunctional proteins. We don't precisely know how DKK1 may be involved, but I've just shown you the model that we believe that it's involved in prostate cancer, Ewing sarcoma, and many other cancers, probably. We're trying then to do parallel experiments with human patient-derived xenograft models in these various mouse strains. We're working with breast cancer, head and neck, colorectal, and lung primarily at the moment. And I'll summarize some of these results. We have many of these organoids, and some patients we use then tumors and PBMC from the same patients that generated the tumors. A number of them can have reactions with their tumor that inhibits the expansion in vivo. There are interesting effects on the patient lymphocytes in terms of expression of markers on T cells. We're also utilizing expanded tumor-infiltrating lymphocytes, TILs, and in some cases they home to the tumors, and in some cases we observe tumor regression. And our long-term goal is to utilize the patient, see what happens with different therapeutics with responses to their expanded TILs, and to compare how our in vivo models perform. So the work on DKK1 was done largely by Eddie Che. The DKK2 story is by Jae-Hoon Shin, who's a PhD student, now a postdoc. And Steve and Pam are working on the PDX models. We've had some very valuable collaborators from Omar Yilmaz and Jonathan Braverman at MIT, and all the others are Yale colleagues. Most notably, Dan Wu had the DK2 floxed mouse strain that was so valuable in our studies. Okay, sorry for the software confusion. I'd be happy to answer any questions.
So thank you very much, Professor Bothwell, for very stimulating talk. It was really a very good overview of your work. And now it's open for question answer. So there's a question from Abhishek Chattar. And the question is, if in knockout mouse, does DKK2 stem cell marker express more than control? And does it be a prognostic marker for tumor diagnosis?
I think probably DKK1 is a useful marker for tumor diagnosis. We don't know that yet about DKK2. There is some indication that it's actually a little bit of a marker for type 2 diabetes. What was the first question about DKK2 knockout?
This was about in a knockout mouse, does DKK2 stem cell marker express more than the control?
Does it express Wait, the knockout doesn't, I don't quite understand the question. DKK2 knockout doesn't express DKK2.
In knockout mouse, does DKK2 stem cell marker express more than the control?
Express more what? That I don't quite understand. I mean, most of what our analysis in this particular set of experiments, there's tissue-specific knockout, whereas that has no effect on DKK2 expression elsewhere. We've looked at it basically in intestinal epithelial cells and stem cells. And we see no other alterations, although we haven't looked extensively in terms of DKK2 expression.
So there's a question by Shija. So she asked, is the DKK1 and DKK2 has any role in developing drug resistance in cancer?
I think the answer is very likely yes. All I can say is that we can utilize these organoids to study that, and by doing RNA-seq organoids and exposing them to different chemotherapeutic drugs that do result in resistance. We can then, by RNA-seq analysis or other types of analysis, we can identify pathways that are affected by drug resistance. And so I do believe that will likely occur, although that's very preliminary ongoing experiments.
So any more questions?
So I have one question, please.
Yes, please.
The question is actually in case
can you speak loudly?
Question is regarding the natural pathogens which reside inside our gut, and they have been shown that there are a lot of bacteria, pathogenic bacteria like fusobacterial species are there, proteobacterial species are there, which have been shown to induce the genotoxics like lethal distendin toxins or colobactins or galganic metabolites, which have been shown to upregulate the Wnt beta-catenin pathway. So I want to need some suggestions. What will be the feasibility to correlate these metabolites or these molecules secreted by these pathogenic bacteria inside the gut, which upregulate the beta-catenin pathway and also induce the colon cancer?
I think that's a very worthwhile line of investigation. Again, we know most of, in terms of in no case have we seen that when there are alterations in DKK as we have studied them, there is no effects on localization or changes in levels of beta-catenin expression. But that's just, that's the canonical pathway is very much involved in tissue repair and in many ways. So that's just yet another set of biological responses that are affected by these pathways. So that's probably just increasing. Increases in DKK expression are going to increase the inflammation, and that may drive things. But those are pathways that are somewhat independent of what we've been characterizing here that I've described. But I think this is actually studies for the future. This is a very important area.
Yeah. Thank you.
So any more questions? Okay, so there's a question. Okay, all right. So there aren't any questions. We would like to thank Professor Alfred Bothwell for a very nice talk. So let us give him a big applause.
Thank you.
So thank you very much, Dr. Bothwell, to be with us today. And we really enjoyed, and we are really sorry for the interruption in between. But irrespective of that, we really enjoyed your presentation. Thank you very much.
You're welcome. Very pleasant.
Thank you very much. So now, can we request our next speaker? Our next speaker, I can't see him. So, Dr. Asutosh, can you please present your data now? So, your presentation, if you're ready, please come in.
Oh, I can do it now?
Yeah.
Can you hear me?
Yes.
Okay.
So, if you can load your presentation.
Yep.
Try loading, otherwise, we can do from here.
We can do either way. I will try to do it from here. If it doesn't work, then I will go for that route.
Sure, sure. Yeah.
Can you see this?
Yes, very well.
Okay.
Lovely. So here we have now Dr. Asutosh Mangalam, and as already I've given an introduction, but people who might have joined later. So let me tell you that Dr. Mangalam, after doing his PhD from Sanjay Gandhi Postgraduate Institute of Medical Sciences, Lucknow, he moved to US, and after doing some postdocs, then currently he's a faculty assistant professor of pathology at the University of Iowa Healthcare. And today he'll be speaking about the gut feeling, breaking down the role of gut microbiota in autoimmune diseases. So it's a very, very interesting topic these days. Anything you see, it is related to gut. Right, before COVID-19, you pick up any journal and it has got a paper on gut. So we welcome you, Dr. Asutosh Mangalam, and you please start your presentation.
Okay. Thank you, Dr. Agrawala. It is nice, and sorry for our technical. I have to use 3 computers to reach that space. I don't know if you can see in background, we have different computers running, but nonetheless, so first I should get disclosure out that one of the bacteria while working on this work, we have licensed to a company. And right now it is in clinical trial for psoriasis, as well as they are doing a COVID-19 trial, this too. But I'm not going to talk about that. So I know this is a virtual, but I thought I will just let you know. Can you see my cursor? Hello?
No, cursor, we can't see you.
Okay, let me see if I can make a laser pointer. Can you see the pointer now, red color?
Yes.
Okay. So Iowa is in middle of US and northern part of US, and I think IT Roper is northern part of US. I think that's, but I think in between everywhere is joining virtually. So I thought I will give, so University of Iowa is located in Iowa. Before that, I was in Mayo Clinic at Minnesota. So I'm just going south. So we always say that we are the poop lab because that's what we deal with. And the slogan of our lab is that your seat is our treasure. And why we are interested in that, because like Dr. Agrawala said, that gut microbiome, and if you want to study about gut microbiome, although we have microbiome all over our skin, every place of our body, but the maximum concentration is in colon. So that's why we study poop, and they come in different varieties. There are lots of projects going on currently in my lab. And so one of them, which I will talk about, is gut microbiota, phytoestrogen metabolism, and experimental autoimmune encephalomyelitis is a model for multiple sclerosis. there's another project going on. So one thing you have to understand that, and I think I will come to it later, that during evolution, I think we evolve with the bacteria because they help us initially in getting maximum energy from the food. Because lots of plant-based food we cannot digest. So if you look at a herbivorous, they are the biggest animal, like elephant, which is an herbivore, because human is a very efficient machine. So we cannot have a big body to digest that plant food. So that's why we take help of the bacteria. And that's why diet play a very important role in shaping the microbiome, in fact, more than genetics. Then recently we have started a project on gut microbiome, which is fungi in multiple sclerosis. And it is still going on. We are trying to write our first manuscript there. And if I get time, I will talk about this story of IL-17 and how IL-17 during homeostasis can regulate gut microbiota. And then we are also working in a breast cancer, like how phytoestrogen metabolizing gut bacteria can play a role in breast cancer, as well as environmental toxins. So as you can see from all these projects, that gut microbiota is central to everything. So because they are so integrated part of human physiology, anything which can affect gut microbiota can affect us. But coming back to multiple sclerosis, which is the disease model I used, so just give you a little background, because when I was doing my PhD in India, I have never heard about multiple sclerosis. So I'm not sure if you guys know. So basically, it is a disease of central nervous system where you have these myelin sheath, which have this coating, lipid coating. So you can think about these are electrical wire, because that's what they do. They conduct signal. So like in your house, if you have a wiring, you have rubber coating on it so that the signal can go for far distance. The same concept in the body. So what happened that our immune system mistakenly take this as a foreign and they start attacking it. So that's why when these myelin sheath are damaged, so now signal cannot go. So depending on which part of the body the signal was going, so if it was going to your legs, then you have walking difficulty. And if it was your hand, you have numbness and tingling, vision problem. And there are four main kind of multiple sclerosis disease. So one is that some individual who get attacked, but they never develop full-blown disease. So on the y-axis is the disability and x-axis is the time. So in both time and disability, with increasing time, there is increased disability. And RRMS is the type of disease you see in majority of MS patients, because with every relapse, there is increased disability. And then the certain patient have progressive. So some of these RRMS progress to secondary progressive MS, and some patient from start, they never have any stable period because there is disease keep going up. So why I am interested in what gut microbiota have to do with the multiple sclerosis. So most of the autoimmune diseases, if you understand, is that there is a genetic factor and there is an environmental factor. So previously I have worked on lots of genetic factor, and we have used HLA class 2 transgenic mice, which have 1501 as well as other HLA molecule to show that how HLA molecule might play a role in the disease. But in a twin, you have only 30% chance of getting disease. So it means that 2/3 of effect is environment. So what we did is that, so among environmental factor, there are lots of things, but if you look at the disease incidence of MS, where it is found more, and pink or reddish or that kind of color is the higher number of MS cases. So if you see about these cases of MS, and if you try to overlap, if you look at the developed country map, industrialized nation, you see that all these blue color match with where most incidence of multiple sclerosis is happening. So one is that it is there near to equator, near to polar region, but other way you can look about that these countries are the where industrial revolution happened. So what industrial revolution has to do with the incidence of MS or other autoimmune diseases? So if you look I will go back to the history, is that when Industrial Revolution happened, what happened was that lots of people start moving to cities because of this factory setting up cotton. I think that was the biggest thing at that time. So London was epicenter of that. But when you start moving to city was not geared up to take so many people, as well as the herd, like animals they have with them, like horses or cows or whatever, sheep or goats. So then city has to build up a sewage system. And as you can know that every city, even now, if you take any city, any part of the world, it is on a river, because it is a good mode of transportation as well as it is a good source of water. So you take, so this sewage system, they put it in the river. But after they put it, then there was a disease, a lot of disease epidemic. But at that time, nobody knew that what, because all the scientists in that time, they thought that these diseases are airborne. So that way, John Snow, he's considered a father of epidemiology. So that's also so how data keeping is very important in science, because only thing he did is that he went and looked at, because at that time in London, there was two water sources. One is upstream, the Thames, and one downstream. So he looked at that, where is the water source and where people are dying. So I'm not sure if you can look at these black dots in. So there are lots of people died here. So these are the places where there was a downstream water source. And that's how he knew that it is some disease which is coming from a water. But, in a science field, when you're trying to say something new which not everybody accepts, so he used some clever method to say that how these diseases are waterborne without saying water word in there. So if you have interest, you can go in history and look at it. But what happened is that, so once they realized that diseases are coming from water, somebody got this sodium hypochlorite and they chlorinated the water. So once they chlorinated the water, the disease of these epidemic infectious disease went down. So if you look at the typhoid fever, it went down considerably. But at the same time, if you look at the autoimmune diseases, multiple sclerosis, Crohn's disease, type 1 disease, or asthma, which our previous speaker was speaking about, all these diseases just picked up. So although in science there is correlation, is not causation, but we know that there is a maximum density of bacteria in the gut. And if you put acidified water, although it is keeping the water pure, but it might be also killing. But in 1900, nobody knew about it. So what had happened is that, so this gut microbiota which we have, it start disappearing or it start changing. So this was one of the example I gave you, but there are lots of other examples. So like in India, now we see more increased cases of MS. And what might be happening that urban living, away from dirt, because you are not living with this all bacterial community which is there, because you have to remember that not all bacteria are bad. And then lots of chemicals which is coming in the diet, because after World War II, lots of these chemicals which were used for non-perishable food for military started coming up for general population, increased use of antibiotic. So all these, what they did is that they altered the relationship we had with our gut microbiota. So based on all this data, we asked the question, simple question, that do MS patients have a distinct gut microbiota? So as you know that everybody say there's a gut feeling. So that's what the title of my talk is. And therefore yesterday when I was talking to Agrawal, he also said that he had a feeling that gut feeling is going to be a good talk. So, but means we always say about the gut feeling, but what is it really? There is a relation between our gut and a brain disease here. And so just before going into data, some of you might understand, because big data is a thing. So in big data, the concept is that there are alpha diversity and there is a beta diversity. Alpha diversity is simply like if you take what is the difference in diversity in individual about the bacteria. So let's say you take two individuals who have the same number of bacteria, but if you look at kind of bacteria they have, this blue individual have 5 different kind of bacteria. But if you look at this red guy, he has only two different kind of bacteria, B and C. So although the number of bacteria is same in both, the diversity is less in one individual. At the same time, what is beta diversity is that how different is the bacterial makeup of one individual versus other. So this is what is alpha diversity. Like I said, there is a difference in diversity, and the beta diversity is the bacterial composition between two different, so intergroup differences. So this is data which is already published, so I'm not going to go into too much detail about it. So each square or diamond or circle are an individual patient's bacterial makeup. So let's say there are 100 bacteria in one subject, which is patient or healthy control. So this blue is healthy control. So if there are 100, so these are different kind of bacteria which make up. So this blue dot at this point and this green dot here, they are different from each other because their bacterial makeup is very different. So this is just showing that MS patient have a distinct gut microbiota as compared to healthy control. So this was a beta diversity. But if you now go deeper, so that was a 10,000 foot view. So if you go to 1,000 foot view, like try to find out what bacteria are different. So we did Permanova analysis, statistical test to see that. So this is just on your left side, you will see parabacteria as something which is decreased in. So each individual data is here. And you can see that mean is here in the MS patient. But if you look at the control, these blue or whatever color you see is that they are higher and mean is higher. The same way, one of the bacteria, Blottia, is more abundant in MS patient as a control. And then these are the makeup of MS patient versus control for all the bacteria. So I'm not going to go into detail. The point of this slide just is to show that there were lots of bacteria at different level, which was different between MS patient and control. What I wanted to bring in now is that, so in our study, we found that these are the three bacteria which was lacking in MS patient. But in all these big data analysis, you always have to be a little careful because what if it is some data which is not right, like you found it because it is a big data and you didn't do the right statistical test, or there was some error. But what we got confident with is that all the MS study which has come so far have shown what are other bacteria we have found. So we were the first one to show that these detailed study in MS patient that they are different than healthy individual. So Privitella is decreased. Jungi et al., which came this is a Harvard group study which came 1 week because we did the study at the same time. And then this study came 1 week of our study is that Privitella and Parabacter are lacking in there. Miyake she published just before us from Japan. Then Stanford group also shows that this is decreased, and the German group, what they did is that they took bacteria from a twin which have MS and the twin which don't have MS, and they put it in a germ-free mice. So only when you have a healthy twin in the germ-free mice, you see more Adlercrusia. And when you treat MS patient, then there is an increase in Privitella, at least 2 studies have shown. So point of this study is that what we found has been also shown by others, just validating our finding. And this is just an altimetric score, which I never knew, but I was told by journal that this is a score which tell you that how your paper fare as compared to in the science at this time as compared to other paper. So within 3 years of its publication, so this is the top 100 read articles. So one thing I wanted to point out that at that time when I completed the study, I was moving to Iowa for a job and I didn't had time to do more study. I sent it to big journal, but then I said I wanted to get more audience. So I sent it to scientific report and it got published without any review within 28 days. And so the point is that if you have a good study and you believe in it, journal doesn't matter as long as you get the good audience. It is still among the 99 percentile paper track article in the journal as well as all the journals published. So the question becomes is that we found all these, but what does it mean? So you remember that Prevotella is the bacteria, we found that increase. So then we wanted to ask that, can it modulate disease in animal model? Because, Koch hypothesis is that if you think that this is bacteria is important, can you provide it or can you remove it to prove that this bacteria has to do what you are saying to do? So Salas is a postdoc who is from BHU. He is a very hardworking guy. So he did some of these experiments. So so EA is a model for multiple sclerosis, which some of you might know because it is a model. So like if you're working in India, I think Leishmania is a model for Th1 and Th2. But in all Western country, if you want to study Th1, Th17 or Th2, this is a model because Th1 or Th17 are involved in the disease. So anytime you get a molecule, you test in a model because it is a very easy model where you immunize with a CNS antigen with adjuvant, and then you see a disease where you get a paralytic disease. So like I said, in MS patient, they get weakness in the limbs. So similarly, when you immunize these mice, they stall with limb weakness and they develop full-blown paralysis. So in mice, when we don't give any bacteria, so we gave either medium or we gave Prevotella histocola, another species of Prevotella, E. coli, which was a mouse bacteria, and C. sputagina, which is a human-controlled bacteria, because we wanted to make sure with the proper control, not any bacteria can do it. And when we give Prevotella, we see that there was this very significant effect on disease. So as you can see that mouse had a very less severe disease incidence was also low. Another strain of Prevotella had some effect, but it was not as significant as Prevotella histocola. And other ones has no effect, which just showed that not any bacteria can cause this effect. So if you take a mouse E. coli, which is a commensal, it doesn't have any effect. And this is just a cumulative EA score in each group, which again shows that Prevotella histocola had more significant effect cumulative EA score. So with this data, we did, and I'm not going to show, we published this data, and at the same time, we wanted to differentiate this from probiotic. Because like Dr. Agrawala mentioned, that gut bacteria are important, so lots of companies are coming with probiotic. But remember, this is my understanding is that you, like Koch hypothesis, you try to treat a disease with what is lacking, not giving something which is good otherwise. So that's why we coined this term bacteria as a drug when I was in Mayo. And if you follow a magazine Wired, which is like a techie magazine, so it comes with a jargon in the future to watch for. So they pick up our term drug, which is bacteria as a drug, which you can use as a treatment. So don't be surprised in the future when you see drug instead of probiotic. Then we did some other study, which I'm not going to show data because we wanted to see that can we combine Prevotella with a drug Copaxone, which is an MS drug, or Betaseron. And we found that although there was no synergistic effect, but Prevotella was as effective as Copaxone or beta interferon, which is the first-line drug in multiple sclerosis. So it just shows that maybe they are mediating their effect with the same pathway. So that's why we don't see synergistic effect. We don't have gone into much detail, but one thing we know that Prevotella histocola is as effective. So not only we have seen that it works in multiple sclerosis, but it works in a model of RA, as well as recently Mayo Group has checked it in a diabetes model. So it works in a diabetes model. But right now it is in clinical trial for dermatitis or psoriasis because those are the most easy model to try any drug on. So most probably in the future, we are trying to do a clinical trial in multiple sclerosis. So this is about, we found a bacteria which was depleted in MS patient. We saw in animal model that this is decreased. But now the $1 million question is that how does it do that? So like I mentioned in the start that when I was asking these questions and people keep asking is that how you know that bacteria is working. So I went into literature and tried to find out that what control gut bacteria. So if you look carefully, diet is that when you look at the world map, and I was showing you lots of things, one thing which changed everywhere is the diet. And with industrialization, processed food is eaten more in the Western country as compared to India or China or other places. So why diet is important? If you're not convinced, if you look at the human evolution, when we are born, we are almost sterile. And initially we are on the mother milk. So our dietary requirements are different. So we have a different bacteria, only proteobacteria is there. But as our diet changes, the composition changes. So it again shows that diet play a very important role in the makeup of the bacteria. In fact, they have Jeff Gordon group have done some elegant study where they have shown that if you transfer a microbiome from an obese individual into germ-free mice, it become a germ-free. It become an obese. However, if you change the diet to a plant-based diet, these mouse now don't become obese. So
you're left with 5 minutes.
Oh, okay. So what I wanted to do in this 5 minutes is that just show you that what we found is that how diet can modulate the immune system. So these are the 3 bacteria which metabolite phytoestrogen. So we wanted to ask that can they play a role in a phytoestrogen metabolism? So Sam did most of the work with a graduate student, which is completing up. So just to give you a little background, that phytoestrogen are chemicals which are present in plant, and we don't have ability to metabolize them. Only certain bacteria, including the one we found depleted in MS patient, can metabolize them into equal and introlactone. And so we asked this simple question that can phytoestrogen modulate the disease, and is it bacteria required? So what we did is that we put mice on a phytoestrogen-free diet, a diet with isoflavone, which is a phytoestrogen found in soy food. And as you can see that if you don't have phytoestrogen, they develop severe disease, but we introduce isoflavone, they develop a mild disease. And it is not true for one model. We can repeat it in a HLA transgenic model, as well as we can do in a relapsing remitting model, which mimic human MS2, that once mice on a phytoestrogen diet, they develop a milder disease. So then the next question is that once we prove it, the next question is that how does the bacteria play a role? So when we put mice on a diet, the one thing we see that the bacteria which come up is Adlercrucia and Parabacterial. And if you remember that these are the same bacteria which shows depletion in MS patient. So it means that our diet data just mimic human MS. So then we ask the question is that can we use these bacteria when we deplete all the bacteria, give the diet and give this bacteria to see that can the bacteria require these phytoestrogen to mimic their effect, and phytoestrogen require bacteria for the effect. So if you see this, the black dot, when we have bacteria but no diet, you don't see effect. But when you introduce diet, then only you see effect. And if either one of them are not present, like here, if you have a media only, then you are diet only, you don't see the effect. So next question we ask is that, so diet, we know that diet is important, bacteria is important. So how about what immune cells are important? So I'm not going to show lots of data because lack of time. So what we found is that when you put on an isoflavone, which is phytoestrogen diet, you have increased number of regulatory T cell and decrease in FOXP3 ROR gamma T cell. So one thing you have to know is that ROR gamma T expressing FOXP3 cell acquire inflammatory phenotype. So that's how Treg can become inflammatory cells. So we found that was decreased and FOXP3 total expression was increased. And then when we look for CD4 and CD8, so although we were trying to find a CD4 population, but we found that to our surprise that CD8 population was increased in isoflavone mice. So then the question was that we ask a simple question, if CD8 is important, so if we take a CD8 knockout mice, then if you put on a diet, then the suppressive effect should be gone. And that's what exactly happened, that if you take a CD8 knockout mice, which are these, so when there is CD8 is present, you see disease suppression. But when you knock out CD8, you see now that the effect is gone. So we just confirm that absence of CD8 T cell abrogate this phytoestrogen mediated disease protection. So to summarize all the data is that I have shown you that MS patients have gut dysbiosis and especially loss of phytoestrogen metabolizing bacteria. If we put animal on phytoestrogen phytoestrogen diet, they develop milder disease, and the bacteria are required for this disease because when we provide these bacteria, then you see the effect. And CD8 T cell and FOXP3 T cell are playing an important role. And now I'm just going to stop here for the lack of time. And thank you, Dr. Agrawala. And I would just like to acknowledge slide, which is in the end, that lots of lab members who did all this study and the funding support. And I will take any question if you have now.
Thank you very much, Dr. Ashutosh, for a very nice presentation and the way you have correlated very well the influence of phytoestrogen on the cellular and molecular mechanism of the host immune system. So it was a wonderful talk. So it's open for question answers now. So there are a lot of questions for you, and I don't know whether I can take them on because of shortage of time. So this is Amit Rawat. He asked that the microbiota would vary according the state of the disease in the same patient and also by immunomodulation. Would it not be a major variable?
So I'm glad you asked the question because Mayo study we did at one time point. So that's why to answer the same question, we are doing a longitudinal studies where we are following a patient up to 5 years so that we can answer those questions that it is different states. So is it that microbiota change before a disease relapse happen? And at the same time, we are recruiting patients which are treatment naive as well as on different disease modulating agent. And we have some preliminary data that, yes, that affect, but even with, so we see a different kind of effect. When you have on antibody treatment, we see a different kind of bacteria going up. If you take an antifungal drug, which is Tacfidera, you see a different bacterial composition. So it seems that most of the drug act through different modulation or different gut microbiota.
The question from Ritika Kumari, that in the era of COVID-19 pandemic, having each time warm water will decrease gut microbiota, resulting into bad health condition and as well as making us more susceptible for COVID-19 infection.
So I didn't get the first, but I will try to answer it that, so virus has a major impact on gut microbiota. And I think Dr. Agrawala mentioned that he did some study and he found a similar thing. We haven't done anything. We are trying to do some in flu model. But you have to understand that in this COVID-19, I think most of the patients come with the diarrhea symptoms, if I'm not wrong. And one of the bacteria which is depleted is Prevotella in animal model when they use germ-free pig. So in fact, we are trying to use Prevotella as a drug in a patient which have early stage of COVID. So we are doing a clinical trial in Mayo and Iowa. I think still working on the specific of that is that gut microbiota might help in immunomodulation to a better because viruses is wiping out the good bacteria. So maybe if we can change that environment and it will boost the immune system and we can patient can recover faster. I hope that answers the question.
Rahul Gava Guru, he asked a question. Antibiotics are really readily exploited in today's world, and especially in our country, you can just go and get through counter. Does this extensive use of antibiotics harm our gut biota? Does antibiotic-oriented diarrhea have anything to do with gut biota?
I think so antibiotic definitely have effect. So when I give it, here people say that, oh, we take lots of antibiotic. I said, you guys don't take. In India, we can go buy antibiotic without prescription. We don't ask anyone. I think lots of time I had flu and took an antibiotic. So I think antibiotic, what it do is that change your microbiota completely. So it can be a good or bad thing depending on how, because every individual, because you have to think about it, your gut microbiota is like a soccer match. So when you're in a soccer match, you are playing and your aim is to make a goal. But you make a goal only once in a while unless it is a very bad team or a very bad immune system. The same way your gut microbiota is very resilient, it can take care of lots of these things. So coming back to gut microbiota, so some of the MS patient, they say that they got a disease after they had a diarrhea. So one of the patient called me from UK, he was a big author, that he went to a trip in Asia and he had a big diarrhea and he took antibiotic because of that. And when he came back, he had his first symptom. So yes, these changes can affect you, but I think MS and other diseases are chronic diseases. So most of the time it is that slowly you are going from this symbiotic system to a system where you are increasing the inflammatory response. And I don't have time to go through, but I think what is happening is that slowly you are changing your healthy gut microbiota to a microbiota which can induce more pro-inflammatory response. Leaky gut might be one of the things that it will make your gut more leaky, which will allow lots of endotoxin from your gut to go into system, which can induce systemic inflammation, and that can induce lots of different diseases.
So we'll take this last question. There are many questions, probably 25, 30 questions, but it will be a last question because of the constraint of time. This is Yasmin Khan. Does phytoestrogen has any effect on apoptosis pathway?
I think I will not go into that because I don't work on a cancer field. So I have seen some data from individuals who directly seen effect of some phytoestrogen. So what I do is that how phytoestrogen is metabolized by gut bacteria like isoflavone into a qual and introlactone, and how it interacts with the epithelial cells and immune cells. So I haven't done apoptosis work, but I know in cancer field, that's what people thought that that's how it is acting. But I haven't done personally any work and I cannot comment on that.
So thank you very much, Dr. Sutosh Mangalam, for a nice, very educative talk. And I hope in future we'll try to interact with each other, and we'll like to invite you at our institute at IIT Ropar. Probably then you can spend more time with our students. So let us give him a big applause for wonderful talk. Thank you very much.
Thank you.
So our next speaker is ready, Dr. Ramarao. So Dr. Ramarao is a professor at Emory Vaccine Center, and his main interest is on vaccine against HIV. And today, Dr. Ramarao will be talking about HIV vaccine synergy between tissue-resident T cells and antibodies. So we welcome you, Dr. Ramarao.
Hi, can you all hear me okay?
How are you?
Good.
So we are excited to hear you.
Wonderful. And this is, and first of all, I'd like to thank you for initiating something like this, especially to connect with people in India. This is a great opportunity.
2,700 people were registered for this conference, and 1,700, about 1,200 they are watching your lectures.
Oh, wonderful. Right now, I can only see 20 people. I was thinking, maybe I'm just looking at the speakers.
No, what we did, we created 3 links. One link exclusively for the speaker, so that the bandwidth should not be disturbed.
Wonderful.
So we are getting clarity. We can see you very clearly.
Wonderful.
We can hear you very clearly.
Wonderful. And then I assume that the majority of the audience are young, budding scientists, and those are the people I would really like to reach.
Sure.
So I consider this as a great opportunity. I should share my screen, right? Okay. Can you see this?
Your presentation is not visible. Can you please reload? Hi, Rama, can you reload your presentation?
Sorry, can you hear me?
Yeah, we can see you.
Sorry, I think that I'm using the Firefox and then that has a problem. And now I switched to Google Chrome. I'm going to try that now. Yes. So can you see okay?
Good. Yeah. We can
Sorry
see
about
you.
that. I guess they want to monopolize certain browsers. Okay, so in the interest of time that we have about 20 minutes for the talk and 10 minutes for questions, I try to keep my talk simple. So I know that we are all in the middle of a major pandemic of our lifetime, but I do like to remind everybody that HIV/AIDS is still a major problem in the US and across the world, especially in the sub-Saharan Africa. There are more than 36 million people are living with HIV, and we need a vaccine for sure. So about half of these infected people are able to access antiretroviral therapy, but that's not enough. And then the art has a lot of issues long term when people take them, and just not the cost itself. So I want to quickly talk about the early events during HIV transmission. So there is a lot of virus particles that the host gets exposed to, and then few of them gain access to the mucosa, the local mucosal tissue, and in fact, few CD4 T cells or dendritic cells. So we call this as a small founder population. And then there is a low level of virus replication locally, and then these infected cells or the virus gains access to the local draining lymph nodes, and then once the replication occurs there, and then the virus becomes systemic. So there is a maximal opportunity for vaccines to prevent infection during this early stage of infection, which lasts for about 3 to 4 days at the site of virus exposure. So basically the vaccine should induce immune responses at a local mucosal tissue to give highest chance of protection. So if you look at the protective immunity at the portal of viral entry in the mucosa, so here you have the virus and infects the CD4 T cells. If you have neutralizing antibodies that can block the step of infection, so you can get protection. And generating neutralizing antibodies against HIV has not been feasible because of the vast diversity of the virus envelope glycoprotein, and it's also heavily glycosylated. So these antibodies can't really reach the protein portion of the envelope. If the infection occurs and there are other functions of the antibody can still help, like phagocytosis that can clear these infected cells. And the phagocytosis can also work on the virus itself. It doesn't have to be necessarily virus-infected cells. And then you have complement-mediated lysis. And then you have antibody-dependent cellular cytotoxicity. So this is called ADCC. So these are all called as cell-targeting antibody effector functions. So where the constant region of the antibody plays a role, it recruits various effector cells like NK cells or macrophages to do these jobs. Whereas the neutralization is driven by the variable region of the antibody that provides specificity. In addition to the antibody, if you have tissue-resident cytotoxic T cells, they can also clear these infected cells. So a vaccine that can do both all these functions, like inducing neutralizing antibody, inducing antibody with effector functions, as well as tissue-resident CTLs, can do a great job. So I thought I'll keep this introduction to short and focus more on the data. And today what I'm going to present has recently been published and is done as part of this consortium that we call as the Emory Consortium for Innovative AIDS Research. This is headed by Dr. Eric Hunter and myself, and with some collaborations. This consortium has many players, and it has two foci. One focus is on the mechanisms of vaccine protection, and the other focus is on the mechanisms of reservoir eradication. As part of this mechanisms of vaccine protection, we have various groups that are developing various things. One group led by Bali Pulendran, Rafi Ahmad, Jane Crotty, and Max Cooper, looking at durable humoral immunity. David Massafirst is looking at tissue-resident memory CD8 T cells, and my lab is looking at balanced humoral and cellular immunity. And then as part of the mechanisms of reservoir eradication, we have Guido Silvestri, David Margolis, and Charudi, Mirko Payardini. They are developing approaches. We also have a role in there. And then supported by various cores, the primate and live animal imaging core led by myself and Phil Santangelo from Georgia Tech. Antibody function and viral diversity by Cindy Redin, mucosal antibody responses by Pam Kozlowski, plasmablast response and B-cell repertoire by Jens Rammer, and single-cell analysis and transcriptomics by Steve Bosinger and Paul Johnson. So this is, you can consider this as like my acknowledgement slide because it is a collective effort of doing these studies. And then we recently published this paper in Nature Medicine where we show that T-cell inducing vaccine durably prevents mucosal infection even with lower neutralizing antibody titers. So it's basically driven by Bali's lab, my lab, Massafirst's lab, and Eric Hunter and Cindy Redin's lab. So I will review this data now for the rest of the presentation. So we conducted an NHP study using rhesus macaques where we immunized two groups of rhesus macaques with 15 animals each. One group received the BG505 SOCIP envelope protein adjuvanted with 3MO52. So 3MO52 is a TLR78 agonist developed by 3M. And this is encapsulated in nanoparticles. I want to just go back one slide and acknowledge the first authors of this paper, the Prabhu Arunachalam from Bali's lab, Taishina Charles from Cindy's lab, Vineet Jog from David Massap's lab, and Satish Bolimpalli from my lab. And then various other people played an important role. I'll be acknowledging them as I'm going forward. Coming back to the study design, so this is the protein that came from John Moore and Ayavi. This is a clinical product that they're actually testing in people right now. We gave 4 protein immunizations with this adjuvant PMO52 with about 8 to longer 24 weeks gap. This is the schedule here. And then a second group received the same protein immunizations, but in addition received heterologous viral vectors. So that's what this HVV stands for. So what I mean by that is that priming with VSV expressing SIV gag only, there is no envelope, and followed by 8 weeks later with the vaccinia virus expressing SIV gag, followed by AD5 expressing SIV gag. So this is to drive a very strong T-cell response, primarily to gag. So there is no T-cell response here to any other proteins of SIV or HIV. Just to see whether there is a synergy between antibody response and T-cell response. And this protocol was basically developed by David Massapust quite some time back in the mouse, and then we started doing the NHP work. The other important feature of this vaccine regimen is that these viral vectors were delivered intravenously. So I'll come back to this, the importance of the route later on. But it was designed that way so that immune responses can be generated in the tissues very effectively. Then a control group of 15 animals received just the adjuvant only, 3MO52, right before these animals were challenged. So about 4 weeks after the last immunization, these animals were exposed to 10 weekly challenges, intravaginal challenges to BG505 shield. This is a chimera of SIV and HIV, where the backbone is basically the SIV with HIV envelope, BG505. So this is a tier 2 shield. And the goal of this study was what is the level of neutralizing antibody response we can generate against this HIV envelope using this protein and adjuvant combination, how durable this response is, and then if you combine this with an antibody-inducing vaccine with a T-cell inducing vaccine, will there be any benefit for protection? So here is the binding antibody responses in serum. This is protein. The legend is not here. The red is the protein-only group, and the blue is the vector plus protein group. Apologies for that. So you can see that after second protein immunization, there is almost a 3-log boost of the binding antibody. This is IgG and IgA. And then that was, it is not shown is that these antibodies contract quite a bit, but then they get boosted again following the third immunization, and then they contract again some, and then they get boosted by the fourth immunization. So this is 2 weeks before the challenge. Whereas if you look at the IgA titers, they continuously, the peaks of these responses continuously increase almost up to the fourth immunization. So they follow different kinetics. This is the binding antibody responses, again IgG and IgA in vaginal secretions. Just like the serum IgG, you see pretty rapid increase in the IgG responses by second protein, and then they kind of boosted to the same levels, the subsequent proteins. And the IgA also follows the same pattern as in the serum. But more importantly, when you look at neutralization titers against the BG505, for those that normally do not work with HIV vaccines and don't think about these vaccines, inducing neutralization titers against a tier 2 envelope, even the autologous neutralization, has not been that easy until recently when John Moore's lab and others started defining stabilized trimeric immunogens. So if you see what you're seeing here after second protein immunization, after first protein immunization, there was no detectable neutralizing activity. These data were generated by David Montgomery and Celia Lagrange, and then these were further confirmed by Cindy's lab at Emory. So you can see that these appear at low levels. Once the third protein is given, they are significantly boosted in many of these animals. If I remind you, and then the fourth protein, boosted further or kept them at the same peak as the third protein. Whereas if you look at the IgG titers, that between the second protein and third protein, you don't see much difference. And then again, the same is true for the fourth protein. Whereas if you look at the neutralizing antibody titers, you see a big difference. So this is because of the maturation of the antibody. But importantly, although there are some animals seem to show a little bit higher neutralizing antibody titers in the vector plus protein group, overall they are quite comparable. So meaning that having the viral vectors added to the protein immunization, they don't significantly impact the neutralizing antibody response to the envelope. And this is the same data overlaid for the both groups using as a median for each group. So this is the independent analysis that is done by Cindy Dardine's lab, and this is the data we actually used in the paper. And then they correlate quite well between the two labs, because we were doing this just to make sure that things that we are measuring are quite reproducible. Here is the CD8 T cell response on the left, and the CD4 T cell response to the GAG only. Basically, for the envelope, there is very little T cell response because protein immunizations only induce mainly CD4 T cells. They don't induce much of the CD8 T cells. So what you can see with each immunization, you see a robust increase in the CD8 T cell responses in gamma positive cells, going up to over 10% of the total CD8 T cells. This is the power of this heterologous prime boost approach. And then we also saw a pretty nice boost. In fact, going up to 10% of the CD4 T cell responses. Interferon gamma positive CD4 T cell responses is quite high for any vaccination regimen. So this basically induced both CD4 and CD8 T cell responses. And a bit about virus challenge. We did an intravaginal challenge with BG505 SHIV. This came from George Shaw. This is at a pretty high dose, 3.5 times 10 to the 8 virions, 1 ml volume, and then given every week. We had to give this very high dose so that we can get about 30% of the unvaccinated controls get infected at each exposure. So here is the protection data. So the black is the control. You can see after first exposure, about 30% of the unvaccinated controls get infected. They drop off like that as we increase the challenges. By 10 challenges, we infected 90% of the animals. There are only a couple of these animals that showed some resistance. So this is a normal problem with intravaginal challenges. We found that those 2 or 3 animals that took longer challenges, these were actually significantly older animals. So when we remove those old animals from the, we didn't have that old animals in the vaccinated groups. They were there only in the controls. When you remove them, then you actually see all animals get infected by 3 challenges. So importantly, that both protein-only group as well as the vector plus protein group showed pretty nice protection from infection. And although the vector plus protein look like is slightly better, that is 70% of the vaccinated animals were completely protected, whereas opposed to probably 55% in the protein only, they are not statistically significant. However, and then we also saw good viral control in these animals. So these are both vaccinated animals showed close to 2 log lower virus at the peak. This is only the infected animals. We are not, obviously we can't look at uninfected animals because they don't have any virus. So the vaccine also showed better viral control in those infected animals. So when we try to correlate between the protection and the neutralization titer, what we saw was that there was a direct correlation between the number of challenges to infection, which is measured as protection, versus the neutralization titers in the protein-only group. However, in the vector plus protein group, we didn't see that. That association was not there. When you look at all animals, the association still was there, but the R value was weaker than the protein-only group, 0.8 versus 0.6. So then Prabhu looked at the cutoff for the neutralization titer for complete protection in the protein-only group, and then it was determined about a titer of 300. Seems to correlate with complete protection in the protein-only group. It's a little bit over 300, and this is in line with another previous study where came from Scripps Chavi, where they used the interrectal challenges and found a similar titer to correlate with complete protection. However, when we looked at these protection based on this 300 cutoff, so if among the protected animals, there were some, so all animals that if they have greater than 300, they are protected. But in some animals, although the titer was less than 300, they were still protected. And it turned out, and on all the infected animals, that if they are less than 300, they all had less than 300. So in this middle group, which was interesting, that they're all protected despite having lower neutralizing antibody titer. And then it so happened that they all belong to this blue group, that is the vector plus protein group. So then when we looked at the acquisition of infection based on the neutralizing antibody titer, in both groups, you can't see the blue group here because it's right behind the red group. If they have titer greater than 300, they're all completely protected. When they have titers less than 300, then you can see that there is an advantage for protection in the vector plus protein group compared to the protein-only group. So this clearly showed that if while combining the viral vector immunization with protein-only immunization has a significant advantage of protection. So it looks like I'm running slow, so I'll quickly, and then the correlation was with the...
You are left with 5 minutes.
Yeah, I realized that. Sorry about that. So there was a strong correlation with vaginal IgG, and then there was also a strong correlation with the antibody effector function in these mucosal tissues, which is shown here. This is the ADCVI activity. And the more important thing is that when we took those animals that were protected from the first round and then exposed them again almost 6 months later with the second round of 6 challenges from both groups, we took 6 animals from each group, and then we still see protection in the vector plus protein group, but not in the protein only group. So showing that again, this protection is durable with the viral vector vaccination. So to conclude this, that BG5FH SOSIP immunizations in PMO52 nanoparticle adjuvant, they induce a strong autologous tier 2 neutralizing antibody response. Animals with neutralization titer above 300 are completely protected. Vector plus protein vaccination enhances protection even in low neutralizing antibody titer animals, and protection in the vector plus protein group is durable. We are doing follow-up experiments where we are asking the question, is intravenous vaccination important to generate tissue-resident T cells and protection? Can we replace this viral vector regimen with clinically approved vaccines? And mechanistically, how antibodies and TRM work together? So we have an experiment set up to answer these questions. So I'll stop there.
So thank you very much, Dr. Ramarao, for presenting very interesting and intriguing data. So I would like to ask first question before I... okay, so the question is, how long this protection persisted, and were these it generated long-lasting memory T cells?
Yeah, so if you see the time of the second set of challenges from the time of last vaccination, it was about 1.5 years. So I would say that at least for 1 year we can see this protection. And the way these memory cells work is that there is a significant contraction of the immune responses. They happens within the first 3 months after vaccination. And then after that, the contraction of the immune response is going to be very, very low. So if anytime when you see protection is about 6 months after final vaccination, it's very likely to last for a long time.
Okay, now it's open for question answers.
What was the question? I missed it.
Okay, so there are many questions for you, and now I'm going to read for you one by one.
Yeah.
So this is Abhishek Chakkar, and he says that he would like to ask Dr. Ramarao that vaccination strategy that can both CD8 and T cell and antibody responses confer enhanced protection against infection. How does your late study help in it?
Sorry, how does this study help in it?
His question is that whether your vaccination can generate both CD8 T cell and antibody response to confer protection.
Right, that's what we showed, right? That the viral vectors are designed to generate the T cell response, and the vaccination is designed to generate the antibody response. And when you have a combination, it can actually give you protection even in the absence of presence of low levels of neutralizing antibody.
Okay, so second question is from Lakshana Mahajan. And how does mode of attaining immunity vary if antigen given internationally or through blood or through some other route?
Yeah, so the immunization route really can clearly change the quality and the magnitude and the location of the immune responses. So, for example, we have shown that if you give intramuscular immunizations, you can get strong, using our DNA and via vaccination, you can get pretty strong T-cell response, whereas if you give intradermal immunizations, you can actually get better antibody responses. And we are now trying to figure out what is the influence that the route of immunization has on using T-cell responses in the tissues. So that's the experiment is ongoing, and we think it may be severely impacted by the route of immunization.
So there is another question from probably you answered that. This is on memory. How long memory response is generated by Ebarami Sitaraman? How efficient is memory response triggered by your vaccine? So probably you answered that. There is another question by Anuj Bandral. What is the main constraint in the development of HIV vaccine in addition to high mutation rate of virus?
Well, the other main problem is because the virus integrates into the host cell. So once there is an infection because of this virus integration, then viral reservoirs are a major issue. So that's why the antiretroviral drugs can only suppress the virus, but they can't clear these viral reservoirs. As a result, for a treatment approach, you do need to have approaches where you do T-cell function as well as eliminate the viral reservoirs.
So there's a question from Rabia Hamid. What is the potential of lectins in being used against HIV code glycoprotein?
So there are a lot of other cells in our body express lectins, right? So I don't think just using lectin as a way to inhibit. So this is something in a prevention setting, you can't be injecting lectin into somebody all the time because they also have certain half-life. So it's not going to work as a vaccine approach or as a prevention approach.
Okay, so because due to constraint of time, we won't take any more questions. There are many questions people have. And now let us thank Dr. Ramarao for giving this wonderful lecture. Give him a big hand, big clapping. So thank you very much, Dr. Ramarao. So our next speaker and the last speaker of today's session is Professor Imtiaz Ali Khan. So Professor Imtiaz Ali Khan is a collaborator in this project, and we together got this project from SPARC. So he's a professor of microbiology at the George Washington University, and he moved to US after completing his PhD from Institute of Medical Sciences, BHU, Varanasi, and his area of interest is autoimmunity and immunological responses. And today he's going to talk about CD8 positive T cell immunity against cellular parasitic infection. So we welcome you, Professor Imtiaz. You please unmute your mic and share your presentation.
Okay, can you hear me now?
Yes. So you can share your presentation.
Okay, just a second.
So today morning, Professor Peter Doherty, he released our monograph.
So,
which is also part of this project.
So can you see, can you
share your presentation?
Yeah, that's what I'm trying to do. Share. Can you share? You people have my presentation. Can you share from there, or is it not possible? Because I
okay, we'll share. We'll share from here.
Okay, because I'm having problems.
Sure. Share. So just in a moment, we are sharing the presentation.
Just a second. So where will I do that? Will it? Is it asking me somewhere to share the presentation? Is there a? So how can it give you the permission? From iCloud? Just a second, I'm having some glitches in my computer. You want me to share it from Gmail? Is it okay? Okay, okay. Yeah, now we can see it. Yeah. Hello? Yeah, so shall I get started? Yeah. So my topic of my short presentation today is as first of all, I want to thank Dr. Agriwala for giving the opportunity and putting all the effort for organizing this workshop. So my lab has been working on the CD8 T-cell immunity against parasitic infections, especially Toxoplasma gondii. And first of all, as a parasitologist, whenever you talk about any parasite, you have to be acquainted with the life cycle of this parasite. Can I have the next slide? Okay. As far as the life cycle of the parasite goes, Toxoplasma is the obligate intracellular parasite. It always needs a cell to survive. It cannot survive extracellularly. And it has two hosts. Cat is the definitive host because the sexual life cycle of the parasite takes place in the cat gut. The male and female gametes are formed. They fuse together to form oocysts. And oocysts are liberated when the cat defecates. And when it is doing that, the oocyst has two sporocysts. And a cat or animal or a human being dealing with the soil, which is contaminated with the cat litter, ingest these oocysts, especially if they don't maintain a good hygiene.
And your voice is quite low. Can you please increase it or come near mic?
Shall I speak loudly? Normally, my people have complained that I speak too loudly. This is the first time I'm hearing that.
Yes, quite low.
It's good. How about now?
Needs to be better. Just come near mic.
Okay. Okay. How about now?
Yeah, better.
Okay. So when the sporocyst, the oocyst with the containing two sporocysts are ingested by the humans or animals, it can be sheep, it can be pigs, it can be birds, and under the acidic environment of the gut, the oocyst wall is basically dissolved, and the sporocysts liberate the sporozoites, and sporozoites convert into tachyzoites. And these tachyzoites are very promiscuous as far as the infecting the cell is concerned. They can infect every nucleated cell of the body, especially excepting RBC, which doesn't have a nucleus. And it travels from these cells. It could be there that people have shown whether it could be macrophages or it could be dendritic cells take up, and these first disseminate to this is a stage of the acute infection. They disseminate to the nearby organs like mesenteric lymph nodes, liver, and so on, and ultimately they have some kind of predilection towards the brain. And it is not known clearly, but it is believed that under some immunological pressure, these tachyzoites undergo a stage transformation and they become bradyzoites. And these bradyzoites can be found in brain or in skeletal muscles. And their bradyzoites are covered with a cyst wall, and these cysts remain dormant for as long as the person's immune system is intact. Human beings get infected by eating meat which has Toxoplasma cysts, or they can get infected by handling the cat litter box, which is contaminated with the oocysts of the parasites. Now, as long as your immune system is fine, you can basically not even know that you were infected. It will pass as some kind of flu, slight enlargement of lymph nodes, and so on and so forth, and you will not know you are infected. The seroprevalence is very high in Europe, especially France, where it was believed that 70% of the population has been exposed to Toxoplasma gondii infection. Though I'm not very much familiar with French cuisine, but they say that and that they eat a lot of, the part of their cuisine is the undercooked meat. In this country, United States, at least 30 to 40% people have been exposed to Toxoplasma gondii and are seropositive for Toxoplasma, the parasite. As I said that in the immunocompetent people, is not a problem. Earlier, it was a problem for, or it still is, for pregnant women. If woman is infected in the first trimester of the pregnancy, it leads into abortion. If woman is infected during the second or third trimester of her pregnancy, the fetus will be born, but it will have a lot of malformations like hydrocephalus, chorioretinitis, so on and so forth. So the parasite gained a lot of importance due to HIV epidemic. Before this HEART therapy, in one of the studies conducted by Jack Remington's group at Stanford, they had shown that 30% of HIV-infected population suffers from the disease due to reactivation of latent Toxoplasma infection. And similarly, it's a problem for transplantation patients or anybody who is under immunosuppressive drugs. It results in the reactivation of latent infection. The brideozoites releases the parasite, they get converted into tachyzoids, and they spread throughout the body. And most of the time, the person dies of toxoplasmic encephalitis. And this was one of the common problems in the HIV-infected population before the HEART therapy started. Next slide, please. As far as immune response to Toxoplasma gondii infection is concerned, it induces a strong innate as well as adaptive response. Before the adaptive immunity sets in, the immunity is maintained by natural killer, even neutrophils, dendritic cells, and natural killer cells. Natural killer cells produce a lot of interferon gamma, and basically dendritic cells produce a lot of IL-12, and Toxoplasma infection we have seen in the animal model. I don't remember of any pathogen which induces such a strong IL-12 production. We find that when we measure serum IL-12, it is a matter of few hours you get a spurt in IL-12 production, and it is believed to be by what Alan Shear's group has shown by dendritic cells. CD8 alpha positive dendritic cells produce this IL-12, and then it has also been shown by Eric Dinkers that IL-12 is also produced by the neutrophils. So IL-12 induces NK cells to produce a lot of interferon gamma. Interferon gamma is very critical for protection against Toxoplasma gondii infection. My very favorite quote is, if you have interferon gamma knockout mice, you don't have to give them Toxoplasma tachyzoids. They can smell Toxoplasma and they die. That is how critical interferon gamma is for the protective immunity during acute infection. The innate immunity, after it wanes out, this adaptive immunity sets in. Because there is so much IL-12, there's a polarized Th1 response, which is manifested by CD4 T cells producing a lot of interferon gamma. And interferon gamma, along with TNF alpha, is responsible for production of nitric oxide by the macrophages or dendritic cells, and that will stop the multiplication of the parasite. But ultimately, during the chronic infection, CD4 T cells provide a good helper response for generation of CD8 T cells. And CD8 T cells are the major effector cells, especially during the chronic infection. And cytotoxic response, they are also good interferon gamma producers, and they exhibit antigen-specific cytotoxicity, which is mediated by perforin. I'm sorry about that. And for the long-term infection, for the chronic infection, that is contained by cytotoxic CD8 T cells. Now the question is, why do HIV-infected population, which have downregulated CD4 T cell response, get reactivation of toxoplasmosis? The reactivation of toxoplasmosis takes place during the advanced phase of HIV infection, when not only CD4 T cell response is down, but CD8 T cell response is also compromised. That is the point when the HIV infected population undergo reactivation of toxoplasmosis and develop toxoplasmic encephalitis. Next slide. Mouse is a good excellent model for studying Toxoplasma gondii infection or immune response to Toxoplasma gondii infection. And a lot of their symptomology can be compared to the humans. What we see, we take a C57 black 6 mice. If you give them very low dose of type 2 moderately virulent strain ME49 parasite intragastrically, what happens? Most of the death due to acute infection takes place at day 7 to 9 if some animal has to die, because 10 cysts is a very low dose infection. But what we have observed, as it passes and we think the mice have recovered, they start to die. And if you, in this, as data, the slide which I'm presenting, by day 125, which is a lot of time after infection, all of these mice start to die. So what we did by real-time PCR, we measured, because if the animal is dying of Toxoplasma infection, it should have a lot of tachyzoids, which is the acute infection, which is the representative of acute infection or representative of reactivation of infection. And we use stage-specific antigens and determine the level of these antigens by RT-PCR. And what we find in case of the INDO-2, which is the representative of tachyzoids, which is a acute stage, it goes up at week 3. Then it comes down at the week 5, meaning that they have converted to brachiozoids. But it starts again to go up at week 7 and week 10. Similarly, INDO-1, when the cyst, which is the brachiozoid specific antigen, representative of chronic infection, at week 3, you start to get brachiozoids, but they start to go down. And they go down by week 7 to week 10 infection. That means during the long-term infection, there are not only brachiozoids indicating that infection is being controlled and the chronicity is being maintained. Some of these brachiozoids probably are converting into tachyzoids, resulting in acute infection. Next slide, please. We have in number of papers which came from my laboratory and others also, there is a consensus that CD8 T cells are critical for controlling the chronic infection. And if you look at week 5 post-infection and look at CD8 T cell response, at week 5 point infection, it is 11.4 in the spleen, and week 7 infection, it goes down to 6.2. I mean to say interfunctionality of CD8 T cells, measuring interferon gamma and measuring the granzyme B. Measuring the polyfunctionality of CD8 T cells, the interferon gamma and cytotoxicity, and both of them, which is represented by granzyme B, both of them, these functions are important for controlling chronic Toxoplasma gondii infection. At the polyfunctionality at week 7 goes down as compared to week 5. So it happens in spleen and it also happens in the brain. Brain, it goes drastically down. And in those days, now a lot of people are talking about the exhaustion or the dysfunctionality of CD4 or CD8 T cells during chronic infection, which was first reported by Brian Barber in Rafi Ahmed's lab, and during the LCMV chronic strain of infection, there is upregulation of PD-1, major inhibitory receptors, and now these what is called as checkpoint inhibitors. There are a bunch of checkpoint inhibitors, PD-1, LAG3, TIM3, CTLA4, CD160, so on and so forth. And this anti-PD-1 therapy or in combinatorial therapy with anti-CTLA4 and so on is being used in more so in cancer patients, not in HIV as yet, where also it is important. What we saw at this PD-1 expression, the major inhibitory molecule, in case of both spleen and brain, is increased at week 7 to week 10 post-infection. That is a time where we see the downregulation of functionality of CD8 T cells. As you can see from this slide, it almost doubles from week 5 to week 7 to week 10. So there is increased expression of PD-1 in these mice during the chronic phase of infection. In the late chronic phase of infection, I should say. Next slide, please. Well, if PD-1 is responsible, can the anti-PD-L1 blockade reverse this dysfunctionality of CD8 T cells? And what we have in the slide, what is marked as C, when you give anti-PD-L1 administered, because you have to give it, and there's also the dosage has to be worked out, because you cannot give anti-PD-L1 continuously, because otherwise they will die of inflammation and autoimmunity. So we had to work out the schedule. Raj Badra was a graduate student in my lab. He had to spend some time working which is the best possible PD-1, anti-PD-1 dose, and how you can give it without causing acute inflammation in the mice. And what we found, the mice which were treated with anti-PD-L1, their functionality of CD8 T cells increases in spleen as well as the brain, both cytotoxicity as well as the interferon gamma production. So anti-PD-L1 treatment does restore to a certain extent the functionality of CD8 T cells in these chronically infected mice. So what happens to die? As if you remember the earlier slide I showed, they slowly die, like it's shown in when you give the control antibody. These animals, they survive for the acute infection, but during late chronic infection, they start to die, they succumb to infection. But if you give anti-PD-L1, only we had 10 mice in this group, and this was repeated about 4 to 5 times, we find that only one of the 10 mice died. That it had a major effect on the survival of these mice if you block the PD-1, PD-L1 interaction. And is it due to CD8 T cells? Yes, it is due to CD8 T cells. The other slide, if you look at the right-hand side, shows that if you give anti-PD-L1 antibody, all of the mice survive, but you deplete and give anti-PD-L1 plus anti-CD8, all of the mice come to the infection. So what they showed us that CD8 T cells are important for Toxoplasma gondii in the chronic infection, but during the late chronic phase, there's upregulation of PD-1, and this PD-1 causes CD8 T cell dysfunctionality, and they cannot control they cannot maintain the chronicity of the parasitic infection, and they die of reactivation of latent infection. Next slide, please. So this is the case for CD8 T cells. What about CD4 T cells? Our previous work had shown that CD4 T cells that CD8 T cell response against Toxoplasma gondii can be induced without CD4 T cells. If you have a CD4 knockout mice, you can get a normal CD8 T cell response during the early phases. But after 3-4 months, this CD8 T cell response wanes out. It cannot be. CD4 T cells are important for CD4 T cell is not needed for the generation of CD8 T cell, but it is very important for the maintenance of CD8 T cell memory. So we looked at the CD4 what happens to inhibitory, what happens to CD4 T cells? We are reported to see there is CD8 T cell exhaustion, but what about the CD4 T cells? And one of the important molecules we looked again is PD-1. And because we wanted to look at the antigen-specific cells, but problem with the Toxoplasma gondii is that there are very few tetramers which are known for which are CD4 directed. Only one tetramer is AS15, and that is very small percentage of CD4 T cells. So if we started to look at the tetramer positive cells, we will not be representing the total CD4 T cell epitopes. So what we used as one of the study which was used in John Harty's lab is looking at the surrogate markers. And surrogate markers for CD8 T cells are CD4 T cells we used was CD11A and CD49D. And CD49D, CD11A. And when we looked at we also did a control experiment where we took these AS15 positive, the only known tetramer for for CD4 T cells and CD57 black cell mice, and we found that 100% of these tetramer positive cells were CD19, CD11 high, and CD49 high. So we consider these surrogate positive cells as antigen-specific cells, and we looked at the spleen and the brain at week 3, week 5, and week 7 post-infection. As you can see the slide on the left-hand side, we see the increased expression of PD-1 in case of the spleen as well as the brain at week 7 post-infection.
You're left with 5 minutes.
5 more minutes? Okay. When you look at the, I will try to wrap it up, then other part I will not, I had another part also. And when you look at the functionality in the spleen as well as in the brain, you can see the PD-1 low cells have a good bifunctionality, but PD-1 intermediate has increased, because that's another story of why PD-1 intermediate are better functional, because PD-1 is also an activation marker. But when you go to PD-1 high stage, there is much decreased functionality in PD-1 high expressing CD4 T cell. It is the case is similar in the spleen as well as in the brain. Okay, next slide, please. Now we looked at the transcription factors, because there was a report that by John Verry's group that there is a BLIMP1 is a transcription factor for the exhausted CD8 T cell. And what we found, BLIMP1 is a B lymphocyte-induced maturation protein. It is encoded by PRDM1G. Increased expression of BLIMP1 has been reported to cause upregulation of inhibitory receptors on CD8 T cells. And BLIMP1 is antagonistic to BCL6. If BCL6 is up, BLIMP1 goes down. And when the BLIMP1 is go up, BCL6 goes down, which is an anti-apoptotic molecule. And what we found, as far as the transcription factors is concerned, the Tibet goes in the antigen-specific cells, Tibet goes down, EOMs is increased, which happens in the exhausted CD8 T cell, and BLIMP1 is tremendously increased at week 7 post-infection, and BCL6 goes down at week 7 post-infection. So there is a increased expression of BLIMP1, which is a transcription factor responsible for the upregulation of inhibitory receptors. Next slide, please. The next study which we did was that, what if you transfer non-exhausted CD4 T cells, CD4 T cell isolated at 2 weeks post-infection in the mice which were infected 5 to 6 weeks earlier? Can these fresh CD4 T cells, exhausted CD4 T cells, non-exhausted CD4 T cells, basically rescue the CD8 T cell exhaustion? And that's what we did. We infected the mice with CD45 congenic mice system we used, and we gave them these cells isolated at 2 weeks post-infection. I gave them to 5 week post the mice which were infected 5 weeks earlier, and looked at the CD4 and CD8 T cell response at week 7 post-infection. And if you can see, PD-1 levels in those mice which were given CD8 T cells was decreased, BLIMP1 was decreased, but interestingly, CD4 T cells had no effect. If you give non-exhausted CD4 T cells to the mice, the CD4 T cell is not rescued from the exhaustion, but it is the CD8 T cells that are rescued. And it is shown in this slide. If you look at the functionality in terms of interferon gamma as well as granzyme B and TNF-alpha production, the bifunctionality of CD8 T cells, where you give toxoplasma gondii specific CD4 T cells, increased. While if you give non-specific, it doesn't have much effect. And obviously, when there is no transfer, it is basically the functionality is down. Same is case when we measure TNF-alpha and interferon gamma. It is very much down, but it does slightly go up when you give TNF-alpha first in flow cytometry. It is very difficult to measure, but it slightly goes up. Next slide, please. Now, what we did, we took conditional knockout BLIMP1 mice, PRD1 conditional knockout mice. In these mice, when you give tamoxifen, the BLIMP1 gene is sliced, and you have a control on which time you can select, when you can basically knock off the BLIMP1. We took the CD4 knockout mice which were treated with injected with the PRD1 conditional knockout mice, the CD4 T cells which don't have BLIP1. And after 5 weeks post-infection, when we give tamoxifen for 1 week, we did the assays. And what we found, when you give the BLIP1, tamoxifen, to these mice, the interferon, their functionality increases. As you see interferon gamma and the granzyme B, it goes from 1.27 to 3.26. And interferon gamma and granzyme, in case of, this is it is not written here, it is spleen and it is brain. And in the brain also, the bifunctionality increases. And if you look at the parasite load in these mice, now look at the Toxoplasma gondii. When you knock out the BLIP1, the parasite load goes sufficiently down in blood. While these are the wild type, these are CD4 knockouts, and these are conditional knockouts with tamoxifen and without tamoxifen. And all in 3 organs, blood, brain, and the spleen, you have that when you knock out BLIP1, the level of parastemia goes down. And as when you do the immunofluorescence staining, you have wild type mice which have tachyzoids. CD4 knockout mice has more tachyzoids. And when there is no treatment, there's no tachyzoids. But if you give tamoxifen, knock out the BLIP, the number of tachyzoids decreases substantially. Next slide, please. So I will go to the conclusion part that CD8 T cells are exhausted during chronic toxoplasmosis. CD8 T cell exhaustion is primarily mediated by increased PD-1 expression, though I can qualify my statement. We have found out there are other receptors, especially the TIM3 is very important, and LAG3 to a certain extent also important. And I don't have time to talk about that. And it can be reversed by PD-1, L1 blockade. CD4 T cells are critical for maintenance of CD8 functionality. CD4 T cells are exhausted during chronic toxoplasma infection. Adaptive transfer of non-exhausted CD4 T cells reverses CD8 T cell dysfunction and prevent reactivation of chronic toxoplasmosis. And CD4 exhaustion is attributed to increase in BLIP1 expression. Thank you very much. And I'll be more than happy to answer any question you have.
So thank you very much, Professor Khan, for a very exciting talk. And now this is open for question answers. And let me take first opportunity to ask you a question. That since interferon gamma is quite crucial for protection against Toxoplasma gondii, so in such cases, if you inject interferon gamma in these animals, does interferon gamma at the same intensity can protect mice from infection from Toxoplasma gondii?
Yeah, if you inject interferon gamma, obviously it will go. But what people have shown in the chronic infections, it is the cytotoxicity of T cells is important also. Interferon gamma. For acute infection, it is just interferon gamma. They're like if you have a peripheral knockout mice, they will survive fine acute infection, but in the chronic infection, they succumb to chronic infection.
And second question is, what is the mechanism by which CD8 T cells, they were rescued by CD4 T cells?
That's a good. That's what we are showing in the. We have that data. That was the next part. It is IL-21 mostly. And another molecule which we are showing is that, which is in a recent study and on which we have an NIH grant now, is that upregulation of co-stimulatory molecules on CD4 T cells. And one co-stimulatory molecule we are targeting is 4-1BB. 4-1BB basically gets upregulated in CD4 T cells. It increases the IL-21 production, and IL-21 restores the cytotoxicity of CD8 T cells.
Okay. So it's open for questions. Okay, so let me read the questions on